Hello.
Good morning, everyone. You are requested to put your phones on silent. Ladies and gentlemen, welcome to day 2 of the first international conference on sustainable innovations in drug development. I hope yesterday was a scientifically fulfilling day. The lectures and poster sessions have been informative and motivating to you. I hope you had a great stay at IU campus and enjoyed the cultural program and dinner last evening. We will now begin with session 6. For this, I would like to invite Dr. Vinay Kumar Srivastava and Professor Sivapriya Kirubakaram.
Good morning to all of you. The voice is very low. I think you did not have good breakfast. So I think the Vishnu Maya will take care of good lunch at least. So anyway, the tone has already been set yesterday to next level, where we have witnessed various eminent speakers who have presented their work before this august audience, and followed by the student interactive session, which otherwise did not happen, like student interactive, because we hardly had one question from a student. So among our panelists, one really played the role of a student and made excellent questions, and again has set a high standard of this conference, and followed by wonderful cultural evening by a student of Indrashil University, showcasing our cultural heritage, especially of Gujarat. So continuing with the same standard, we are moving forward next step by starting the day 2 session with the plenary lecture by one of the eminent speaker, Professor Tapas K. Kundu, who is Professor, Department of Chemistry, IIT Indore. Sorry. So I think need the correction, because here it is mentioned Professor, Department of Chemistry. Okay. So presently he is holding the position at JNCASR Bangalore and CSIR Central Drug Research Institute, Lucknow. You were earlier. Okay. So I think it will come forward in his introduction. So let me introduce him. Professor, sorry.
No, this is fine.
Anyway, so Professor Kundu earned his PhD from Indian Institute of Science, Bangalore in 1995, receiving the Best Thesis Award. He pursued postdoctoral research and visiting foreign research associate at the National Institute of Genetics, Japan, followed by a postdoctoral fellowship at Rockefeller University, USA, from 1996 till 1999. Joining JNCSR in 1999, he has made significant contributions to gene expression regulations, epigenetics, and therapeutics, elucidating transcription regulations mechanisms and developing novel diagnostic and therapeutics. With numerous publications in international journals, his research led to several granted and pending patents, including commercially viable research reagents. A fellow of 4 major Indian academics, he has served as an editorial board member of the Journal of Biological Chemistry from 2011 to 2016 and currently hold editorial roles in Journal of Biochemistry, Oxford University Press, eLife, and Subcellular Biochemistry. As a director CSIR, CDRI, Lucknow, he spearheaded academic and drug development programs leading to multiple drug candidates to phase 1 clinical trials, licensing them for further development, and completing a phase 3 clinical trial for anti-COVID-19 therapy awaiting DCGI approval. He also played a key role in establishing the Asian Forum for Chromatin and Chromosome Biology and the Chemical Biology Society of India. He has earned selected recognition, which I'll just mention here. Shanti Sharoop Bhatnagar Prize from CSIR in 2005, Fellow of National Academy of Science, India 2005, Fellow of Indian Academy of Science 2008, the National Academy of Science, India Reliance Industry Platinum Jubilee Award 2008, Fellow of Indian National Science Academy 2009, J.C. Bose National Fellowship from SERB DST 2010, the G.D. Birla Award for Scientific Research 2011, the Ranbaxy Research Award in 2011 in the field of medical sciences, basic research, and Indian innovation. Award 2012 given by Merck Millipore 2012, Shri Om Prakash Bhasin Award for the year 2019 in the field of health and medical sciences by Shri Om Prakash Bhasin Foundation, New Delhi. The National Academy Medical Sciences 2021, ICBS 2022 Global Lectureship Award by International Chemical Biology Society, Sir M. Vishwasaraya Senior Scientist State Award for the year 2021 by the Government of Karnataka Department of Electronics, ITBT and SNT instituted by Karnataka State Council for Science and Technology 2023. So I think his award itself speaks about the depth of knowledge he holds with him. And we all are very lucky to have him here and share all his experience. His title of the talk is Epigenetic Regulation of Neurological Disorder, Role of Lysine Acetyltransferase P30CBP. So welcome you, sir, on the dais.
Thanks a lot. सही लगेगा वो उस तरह से अब बंद करो पहले इसको ठीक है चलेगा कोई बात नहीं हाँ So thank you very much for, you've gone through my whole CV. First of all, I thank the organizer to give me this opportunity to come here, which was due since a long time. I'm very closely associated with Cadira. The reason is, during my directorship, Dr. Rajiv Modi was the RC chair of my institute. And many of you work with him, so you know how you have to be always alert. And he made a significant contribution as a RC chair. And I often used to come to CADLab to discuss with him many things. And visit to this university was due for me quite some time, because another beloved person and whom I respect a lot in the field of drug discovery, Dr. Yadav, was also is one of the beginners of this institution. Thank you very much. And I must say, you know, Dr. Vishi, she is so responsive for everything, so prompt. Vishnu Maya. Okay. I'm going to talk to you. If you can remove this top part, will be very nice. The epigenetic regulation of neurological disorders. What is epigenetics? We all have born from a single cell, okay? And this cell inside your mother has been differentiated and given many types of cells to us: neural cell, kidney cell, blood cells, and many, many things. But if you take the DNA sequence of these cells, and these cells all are equal, but the cellular behavior looking to these cells are completely different. So DNA sequence is not the ultimate. There is something above the DNA sequence, and that is the science called epigenetics. So your DNA is not your destiny. If this is the DNA, you have a dictionary, this is a non-functional. Unless this DNA is properly leveled, properly modified, it has no role to play. So if you look at these two mice, they have identical ATGC sequence, but they look very different. Why? Because the DNA is differentially modified by simple chemical modification, methylation of the bases. Now, what is the basis of it? Our genes are organized in a beads and a string structure, which makes an individual chromatin territories inside the cell. And these territory folds unfolds to express the gene by any kind of signal. What is the signal? Signal could be your happiness, signal could be your sorrow, signal could be your food, signal could be your anxiety, signal could be your infection. And that will lead to the different kinds of modification like DNA methylation, DNA associated protein like histone modification, chemical modifications, and small RNAs, which will help to fold and unfold the genome, make it functional or non-functional, as and when it is required. So that is the science called epigenetics. And you can easily imagine these are highly metabolically regulated. Methyl group has to come from S-adenosine methionine. Acetyl group has to come from acetyl-CoA. And that metabolism is very, very important. So the song of the life will be only sang properly if it is properly modified. It's properly organized. Otherwise, it is not. So if you look at a simple modification called acetylation, so lysine residue will be acetylated in the histone molecule associated with your DNA. So when it is acetylated, genome will be open. When it is deacetylated by another eraser enzyme, it will be open. So, and there are a large number of enzymes are involved in that. I will talk to one group of enzyme P300 CBP downstairs. So something called acetylation homeostasis. This homeostasis, opening or closing of the genome, is very, very important. Acetylation will be terribly down when you are depressed. Depression has a direct connection with acetylations. And so as many, many disorders, neurological disorder and cancer, you will see this homeostasis is lost. And that is why this is an important target for therapy. I will show you today one of the example of it. And so I will get into you in a neurological disorder. There are two types of neurological disorder. One could be developmental disorder, one could be neurodegenerative disorders. So in this, the factor will be genetic factor. This factor will be also epigenetic factor. Genetic factor modification is very, very difficult. But today we know a lot the biochemistry of the epigenetics, and thereby you can have many kinds of modulators. Some of the inhibitors, and our discovery is the activator. This will alter the epigenetic cassette. So fundamentally, you can play around with epigenetic modifications. I will show you a couple of examples today. So suppose you are all listening me. So before activity, when you are not listening, now you are listening. Your memory getting consolidated. Your calcium signal is on. Your genes are recording it. So for that, the acetylation has to happen, the genes in your brain. If this is stopped or disturbed, your memory will not work, neither the short-term memory nor the long-term memory. So you imagine how it is connected. As you grow old, the acetylation level in the brain will dramatically alter. Methylation will increase. So therefore, the acetyltransferase, lysine acetyltransferase, SCAT, activity is very important. So if it is a hyperacetylated, there are many diseases will cause. Hypoacetylated, so as that. Therefore, you need to balance this. And if you can balance, there is the hope of the therapy. How did we enter into it? So we were screening the P300 acetyltransferase modulators. The first hit was the natural compound called anacardic acid. This is basically aspirin. Okay. Now, this is a strong inhibitor of acetyltransferase activity as a whole. Many people, after we discovered, they are using it. You can see in the catalog. But we were not very happy with this simple molecule, especially my postdoc. So what he started modifying this molecule. He added a flutamide along with this. The surprise of the surprise was this no longer remained an inhibitor, it became an activator. So this is the this immediately catch eye of many people. We got the US patent. Many companies start selling this compound. You can go and see in the catalog. But we were not happy with it. We are not happy with it. The reason is this was not a very good cell permeable molecule. But chance favored the prepared mind. Next door of mine, Professor Ishwar Murthy's group was working in carbon nanosphere derived from glucose. And these carbon nanosphere, when my student Selvi looked into it, she saw this is a cell fluorescent molecule. And this cell fluorescent molecule, when we put into the cell, what we find that it is a cell permeable as well, this nanosphere. So this is 1 hour, 3 hours, and 24 hours. Chandrayaan land on the moon. So it was a miracle that glucose-derived carbon nanosphere having many properties of glucose, not carbon nanoparticle, don't mistake with this, can enter into the cell. When we put this along with this, it could enter into the cell, this molecule, which was not cell permeable very much, and start inducing the acetylation. The most surprising thing was waiting for us is that when you inject this to the mice, subperitoneal injection, we find that it could cross the blood-brain barrier and it could actually activate the acetylation in the mice brain. This means a lot. Many people before us was trying to inhibit the deacetylase and induce the acetylation. So now with this device, we could activate the acetylation in the brain. And what we find that we made this molecule little civilized. We cut the tail and we made it little smaller. We gave the name TTK21, second generation molecule. And this TTK21, we covalently conjugate with the carbon nanosphere. And then we see that it is inducing the acetylation in the cell. And when you inject to the mice, we find that in the day 3, there are lots of fluorescent particles in the brain, and that could induce the acetylation in the brain, especially the P300 mediated acetylation. H3 acetylation, H2B acetylations, these are the histones. Now, where in the brain is this acetylation is happening? To cut a long story short, what we find that this is a very important area of the brain, which is hippocampus. It's very important for your memory consolidations, memory formation, storage. And there is a place called dentate gyrus, and we find that this acetylation is hugely upregulated in the brain upon injection in the dentate gyrus area. So dentate gyrus area is a very important area. And this is the area where the neurogenesis happens. Neurogenesis is connected to many, many phenomena, including depression. So we ask the question, is this induction is inducing the neurogenesis? So what we find that indeed, when you put CSP alone, that's the carbon nanosphere, this is the dendritogenesis. And when you put CSP TTK21, you find a huge dendritogenesis or axonal growth has happened. this was something very important. If this is the case, what is happening in the memory? We did a memory test. This is the Morris water bath test. So you put the mice here. First day it takes long time to go to this hidden stage. Second day it doesn't take long, little less time. Third day it takes very little time to find out the stage. That's the memory. To cut a long story short, what we find that if we treat with the CSP TTK21, induce the acetylation, then even after 16 days, mice is remembering where is the stage. Clear-cut induction of the long-term memory in the normal mice. Well, so we try to see which gene it is. We find out that indeed this is the NeuroD1 and BDNF which gets upregulated, where the acetylation in the promoter is up. Then the question we raise that can we use this for something else? First thing we look into the tau mice. Tau mice is the Alzheimer's mice. And if you see that after the activity, Morris water bath training, if we look at to the dendritic spine in the dendrite, so you see this is the mushroom, okay? These mushrooms will be created upon memory. Now, if you take the tau mice, you see mushrooms are hardly there. But if the mice is treated with the drug, you see the mushrooms are normal upon learning. Clearly indicates that in the tau mice, where the memory is lost, the memory is supposed to come back. Let's look into it. So initially we tried to see by RNA-seq analysis, and we could see that there are 81 very important genes are upregulated because of that. Well, few genes are downregulated. Most important thing, when you do the chip-seq analysis along the gene, we find that very interesting H2B tetraacetylation peaks have come up. And these are actually indication of the activity of super enhancers, the enhancer which induce the gene expression. But what happened to the memory? What we find, this is the normal memory of the normal mice, this is the tau mice, and this is the tau mice treated with the drug. So memory is completely back. This was something, a big news for us. It got a place for a big paper as well. Now the question comes is that if you induce the acetylation, in the tau mice, you get the memory completely back, and many cognitive behavior we tested that is come back. So this was very encouraging for us. So these mice, what we had taken, these mice are young mice, 8 months old. So what happened when the disease is progressed? Mice is 12 months. That was a question. So we took two different age of mice, the Alzheimer's mice as well as the normal old mice. I don't have a time for it, but I just want to tell you one surprise. When you look at to the old Alzheimer's mice, we find there is a dramatic decrease in the cholesterol biosynthesis pathway. So mind the sentence. Cholesterol is terribly down in the old Alzheimer's mice, but not in the old mice. So this we look into more carefully the pathway, and we find indeed that is the case. But is it the case for the mice model? Is it true for human? We managed to get the Chinese brain bank data. And there we find that exactly the mevalonate pathway is down there. So this creates a new discovery that as we grow older, as the Alzheimer's patients grow older, there is a down regulation of cholesterol biosynthesis pathway. So can something this CSP and this TTK21 do for this? We treated the mice, and what we find that this cholesterol biosynthetic pathway, what we find that, first of all, this changes the, mice gets the behavioral, much better. It start doing many behavioral tests. One of them, we just do the recognition memory and all. And then we look at to the pathway, we find that many cholesterol bath pathway genes are upregulated now. So we could reverse back it. How is it possible? So we look at to the each gene and we see the RNA-seq analysis of that, and we find indeed that has happened. So how do we explain so far? The work is not done completely, but for a challenging thing, we had to publish the paper. So what we think in tauopathy, cholesterol metabolism is down. Because of that, the acetyl-CoA is high, and that inside the mice, these extra-acetyl-CoA go to the glutamatogenic genes, and that gets upregulated, and thereby the cognition impairment happens. But if we treat with CSP-TTK21, we can induce the cholesterol biosynthetic genes, and these genes doesn't get up, so cognitive improvement happens. Well, there are many questions can be asked there, how it is happening, but this work is very, very significant therapeutically. Why? Because FDA, they prescribe that the Alzheimer's patients can be given statin. Okay. Now, statin is a lipophilic molecule. It can go to the brain and it inhibits the cholesterol biosynthesis pathway. So our data clearly shows that the lipophilic statins cannot be given to the Alzheimer's patients, because statin will go and it will inhibit the cholesterol biosynthesis pathway. And that's the whole reason we had a very tough time to publish this work. Many statin drug companies, they didn't want that. And Nature Neuroscience didn't want to take us. Well, so this work we are continuing. I will slightly switch over it, a side finding. When we are doing this CSP-TTK21 mediated activation of axonal growth, then the dendritogenesis is happening, neurogenesis is upregulated. One of my colleagues from Imperial College London, who works in spinal injury, spinal injury, wherever that happens, the rest of the part is numb, gone. He had a fantastic finding. His finding was, Simon Govini, he finds that upon spinal injury, when the mice is kept in a enriched environment, okay, you all know this is another important aspect of epigenetics. When you are in an enriched environment, like in Industrial University, your many positive epigenetic factors are upregulated. This happiness can even repair your spinal injury better. That was Simon's observations as compared to standard housing. Simon finds that this is happening because of the CBP or P300 homolog activation. And when you look into that, he sees that if he takes the knockout mice, this repair is not happening. He asked me, can you give your molecule? Instead of keeping them in enriched environment, I can treat with your molecule. Then I think we will be able to get the spinal injury repair much faster. First, he used CSP and the CSP-TTK21. You see, when you add CSP-TTK21 in neurite growths in the in vitro culture, there is a huge activation happens. Then he did that. This is the spinal cord. When you put CSP-TTK21, you find that it is repairing very well. And this repair is functional. Electric current is passing by through that. So this clearly indicates that the spinal injury could be repaired by inducing the acetylations. But we submitted the paper to Science, and Science didn't want to take it. First, they said you have to test it in another model. We test this in another model. That is in the rat model. I had to send the material to Geneva. Look at this very carefully. This is only CSP. This is CSP-TTK21. It has got complete repair. Well, but still, they had a discussion, Science and Science Translational Medicine editor. They bought our paper on the cover page of Science Translational Medicine. Since then, this became very, very popular. We had many offers for taking this to the next phase. We are working on that. But there was a major hurdle. So far, what happened? Okay. So far, what we were doing, we are injecting it subperitoneal. Then we started a new protocol and start feeding this. Will it be able to repair? That's what the question. because unless you have the oral drug, it will be very difficult to use it. We find that upon feeding, acetylation is up in the brain. We also see that the electrophysiology, the LTP is also up because of that. We created a rat model ourselves in CDRI. And when you treat these, you see this is upon treatment of the CSP-TTK21, the mobility of the mice has come back to the normal as compared this to this, suggesting that indeed the oral administration of activator can induce the spinal injury repair. We look into the acetylation mark, we look into the regenerative genes, all are activated as we had the indication before. This paper appeared as the cover story of ACS Chemical Neuroscience, and that was a very interesting and happiness for that for us. And then we are tried now, DBT has supported, we are trying to develop the molecule for IND. Can I have a few more minutes, 5-7 minutes? Okay. So the last part, I'm going to show you an interesting story. The first thing I showed you in Alzheimer's, second I showed you in the new concept in the spinal cord injury repair. The third, I'm going to talk to you on autism. So you all know that when we newborn, this is your neural plasticity, that is your neural network. And when you are 14 years old, these things has been properly established. More or less, this is what, and then it changes because of many other reasons. So there is a specific time period for sensory pathway, language development, higher cognitive functions, and that can happen up to the age of 15. So there is a definite time scale for neural development. And these, if you look at, if it is accelerated or if it is delayed, in both the cases, the case will be ID, intellectual disability. It can happen for many reasons. It can happen for gene mutation. It can happen preterm birth. There is a hypoxia in the brain that can have a permanent injury. So people have started thinking that could it be possible that autism also connects to the epigenetic? We also had a very nice data search, and we find 450 genes has been implicated in ID. Among these 450 genes, these P300, CREB, and DNA methyltransferases are very prominent. So we got a mice model, which is called SINGAP model. SINGAP is a human disorder. I thought it is very, very rare, but in the southern part of our country, there is a huge number of SINGAP patients. And this is what happened. There is a advanced development. So this is the more excitation happens because of the mutation in the SINGAP gene. And that created an ID, autism spectrum disorder, and epileptic enthalpies. So SINGAP mice, the heterozygous mice, shows exactly all the behavior of human. When you work on these mice, so many effort has been done to reverse the SINGAP phenomenon, even to put back the wild type protein, but there is no substantial results came out. We had a unique observation, which turned out to be true much later of our publication, we got to know. What we see that simply my student joined me as a master student. I said, okay, just look into it. So this is the hippocampus dendrogyrus area of wild type mice. And this is the SINGAP mice. We chase with P300 mediated acetylation antibody, H4K12 antibody. Do you see the dramatic reduction? This first time connect to the acetylation with the autism. There's a dramatic reduction. Much later we got to know through the computational neuroscience network program that indeed P300 gets downregulated. When we treat our compound, we find that this SINGAP heterozygous mice, compare this versus this acetylation has come back. So we could induce the acetylation by oral feeding CSP-TTK21 in the autistic mice. We look into the, so what happened? There is electrophysiological change happened. LTP alters. And we find that these the deficit could be recovered by treating the compound, which almost to the wild type level. Then we decided a series of behavioral tests. I will not take you through, I will not make you bored by. This is just to tell you that at different stages, we did an open field test, novel object test, social preference, and Morris water bath test for these mice we treated and untreated. The first results was the open field. That was not very, it's a test for the anxiety. Autistic patient has that. This could not reverse. Second test is the novel object. And we find that normally what will happen, these autistic babies or autistic subjects, they will be losing the ability to recognize something which is novel and not novel. We see that we can completely reverse it back by the treatment. The novel object ability, which is terribly down because of the autism. Then the social preference. Their tribe will be very shy. They will not be ready to come and join. And in this, what we find that these mice, when we treated with the compound, the social preference has, they are mixing with they're staying with the, new mice, same equally as the wild type mice. That means that preference has gone. Okay, this is just to show the video of that. I'm not taking you through. Morris water bath test, I told you that this is the water bath. There is a stage here. They will prefer to come here. First, they will, even if you take out the stage, once they remember, they will be remembering it. That is the memory test. So, memory acquisition, the third day, it will be going there. So, what we find that when you treat the compound, then the SYNGAP mice, which was roaming around all over, now it has acquired the long-term spatial memory as compared to the wild type. So the compound has increased that as well. Working memory. So even if you take it out, still it will go. And they see they are waiting at the same time as the wild type mice is waiting. So it improves the working memory. When you look at the normal SYNGAP pathway, that hasn't changed. So this is happening all towards the epigenetic alterations. Well, why it is happening? Please bear with me 3 more slides. Why it is happening so? 3 questions we have to answer here. Number 1, is it inducing the neurogenesis? First of all, is the neurogenesis is low in the SYNGAP mice? And we find that indeed, when you look at the SYNGAP heterozygous mice, neurogenesis is terribly compromised as compared to the wild type from the dentazidus. When you treat that with the compound, it could induce the neurogenesis same as wild type level. So neurogenesis is upregulated. We ask one more question. That is the neural wiring. This is a very interesting experiment. I also did not know. My collaborator taught me. So this is when you cut the whiskers of the mice, which is their sensory organ, then they regrow it. When they regrow it during the memory consolidation, they in their dendrites, because of the memory acquisition for the normal mice, they will get back these mushrooms. But the SYNGAP mice, because of their deficiency of memory, they will not be able to grow that. If when they will regrow their whiskers. So we took the prenatal, neonatal, adolescents, and adult mice. Whisker trimming happens, then we add the drug, and then we follow it up. And then we try to see the regrowth of the spines. And what we find that the Philadelphia density, that is the dendritic spine, when the drug is treated, it is almost to the level of wild type. Very significantly, it has come back. We did the RNA-seq analysis, we did the pathway analysis. Now we know exactly which pathway it is doing the job. So, in summary, for these autistic mice, we could induce the acetylation, rescue the LTP deficit, that is the neurophysiological deficit, showed improvement in the behavioral abnormalities, and increased dendritic branching. That conceptually proves that autism can be addressed genetic modification if we know the right changes. I'm very happy to say in this month itself, so this paper has appeared as a cover story of Aging Cell. And here is the team. These are my collaborators. This is the student, Akash, and Ishar is the chemist. He is an electrophysiologist and also expert in SYNGAP mice. These are all different funding. One of the Indo-French funding has helped us a lot. This is the present team. Akash is here. He is now in Albert Einstein College of Medicine, just has left last year. Thank you very much for your attention.
Thank you, Professor Kundu, for such a wonderful talk. And in fact, the work which has been carried out for such a long years has been presented in such a short time and very, very nice presentation. May I have few questions?
Thank you, sir. This is one of the best epigenetics lecture I ever hear. So it's wonderful. Now we are very curious and alert mode about the epigenetic changes. So like the methylation, acetylation, phosphorylation, like the things. So what we need to avoid, what we need to not to avoid, because you early explaining about this behavior, thinking, talking, discussing, and all the things is coming and altering the behavior of the epigenetic level. DNA is not changed, but the behavior of the gene expression is going to change. So now what things we can keep to utilize our full potential and the help for the better health?
So first thing I would say that, this is the coincidence. Being a political leader, he touched the right part of epigenetics. Modi ji said, Swachh Bharat, Shustha Bharat. So we have to be clean. We have to clean our premises. We have to enhance our, the surroundings. And we have to increase our environment to more enriched. That is the first thing. Enriched environment is very important. It is in your bus, train, roads, flights, institutions, in your home. And this is proven now. And we have to be happy. We have to be happy. Not too much, I need more, I need more, I need more, I need more.
in radical base approaches in heterocycles. Over to you, sir. Sir, to keep the talk for 25 minutes and 5 minutes for question hour session. Sure, sure. इसमें आ रही थी नहीं इसमें जा रहा है तो ये प्रॉब्लम है इंटरनेट का 5 मिनट किया No, that's me. I'll finish in whatever time. Only thing is it has to start. Because of the internet connectivity, there is a problem. Thank you. Entire screen हम तो करके देखा था ना चेंज हुआ है कि खाली तुम्हारा वाईफाई में है मेरे वाईफाई में इसको भी आप सेम नेटवर्क से कनेक्ट करो ठीक है
Okay, am I audible at the back? Okay, sorry for that technical glitch a little bit because of the internet connectivity issues, and we have to share this particular screen on your Zoom as well. So we are having this particular difficulty. Apologies for that. In the next 17, 18 minutes or so, what I will try to do is share the story of one particular project that we are working at IIT Bombay. So before I actually start with my story, let me join other speakers in thanking the organizers, particularly Vishnu Maya, Dr. Yadav and his team for kind invitation and wonderful hospitality that is being provided here in this beautiful place, beautiful campus of Indrashil University. Almost 2 decades back, our research group started working with this focus that we will use functional groups for carrying out synthetic transformations, developing new strategies. And now this is going to be a little bit of a different thing than what we are used to seeing, right? Typically, functional groups are relegated to textbooks only. Most often, people talk about using catalysis for developing chemistry, solving total synthesis problems, so on and so forth. And here I am talking about functional groups. It's almost like going back to the basics. But believe me, when you start asking the relevant questions with the basics, you actually come across very, very interesting chemistry that one could develop. So one of the functional group which is also going to be part of the story today is the one which is highlighted in this particular circle here, right? If you look at it, many of you will recognize it as an acrylate. You would have seen this in Diels-Alder reaction, for example. But perhaps without this X was a carbon probably there, and here we have a heteroatom. If X is oxygen, we would prefer to call it alkoxyacrylate typically if it is nitrogen aminoacrylate, but we prefer to call them as vinylogous carbonates or vinylogous carbamates depending on if X is oxygen or nitrogen. The idea is very simple. If it is OCOO, it's a carbonate. If I insert a vinyl group, I call it vinylogous carbonate. That's the genesis of this particular terminology. When we started our research, this functional group was known to be excellent radical acceptor, and I will showcase some examples of that. But we also developed chemistry around that, taking it in a non-radical regime, but that's a story for some other day. While working with this functional group, we started working with alkynes, and later on, I'm very sure we'll hear more about alkyne chemistry. I see Raji is here, who has been contributing very nicely in this particular domain. And we have been working with alkynes typically in a non-metal catalyzed regime. And some of it you will hear it probably later in the afternoon from my students. While working with these functional groups, we also try to be opportunistic. Wherever we develop a method, we try to use it in the total synthesis of small molecules. So the idea here is not total synthesis for the sake of it, but rather try to highlight our methodology's usefulness in the total synthesis. So homegrown recipes is what we prefer using in total synthesis of small molecules. Apart from that, we also collaborate with many colleagues who find our molecules useful, and that is also something which we work very passionately about. But today's story, like I said, revolves around radicals, starting with vinylogous carbonates. And to cut the long story short, we had developed a lot of different type of methods which are shown in this little bit busy slide, where we could show that you can tether a radical onto the vinylogous carbonate and do a simple radical cyclization reaction, and you can form rings. That was the basis on which we developed strategies for making heterocyclic compounds. And this particular story starts with this particular vinylogous carbonate tethered alkyne, where we added stanyl radical, so SN dot, which will add onto the alkyne and then follow it with generation of vinyl radical, which will add in turn to vinylogous carbonate in intramolecular fashion, leading to the formation of a dihydrofuran derivative. If we replace that tin with the sulfur, what you get is the thiol radical, and that thiol radical again will follow the same path, but the product formed is furan. Now, this was a very interesting observation, and we were quite excited about this particular strategy for making furans and dihydrofuran derivatives. But one of the problem with this method, what we always felt, is the limitation of having this extra appendage, either the tin or the sulfur. One can, of course, use these appendages for doing more interesting chemistry, but in the total synthesis regime, we cannot always have these extra appendages because they will require additional steps to remove. So it would have been very nice, we thought, that if we could add a hydrogen to initiate this kind of a transformation. So that was one thought which was recurrent in our mind. And the other aspect which was also there in total synthesis regime is that can we set quaternary carbons in cyclic ethers? And with this idea, Rupali, who joined my group around the time when this chemistry was happening, asked this question: can we have an olefin of this type which can give me a tertiary radical by adding literally a hydrogen at this particular point and give me a tertiary radical? And if that tertiary radical adds on to vinylogous carbonate, we will be able to get tetrahydrofuran derivatives, and they will also contain a quaternary carbon. I said, this is a fantastic idea. Why don't we look what that H dot condition that you are going to use? And she came back and said that actually around the time Phil Barron had started developing this hydrogen atom transfer cyclization reactions. And this hydrogen atom transfer using iron and silane, essentially what it does is it generates a formal hydrogen. Of course, it is metal bound, adds to the olefin, and that tertiary radical adds to the acceptor. And this idea was later on taken up by many other groups, Bradshaw, Harzan and Shenvi to make a lot of different kind of compounds. So we thought, can we use this strategy for making tetrahydrofuran derivatives? Uh, she went ahead, tried this reaction with iron silane combination, iron acac and phenylsilane, and she found that reaction actually works quite well. Apart from the fact that you get, apart from tetrahydrofuran derivatives in excellent diastereoselectivity, you also form this alcohol as a side product, right? And this particular side product was forming all the way up to 30%. But after some optimization, she figured that you can actually keep it to the minimal and the required product can be obtained in a fairly good yield, essentially. So she went ahead with the optimized condition, looked at the substrate scope. A variety of substrates would work. You can have alkyl substituents here. You can have aryl substituents, and the reaction will work with excellent diastereoselectivity. For the most part, you get the single diastereomer, even when you are forming a quaternary carbon in the way. The diastereoselectivity drops if the olefin, instead of having R1 as Me, if you have H, that means this is going to be a tertiary carbon, then the diastereoselectivity seems to drop a little bit. And that is the only point when diastereoselectivity was not as great. Otherwise, always we saw only single diastereomer formation in this particular transformation. You could also make spirocyclic systems. Not only 5-membered rings, you could also make 6-membered rings. And those 6-membered rings again were obtained with excellent diastereoselectivity for the most part, right? And of course, we have done the structural conformation and so on and so forth. So this reaction was turning out to be quite interesting. Not only that you could make quaternary carbons, but you can make quaternary carbons which are bunched together, lot more difficult to make otherwise. Two quaternary carbons adjacent, it's a really challenging scenario. And this is something which we are able to do using this transformation. So what is the mechanism of this particular transformation? The mechanism of this transformation essentially, like I said, involves formation of iron hydride complex, which will transfer your hydrogen to the olefin, generating the radical. That radical will do the 5 or 6 exotrophic radical cyclization. The new radical, which is formed alpha to the ester, will get reduced with iron 2 to iron 3, and then negatively charged species is going to get protonated with the alcoholic solvent that we are using. And that's how the reaction is going to give us the product. And once you understand this mechanistic rationale, what you realize that diastereoselectivity is function of what textbook chemistry has taught us, that the incipient substituent, the carbethoxyethyl group, typically will prefer to be equatorial, which will be favored rather than the axial. And that's how one can explain the diastereoselectivity of this particular transformation. So this was quite interesting and good. That time we said that we are able to make only cis isomer. Can we make a trans isomer of this particular reaction? And Rupali said, yes, I think we should be able to do it. So what she realized that the byproduct that we were forming, the alcohol that was there, if we treat this alcohol, she said, under the basic condition, then what we should be able to do is an oxamical reaction here. Now, this is not a radical regime. This is a proper anionic regime. And you should be able to form the trans isomer of the tetrahydrofuran. And the proof of the pudding is in eating it. She indeed found that she could make trans isomer of the tetrahydrofuran in about 10-to-1 diastereoselectivity. So we have access to both the diastereomers. However, this is the byproduct which we didn't want in the previous reaction which she is using. So a little bit of thought, we went to the drawing board and I said, why don't we try the reaction in a slightly different way? Instead of making wine-analogous carbonate first by addition of alcohol to ethyl propionate, you first add radical to ethyl propionate. So you will form this kind of a coupled product, and then you do oxamical so that you can make tetrahydrofuran in a trans isomer fashion as well. So this was the basic idea, that you start with the alcohol which is trithered to alkyne, add the radical to the ethyl propionate, and you should be able to get either this compound or directly Michael addition to the acrylate portion and then the tetrahydrofuran moiety. I said, fantastic idea, let's go ahead and try it. But you know, chemistry always has surprise for you. Neither we got alcohol nor we got tetrahydrofuran. What we ended up getting is a ketone. Now think about it. It's a reduction condition that we are using, and product that is forming is an oxidation product. My first reaction was I didn't believe it. I said repeat. And she came back with the same NMR, which is the ketone. And after some experimentation, we realized that this is a repeatable reaction. And then we went and checked in the literature. Is there a precedence of this type of reaction? We found only one isolated example, which essentially was published during that time, which showed that a hydrogen can be moved from one position to another. Essentially, in a 1,5 fashion, one could do this hydrogen atom transfer under the hat radical cyclization condition. This was shown by Sarah Riesman in a highly specialized condition in a total synthesis. That was the only example we found where intramolecular 1,5 hydrogen shift under hydrogen atom transfer condition was reported. Another paper appeared during our work was progressing, which was by Rajan Babu and Nagib, which showed that you can take sulfonate, vinyl sulfones, with the cobalt silane combination, you can actually do remote oxidation. And this oxidation was essentially at the net level dehydrogenation. And that kind of gave us an idea that this 1,5 hydrogen shift probably is responsible in our case as well. And I'll explain to you the mechanism a little later. So we went ahead and optimized, because this looks like a very interesting way of making what are essentially alpha, beta, gamma, delta unsaturated esters. Amongst the student, the moment you see gamma, delta unsaturated esters or ketones or carbonyls in general, the first reaction that will come to your mind is Claisen reaction, Claisen rearrangement. And we are able to do a non-Claisen route to Claisen product in a matter of one step effectively, right? So this looked like a very, very interesting opportunity. Not to forget, the beta position has a quaternary carbon. So that synthetically becomes even more useful transformation. So we optimize this reaction. To cut the long story short, while catalytic conditions do work, the yield is better when you use stoichiometric amount of ion. And that's how we finally stuck to very general reaction. I don't want to bother you with the substrate scope. You can pretty much have any substituent next to hydroxy group. You can have aryls, heteroaryls, alkyls. You can have even rings, and the reaction works quite well. So the gamma delta unsaturated carbonyl compounds, which also has additional ester, by the way, by virtue of using ethyl propiolate in this transformation. So it's a very, very useful kind of a reaction for making these gamma delta unsaturated carbonyl compounds. What is more, you can set, like I said, two quaternary carbons next to each other. Now, those of you who know Claisen chemistry a little bit, they will appreciate that even Claisen, while it can set one quaternary carbon, two next to each other, is a serious challenge there. And this method is able to do that in a matter of a step. So this particular reaction is very, very attractive from that angle. You can get not just the ketone, you can get aldehyde. And this particular aldehyde essentially was used in the total synthesis of a terpenoid, which was made in about 4 or 5 transformations. We are able to do it in single step, all the reagents coming out of the bottle, commercially available reagents. So it becomes very, very interesting and useful way of making these derivatives essentially. In terms of acceptor, what all acceptors we can have? You can take pretty much any alkyne and then the reaction will work pretty much well. You can have substituted, disubstituted alkynes as well, and the reaction works quite well. This particular point, can we do 1,6 hydrogen shift? Now you will get gamma, not delta, gamma delta, but delta epsilon unsaturated ester. Now you don't have many good methods, or any good methods, if I say, for making these reactions do work well. However, the yields start dropping slightly compared to the 1,5 hydrogen shift examples, right? So we probed the mechanism of this transformation. The 1,5 hydrogen shift is what we believe is happening. And we saw that without ion reaction doesn't work, without silane reaction doesn't work. If we don't add the acceptor ethyl propylate, you simply see the reduction, no oxidation of alcohol. So ion is not doing the oxidation. And then we also put a cyclopropyl group here, which is kind of a radical clock. And it turns out that the cyclopropyl ring was intact in the ketone. That means the hydrogen transfer seems to be even faster than cyclopropane ring opening, which is one of the very fast reactions. Now, final proof of the hydrogen shift came by deuterium labeling. So we labeled deuterium next to the hydroxy group, and then we found that the entire deuterium, almost 98% deuterium transfer was observed, nailing that the mechanism is actually involving 1,5 hydrogen shift in this particular transformation. So overall working hypothesis is you add the hydrogen to this particular moiety, you make a tertiary radical, and that tertiary radical essentially will lead to the generation of a vinyl radical, and that vinyl radical will undergo 1,5 hydrogen shift. And that is how the oxidation is going to take place in this particular transformation. Lastly, what we said is all along I'm using alkyne. Can I use acrylates? And a quick thought told us that based on the bond dissociation energies, the energy gain that we are going to get by sp radical taking up the hydrogen versus sp2 radical taking up the hydrogen, this reaction may not actually work. And that's what it turned out when we were using methyl vinyl ketone or acrylonitrile or the acrylates, you got predominantly simple coupling, that is the alcohol as the product, no ketone was observed. It is only when we had olefin next to a phosphorus or a sulfur containing system, that's when the hydrogen atom transfer would happen. And we did a little bit of the easy computation, and we found that the bond dissociation energy of this CH bond, which is about 70, and that of the alpha to the ketone or ester is about 80, right? So you don't get enough in terms of stabilization by this 1,5 hydrogen shift. But same thing if you go to the latter part, where in alkyne case the bond dissociation energy difference is almost 30 kcal per mole, computed one, that is when you see that hydrogen atom transfer will happen. So there is a threshold at which the reaction happens, the hydride migration happens, or it does not happen. How am I doing on time? A couple of minutes or done? Couple of minutes. Okay. So if there are 2 minutes, I will very quickly tell you what we went ahead with this particular idea. Another challenge when we started working on this was, while this hydrogen atom transfer and hat was well known for the olefin addition, there was hardly anything on the heterocycle additions. And that is what Rupali went ahead and found, that there are only a couple of examples where the pyridine as a heterocycle is used as an acceptor for hydrogen atom transfer reaction. So what you do, you have olefin tethered to pyridine, the radical will add to the olefin, tertiary radical will add to the pyridine, and it's kind of a mini-C reaction, if I may call it that way. That's the only reaction which is there. But pyridine is electron deficient. Will it work with the indole which is electron rich? That is the question she wanted to address. And we had a little bit of a history of working with indoles. So this was much but natural that if I have nitrogen heterocycle with tether the olefin, will this cyclization will happen? So she went ahead, tried this reaction, N-tethered olefin, silane addition. So hydride adds here, the radical will cyclize onto the indole, and the reoxidation actually gives you the N-fused indole derivative. So this reaction actually worked out quite well. Very general reaction again. So you can make N-fused indole derivatives, lots of different type of reactions she could show. I don't want to bother you with the substrate scope. One last point I will tell you that at that point, reaction was working with indoles and pyrols, but that was giving us N-fused indole. Will it work for C-fused indole? That was the question. And if C-fused indole has to work, essentially your radical has to undergo 5 endo-trig reaction, which is Baldwin forbidden. And that is what she tried. And guess what? When you try this particular transformation, the reaction actually leads to the formation of the product. The 5 endo-trig radical cyclization also happened in this particular instance, paving the way for getting the C-fused indole derivatives, which we applied in the total synthesis of brucioline natural products, right? So I don't want to bother you with the details of the total synthesis, but method developed in-house could be utilized in the total synthesis of these indole-based natural product like brucioline H and the analogs of those. So in summary, what I have tried to show you that when you develop radical-based chemistry using hydrogen atom transfer, not only that you can use it for making simple tetrahydrofuran, tetrahydropyran, chromine type of derivatives, but you can also see that accidents along the way, this is particularly for the student, don't get bogged down when you don't get what you expect. In fact, you may be actually looking at something which may be more interesting. And that is what we observed when we saw that 1,5 hydrogen shift is one of the side reactions became the main theme in very exciting way. And then we could also use it in addition of this radical onto the heterocycle to make natural products as well. So what remains to be done is thank the people who were involved in this particular journey. So Rupali and Simran were the key player in this particular project along with Deepak and Ajay. We thank the funding agencies who have supported our research over the years, all my collaborators whose work I did not talk, and thank you organizers for giving me this opportunity, and thank you all for your patient hearing.
Thank you, Dr. Santosh, for your new approach in radical base chemistry. May I have few questions?
Yeah.
Sir, it's a very nice piece of work. Instead of oxygen, can we think about NH, protected NH over there to do the cyclization process?
You're
in the
talking about 1,5 hydrogen shift, or you're
furan, pyran, all this synthesis?
they can be used.
Can
They
be
can
done.
be used. Vinylogous carbamate, essentially, you are saying.
Yes.
Yeah. Stay tuned. Next time. Yeah.
Can we develop the method to synthesize trans tetrahydropyran?
Under the free radical condition, method to make the trans tetrahydropyran. Difficult because basically it will be governed by your conformational preferences, and at least our experience has been that whenever you are trying to do any of these, they always prefer to go equatorial. So base mediated reactions are one of the very few where you actually get the trans isomer as the major product. Now, having said this, I must tell you that we had some success with making trans pyrrolidines when we were doing the alkyne cyclization, but not in the radical chemistry. No, mostly cis.
Yeah, so quick one.
Yeah.
So your Brussels synthesis, it's a fantastic one. In BBR3,
Huh.
you also got reduced. Can you go to that slide?
Yeah.
The total synthesis you have shown, the Brucellin.
Give me 1 second. Here.
Yeah. That last step, you say BBR3 you have used.
Right.
So demethylate, but
Yeah.
in addition, it also reduce this alpha diketone.
No. That's a mistake. That's a mistake.
Okay.
Sorry about that.
Okay,
That's a
that's
mistake. Yeah.
Diketone.
It is a diketone.
Professor Gharpure, very inspiring to me.
Thank you.
So I have a question. You are using this metal hydride as a radical generator. But what happened? Why it is not generating radical near to the carbamate, analogous carbamate, carbonate? There are also possibility to generate the radical at the carbonate position.
Absolutely. So actually, if you look at it, these hydrides are very sensitive to the electronics of the olefin that you are using.
Exactly.
Right? So you notice that there are more than one acceptor. I didn't talk about, but we can actually have an olefin and acrylate and a silane with the indole there. And you can see that the radical will add only to the olefin first, followed by addition of that radical to the acrylate, followed by that radical to back to the indole. So I didn't talk about that today.
Yeah.
But you can actually beautifully sequence the addition sequence because they are very sensitive to the electronics of the olefin that is present. Now, acrylate, while it is not as deficient as acrylate, vinylogous carbonates are not as deficient as acrylate, they are worse than the olefin. So olefin is the one which will accept the hydrogen first. That's why these reactions work, but it's a very good observation.
Yeah. Another thing I want to know, you are showing the mechanism for 1,5 hydrogen shape, and you are generating their carbon center radical, then it is oxidized to the ketone. But what happened to that proton? It is releasing as a hydride or ready H dot, and that is again doing the catalytic cycle. You prove that something like that?
We did try a few experiments with that. The catalytic ion is working. So that kind of suggests that that hydrogen actually can go as a radical.
Exactly.
But we could not get exact stoichiometric complete conversion because reactions would stop there. So we had to resort to the stoichiometric ion. So when I use stoichiometric ion, I cannot be 100% sure of what the mechanism it is going. But the catalytic regime working suggests that there is a possibility of hydrogen going as H dot. So, but then unless I have a foolproof mechanistic evidence, I won't want to commit on that.
Okay. Thank you.
Indications are that.
Thank you.
So, thank you.
A big round of applause for Dr. Santosh.
I request Dr. Vinay Kumar Srivastava to felicitate Professor Santosh J. Gharpure. Now I would like to invite Professor Sivapriya Kerumbakaram from IIT Gandhinagar to chair the next lectures.
Okay, once again, a good morning to all of you. It's my pleasure to introduce Dr. Mahapatra as his next speaker. He has a very remarkable contributions to organic chemistry and organic synthesis. He is currently a professor at ISIR Behrampur. Before that, he had a lot of collaborations with, you might have seen from his CV that he has worked with the Professor Grubbs and also Professor Ains from MIT. And he has been there in NCL Pune as well as in IICT Hyderabad before joining Behrampur, I believe. And he also got several awards. CSIR Technology Award, Nasi Reliance Industrial Platinum Jubilee Award, etc. to add. So today's title of the talk is Molecular Mysteries, the Art of Structural Illustration in Drug Research. So I will ping you 2 minutes before your 20 minutes, Professor Mahapatra.
Yeah, please.
I'll get up 2 minutes before your time gets over.
Good morning to all of you. Thank you, Professor Sipriya, for your kind introduction. At the outset, I would like to thank Professor Yadav sir and Professor Vishnumaya for their kind invitations and giving an opportunity to showcase our work. And as everybody knows, Professor Yadav sir, as per Professor Kundu's today's lecture, that he makes you or feel you so happy that we get elevated our happiness, and that motivates to work hard. Because when I started my career at NCL Pune, from there, there is another. Actually, I'm lucky to have three mentors. So one is Padma Bhushan Dr. A.V. Ramarao. Second is Professor Jayesh Yadav. And the third one is Dr. Mukund K. Gurjar, who is the CSO of MCure Pharmaceuticals. Now, I got very lucky to have another mentor called Professor Ashok K. Ganguly. And now he is the director of ISIR Behrampur. And recently, I think 1.5 months before I moved from IICT Hyderabad as chief scientist to ISIR Behrampur, you can see the beautiful picture of this building. This is the academic building. And this is the beautiful building of CSR IICT. But I'm lucky that I'm at the two places for next 2 years. And today I'm going to talk about molecular mysteries, the art of structure elucidation in drug research or drug discovery. And already Professor B.K. Singh have covered almost my introduction part. That way I'm ease out. Actually, I'll move very fast those introduction slides. Before I start my lecture, I would like to thank all the team members. Actually, we are at IICT work together with Professor Jayesh Yadav sir. And this is the team. At one time, we had 40 PhD students in a single lab. And if I remember correctly, so one of the director of CSL lab came to me and asked me, so do you think, are you thinking like you are close to God? So because the question was why and how you have taken 40 PhD students. So I said, it is not taken, it is given. So it's like a God has given. So like that, so it was the combo of two scientists. One is Professor Yadav sir and myself. So we had the nice teamwork, and we had that time almost like 2008 to 2017, 40 PhD students in single lab. And many of them, actually, I'll go to the next slide. So these are like, so this is like, again, 14 to 22. And always I feel proud to show the third slide. This slide Sorry, it's not working. Okay. Because 5 of our students went to Harvard University, work with Nobel laureate Professor E.J. Kore. And the first is already, as per Professor Yadav sir yesterday said, 7 to 8 students have there started their own industry. So the first student, Professor Dr. Ishwar Reddy, has started his industry at Hyderabad. And the second student, Barla Tirupati, is working as as associate professor in the same institute, Aizer Barampur, where I has moved actually as a professor. And Srinivas Reddy already working in the industry in USA. Sudhakar Reddy is already still continuing in EJ Kore's group, and he has got the offer from TIFR as an associate professor, and the process is on. And Yadav Bharat actually moved out from EJ Kore group to some other group. Now with this, this thing always, I'll be always grateful to these students, past and present students, because they're highly committed and motivated students. Because of them, today Mahapatra is what actually I'm today. Yeah. So as a research group, what do? We try to synthesize total synthesis of some complex natural products. And the most important thing is not the target. So we try to synthesize these natural products and try to assign the structure of those complex natural products. And through that process, we try to help these medicinal chemists by designing molecules on that scaffold. And there is another group, very interesting group, join in chemical biology. For them, actually, we had collaborations for biocompatible organic nanomaterials. And through that process, whatever we learned, the skills, we try to apply for industrial collaborative projects. And I need to cover all these points because why total synthesis is required. Because if you go to any medical shops, out of 100 medicines, you'll find 60% of these medicines are either natural products or natural products based. So because of that, it's very important to involve with natural product synthesis. And why complex natural products? Because one of the example is helicontin B, which contains 32 stereocenters. Now you know what is the possible isomers? 2 to the 32. Almost 4 million isomers are possible. If you do not have the skills to synthesize single isomers, you'll end up with the wrong isomer, and that will lead to a drastic effect in the biological system. And out of that molecule, so we have, so this company, particular SI, designed this molecule that is called eribulin and came out as one of the most potent breast cancer drugs. So another example is Taxol. So everybody knows is a again potent anti-cancer agents, and we have made a lot of, like people have made a lot of derivatives. Derivatives, and it has come out as the most potent cancer drug and available in the market. Now, the third point is that already Professor V.K. Singh have said that if you try to isolate this type of compounds from nature, we will end up with very minute quantity. Like if you want, sorry. So if you take one example, it's called trabectin. It's already a cancer drug available in the market. If you start with 100 kg, you'll end up with 100 milligrams of these natural products after the purifications. So it is very difficult to capture the more than 8 billion populations of this world. So for that, the key role of synthetic organic chemistry is to replicate this natural product in the laboratory, and that also not in gram quantity, in kg, followed by tons quantity. So there our key role starts, and again the question arises that synthesis is not important. Importance is you have to consider everything actually. So economic point of view, and how we can achieve in a greener protocol way. So how can you achieve these molecules? Suppose now if you take trabectin, earlier it was reported is 22 to 27 linear steps, whereas very in recent publication 2023, so they have come up to 18 steps, and that also in gram scale and kg scale and ton scale, which is used now in the industrial process. And today's my talk will be based on stereochemistry. That is the third point highlighted by Professor V.K. Singh, sir, that stereochemistry is also one of the key player in the drug discovery program. So any simple change in the stereochemistry can have a very deadly impact on the biological activity of this. One of the very oldest example is thalidomide. So there is a R isomer and S isomers. Earlier it was available as a mixture, And the history is here. In 1950, more than 2,000 kids died and 10,000 people born with physical, or you can say defects, birth defects. And one of the medical doctor actually find out this is the culprit, because that time it was only available as a racemic, but nowadays almost all drugs are available as a single isomer. And because this S-thalidomide is having a teratogenic effect on this. So now I'll not go to this. So as a group, so what we do focus only particularly with respect to natural products. If you see here, one of the example we have given this liquoscandrolite having 2 tetrahydropyran ring systems. When the medicinal chemist asks that is very complex, having 9 stereocenters, and if you to the 9, if you say, it is almost like 256 isomers. Very difficult for an organic chemist to control all the stereochemistry and synthesize this type of natural products. So they asked to make a very simple derivatives of this liquoscandrolite. So after this synthesize this derivative of this natural product, what you found is, you see, the IC50 value of this for a particular in the cancer cell lines, the natural products was showing 0.05 microgram per ml, whereas the design molecule was gone up to 28 microgram. That means it became inactive. So what it indicates that nature has made the, which is like, becomes like a drug or drug type molecule or drug like molecules. So we cannot, sometimes we cannot change these functional groups. So with keeping in mind, so we just did some literature search. What we found that there are so many natural products having potent anti-cancer activity, like helicandrid B already I have said about, liquoscandrolite A, sorangisine A. So what is the beauty is that if you see in this all these molecules, there are 2 common scaffolds are there. One is dihydropyran. If you have a double bond in between, this is called a dihydropyran. If you all are saturated, it's called a tetrahydropyran ring system. Already Professor, the earlier speaker has talked about, and Professor Garpur has said about that, how to synthesize tetrahydropyran and tetrahydrofuran ring system. I'm going to talk about following a very simple protocol, how to control the geometry of these tetrahydropyran ring systems. If you want a trans, you can get a trans. If you want a cis, you can generate a cis tetrahydropyran ring systems, and applying that simple methodology. So how we have synthesized almost like 30-40 natural products. And today I'm going to talk about particular 2 natural products where we have revised the structure. Now there are some more natural products you can see. I'll just go fast because I have very less time. And now this is the literature search. You can make through O1C2 and C2C3 and like C3C4 bonds making system. But our main intention was, there is a lot of methods are available. So our intention was to make this type of scaffolds where you have an internal double bond and a terminal double bond having different reactivities. And then utilize that reactivities for further explorations, and then leads to the natural product synthesis. And what we found actually during that time, only one reaction was known, that's called Sakurai allylation type of reactions with a lactose system. So what we intended, actually our hypothesis was to start with a acyclic system. This is called delta hydroxy alpha beta unsaturated aldehyde. It cannot lead to lactose unless until we isomerize this trans double bond to cis double bond. So that means in one pot we can do 3 reactions, then only it can leads to this type of cyclic compounds. And the another advantage is that here it was giving less than 9:1 ratio of diastereomers. And in the column chromatography, it's very difficult to separate sometimes the diastereomers. So to ease out that, we planned in such a way so that it leads to a single isomer. So how to do it? So if you introduce another double bond, then the rigidity appears here. It's not exactly flat, but it's like a twisted board. And then that leads to exclusively a trans product. So keeping in mind, so we tried these reactions, allylations under Sakurai conditions, using very mild Lewis acid, that is a molecular iodine, and we got 45% yield, and later on optimized, and then we got more than 90% of this single isomer. Now with this methodology, so we optimized this thing, and then this is the mechanism proposed. I'm not going into details actually. So now this is the mechanism proposed and accepted by the reviewers, and with all this. And then our intention is to apply this methodology towards the total of natural products bearing dihydropyran and tetrahydropyran ring systems. And this came out as a cover picture in Chemistry European Journals in 2010. And now I'll go to the, okay. So now if you talk about the natural products, so some generally after the isolation is done, people do the characterizations, and some structures are solved very accurately with the stereochemistry, and some are solved very inaccurately, and some are could not be solved, even though we are living in the 21st century. So now I'm going to talk about two natural products. So this is one of the natural products. We solved the structure, a stereochemistry of this natural product, and then chemotagic chemical picture and JOC. Now you see, now I'll talk about two natural products. I'll go a little fast. So now if you see these natural products known as crypto-digital K, and what is the difference between these two structures? The side chain. And if you see here, there is a hydroxy group at the 11 prime, and on this side 6 carbon, on this side 7 carbon. So how come the two structures have been given in the single manuscript? One appear in the manuscript and one appear in the supporting information. That means from there actually we realize that there is some problem with this natural products characterizations. Second point is that one is at the 11 prime, one is the 10 prime, and if you see this NMR data of this OHCH, it appeared at 3.89 ppm. So in general, as per our spectroscopy knowledge, so no secondary hydroxy CH proton can appear at 3.89. It has to be allylic because we have a double bond. So that clearly indicates that this hydroxy position is also wrongly assigned. So keeping in mind all these 2 points, so we thought, and third is our tetrahydropyridine system is there. So we wanted to apply our methodology for the synthesis of this natural product. So I'll go for the retrosynthesis. So 2 people who are actually in the Courage group, actually they're involved. And if you see, this is called double bond can be generated through olefin cross metathesis. And this double bond, terminal double bond can be isomerized under Dobbs conditions. And this allyl compound, this diene compound can be synthesized or prepared from our method. And starting from epoxide. And this side chain can be prepared from epoxide opening through your alkyne substrate. So I'll not go details in the chemistry. So these are our chemistry applications of this methodology. And then we followed it following the standard protocol. Now we went for the cross metathesis. Now we synthesized both the isomers S and R, 11S and 11R actually prime. And then we tried to correlate this data. And before that, we have to synthesize another 2 isomers having this side 1 carbon less and this side 1 carbon more. So we followed the same protocol, cross metathesis, following our own iodocyclizations. And yeah. Now this is 10 prime, 2 isomers. And then after having 4 isomers, we now compare the spectroscopy data with the natural product. What you realize that whatever we have predicted, with that 3.89, it came appeared as 3.58. That clearly says that, so this is not a aliphatic OECS. This will be definitely a allylic CH-OH carbon. Now the next is we published as such, stating that the large discrepancy in the spectroscopy data of synthetic crypto-residual K from the natural product suggests that the structure of the natural product crypto-residual K requires revisions. With that we published. And then the original, whoever isolated this, he's a very big guy. So he was the chief editor of Journal of Natural Products. And then immediately he wrote the mail. So this is the mail he wrote. And what he said, I apologize to you for the effort you spent to synthesize the wrong structure. All these 4 natural products are not actually the natural products. But the students put a lot of effort to synthesize not 1 natural product, 4 natural products. But everything came out as wrong. So now what is the next effort is to synthesize the correct natural products. So now we predicted. So this also, then he also highlighted in his paper actually the corrigendum. And now we said that this could be the actual natural product structure. With that, now we have to synthesize this, whatever we have predicted. And then we started the synthesizing. It's very simple, starting from the same synthetic protocol using this our diallyl compound. I'll go back to this thing. Okay, this is the diallyl this thing compound number 7, and with that we did only the oxidation of this double bond to aldehyde, And then this double bond was introduced through another protocol called Nozaki-Hiyama-Kisi reaction. This is nothing but a simple Grignard reactions in presence of chromous chloride. So I'll not go again the details of this chemistry. Okay, now we synthesize both the isomers because this hydroxy center was not assigned. So we synthesize S as well as R isomer because all these three centers are fixed through NMR data. But this was not fixed. So with this, what we did actually S and R, now we compared the data. You just see exactly overlapping with NMR as well as the C13 values. And which clearly says that the correct structure of cryptodigital fluoride K is the S isomer, and this is the correct structure. So with this, we published another paper. So now I have only another 2 minutes. I'll finish this molecule. So this is another molecule known as monosilin VII, and it was isolated in 2017. And looking at the molecule itself, we can, with our expertise, we can judge whether the structure is correct or wrong. How? Now let us go to the next slide. So we have already synthesized this type of natural products known as resorcilic acid lactams. And from there, what the experience we got actually, if you have a beta methyl, the value of the C13 value is above 70, sorry. And if it is alpha, it should be below 70. Okay. Now, if you see, look at the natural products, this is a 10 prime is alpha, it should be below 70. But how much they have given? Above 70. That clearly indicates that this should be beta. By mistake, they have written alpha. So now the next is to synthesize and prove it. So there are two methods we followed. One is our protocol is called isocarbon synthesis developed by us, and another is Nicholas protocol. And we've only succeeded with Nicholas protocol. Okay. Now we synthesize this proposed natural products. And then when you compare with this proposed natural product data, you see how much differences are coming. 4 prime, this is the 4 prime, 8 prime, and 10 prime. All are like huge difference of the delta values. That clearly indicates that our hypothesis is almost like correct. But how to prove it correct? You have to synthesize the other isomer. And then we followed the same protocol and then synthesize our hypothesized molecule that is the revised structure. And you can see here the protein and C13 values both are overlapping with the natural products. That clearly indicated that the natural product structure should be this one, not the earlier one. And this is the way actually we have corrected. But see, the chemistry involves a huge chemistry involved. With the 20 minutes science, very, very difficult to talk about the story, what behind that actually. And the next is these two molecules, even though it looks very small, your 4 centers are there. How many stereoisomers are possible? 16. So that means you have to make 16 natural products or derivatives to prove the correct structure of dipropyrrole, even though it looks very small one. Same way, pochonin J also having the stereochemistry issues. Now we are in the process of revising structures. And what is the final conclusion is that molecular puzzle of dipropyrrole and pochonin J are still unsolved, and the story will continue. If I get a chance, then I'll maybe come back with all this, how we have resolved these issues of the stereochemistry or the structure conformations. That I'll come back in the next meeting if I get a chance. And with this, I would like to thank once again the convener and the organizing committee for giving the opportunity to showcase our work here, and the SARB, DST, CSR, and industry partners for the funding. And thank you very much for your kind attention. This is a very beautiful campus coming up at ISR Barampur. You can see I have given the title Blending of Natural Beauty with Fabulous Architecture. So, and this is the sea, complete sea, and our campus is only 1.5 kilometers away from sea beach. So we have a very nice ISR sea beach we are developing soon. And you people are most welcome to come for MSc internship for 2 months. We'll be happy to welcome you people for students. And thank you very much for your kind attention. These are all our cover pictures. So with this, I would like to thank once again.
Thank you, Professor Mahapatra.
Yeah.
Because of time limit, we can have very quick 1 or 2 questions.
Yeah, it's a very nice actually. The way you have shown that those stereogenic center setting up. It's really so. What kind of processes you have used? You make all the fragment. I mean, all the diastereomers, and then finally set it up, or
not exactly. So we have done the analysis of spectra, and from there we find out which is having the highest difference in the delta values with respect to C13. and proton.
Okay.
So
okay.
as for that, we decided to synthesize that isomer fast.
Okay.
And luckily we got that. With that isomer, we got the structure clarity.
Yeah,
that this is the
otherwise,
best.
you know, you
Otherwise you have to make 16 isomers.
Exactly,
Yeah,
yeah,
16
yeah.
isomers. It's
Mm.
not exactly. And second thing is that for dipole iron, we are taking the help of DFT studies.
Okay.
Yeah.
Oh, that
A-
really
A-
helped.
A-
Okay.
yeah.
Thank you so much. It's really fascinating.
Good talk. You used iodine and allyl silane.
Yes.
So you don't see Hosomi Sakurai type of a Michael addition?
Yeah,
at any point
that
in
is the
time?
Hosomi Sakurai reactions, modified versions, and
Okay.
under mild conditions. That's why I said Sakurai reactions, and it is only lactol reported.
Right.
Ours is uncyclic, and we have a trans double bond, unless until isomerizes. So under iodine condition is, can I go back to the mechanism?
No, I got that
Yeah.
particular
So
part.
it isomerizes to cis, then
and
becomes
then the
lactol
cyclic.
with a double bond. Same Hosomi Sakurai,
Okay.
but it's the uncyclic, not cyclic one.
Right, because
Or
normally
acyclic,
it will do a Michael addition of the allyl fragment in the
No,
traditional.
that was also like a reviewer asked. It
Huh.
may be going through Michael addition and then all those things, but that is not happening in this case.
Okay.
So we have already proved that it goes via lactol. We synthesize the lactol. We synthesize the OTMS compound, the silyl compound, and then we did the allyl addition. That was the reactions going. So those are all control experiments. But it's very difficult to explain everything what is going on. If I start explaining chemistry, it will be 2 hours.
No, I understand.
Sorry.
I was not the reviewer.
Yeah, sorry.
Yeah, thank you, Dr. Mahapatra, for the wonderful talk. So let's thank the speaker.
I request Professor Sevapriya Kirumbakaran to felicitate Professor Devendra Mahapatra.
Alakesh Bisai. It's privileged to introduce him because the way that he presents his natural products, most complex natural products with such an elegancy, I've been hearing him for last few years, at least being in several conferences. He's one of the pioneers in natural product synthesis in our country, young pioneers, I would call. And he's finished his postdoctoral stints from UC Berkeley with Professor Richard Sarpong. And he has been a faculty at IISER Bhopal and also now at IISER Kolkata. He is going to talk about and several awards, global recognition in organic synthesis. He's going to talk about architecturally complex natural products of biological relevance, a platform for the discovery of new strategy or methodology. 18 minutes, I'll get up, Dr. Alakesh.
Thank you so much. Thank you. That I like to talk about the synthesis of complex natural product. Now, the question comes, why natural product? And that too complex one, because you have already seen yesterday, Professor Vinod Singh showed some of the slide, including Jadhav sir's contribution, in discodermalide. Even today, we have witnessed talk by Professor Mahapatra. Also, Professor Gharpure showed some of the natural product. So, if you look at this slide, the long-term goal for synthetic chemist is either drug discovery or make a material for societal benefit. Now, if you come to the drug discovery, which principally follows ADMET principle, where you will see that metabolism actually the most crucial part where pharma industry actually struggle. Now, if you look at this approved drug, this is the literature where you can see these are all based on the either natural product or their derivative. Of course, there are still, this is the portion actually occupied by synthetic drugs. But as you know, 1960s thalidomide story that shows that enantiomer molecule which is having three-dimensional architecture, probably the very good fit for the drug discovery. Now, if you look at the drug discovery principle, which mainly follows ADMET principle, where this is where the problem comes actually, metabolism, so now it also tells us why, to become a good chemist or biologist, one has to follow nature. Today, Professor Kundu has shown very nice introduction or very nice research on how synthesis of chemical actually processes happening in the biological processes. Therefore, his interesting research on this HDAC and other related, epigenetics is been, it's a very interesting one. This also tells us why heteroatom is required for the drug discovery. And now, if you look at, the good catalyst is nothing but a three-dimensional architecture-based organic molecule which goes inside the biological system and it does work, and then it should leave it from the system, or it should go and take part in the other bioprocessors involved there. So therefore, the circular chemistry, which is already operating in nature, that has to be followed in the biological processes. You can see that natural product like folic acid, vitamin B line is a well-known drug molecule. Now people have actually did little manipulation from folic acid to methotrexate by changing the N-methylation as well as aminolysis, and this pteridine ring would lead to anti-cancer drug. So of course this the synthesis of folic acid takes place, starting from GTP, through oxidative processes. So what I'm trying to say here is that natural product has been the inspiration for research in chemistry and biology. You look at the well-known doxorubicin and darorubicin, they are nothing but, natural products isolated from nature, and the polyketide biosynthesis takes place for their synthesis. So here is one natural product called galantamine, which is a marketed drug, which is among 3 drugs available for Alzheimer's today. So one is galantamine, second one is rivastigmine, which is again another chiral molecule, and then there is molecule called physostigmine and denopagil. Denopagil doesn't have any sterogen. Racemic compound has been utilized, but this is where you can see the stereogenic center as well as three-dimensional architecture with heteroatoms actually play, crucial role in this kind of drug molecule. So very recently, this kind of complex structure has been isolated from nature in 2021. You can see that this kind of partner is already there. However, axially chiral partner actually attached here. Therefore, it leads to a pair of atropodiastereomers. I'm not going to discuss the synthesis. Just want to tell you which are those atropodiastereomer like narcicachinin A and B. They are nothing but a pair of atropodiastereomer, because this bicycle, this biaryl actually axis is leading to a pair of atropodiastereomers. Narcipablin A and B, it is the same thing, but where indolin is nothing but in the oxidized form like indole. So we have shown a strategy very recently, and it's also good to see that Professor Eric Carrera actually highlight this in SynFact. This also gives us a lot of boost for the student to do further research. So this is another class of alkaloids, cyclotryptamine alkaloid. This is started one of my first student, Dr. Santanu Ghosh, who is also going to give lecture today evening or tomorrow. So he has elegantly showed that how one can construct this vicinal all-carbon quaternary stereogenic center in this kind of complex molecule. Now you can see that chimonanthin and chylacanthin, they are nothing but, if you treat with acid, they will rearrange to a complex structure like this. Recently, we have also synthesized Hodgkinsin. Currently, the effort is ongoing for the synthesis of quadrigamine C and cytolin. These are the references where the other professor like Professor Larry Overman from UC Irvine, Professor Mobasaki from MIT, Masanuga Kanai Shibasaki, as well as Barry Trost and Neil Garg. They are actually the key player in this particular area. So we could able to show a number of strategies for these classes of alkaloid. So here are some of the representative, publication. So Dr. Santanu Ghosh is there in the audience. He will be speaking tomorrow. So these are the complex structure we have synthesized. Now this is another area. This is called indolosesquiterpene alkaloid. If you look at their the biological activities, actually they exhibit activities against RNA synthesis protein NSP10, which is the major activator for SARS-CoV-2 replication. Therefore, people are thinking that this class of alkaloid may be utilized as a drug molecule, for this purpose. Among all these congeners, geomycin A exhibits selective anti-HIV activities. Therefore, there are interest to develop strategies, unified strategies. Now, if you look at geomycin and auridamycin, they both are of same class of alkaloid. However, they are diastereomer of each other. Therefore, setting one strategy to address both these diastereomer would be very interesting. Further going towards in this direction, you can see diamycins A, B, C. They are nothing but dimer. Now, NN bond forming reaction could lead to a pair of atropodiastereomers, and they can also form a CN bond leading to diamycin C. So these are the isolation paper. What literature shows that NN dimer shows 10 times better activities than their monomer siblings. So therefore, setting this kind of NN bond and therefore leading to a pair of atropodiasteromer. Look at this particular chiral axis. They are not same molecule. Okay, they are actually atropodiasteromer of each other, diamycins A and B. We thought of developing a unified strategies for them. Not only that, in these kinds of alkaloid, you can see that sulfur dioxide actually incorporated between these two molecules. So this also talks about how biosynthesis of this kind of alkaloid might be taking place, because if you know that, that will give us the opportunity to understand what kind of methodology one should actually develop to make them conveniently, like in this case, this is sulfonamide, this is diaryl sulfone. Now sulfadiamycin C is diaryl sulfone, but this is a positional isomerase compared to sulfadiamycin B. So although these have been isolated very recently, but there are no synthetic effort. Common synthetic efforts are reported. So in one slide, I'm summarizing our effort. It is 2014, Phil Baron from SKIP showed elegant approach to diamycin B. However, their method could not able to synthesize diamycin A. After 10 years, almost 10 years, in 2023, we have shown that a copper-mediated aerobic oxidation to form NN bond, therefore a pair of atropodiastereomer for the first time, and that also tells us the first total synthesis of diamycin A that has been reported in 2023. Very recently, we saw that this kind of NN bond forming reaction could be derived from photoredox catalysis leading to NN bond. Of course, we could also do CN bond type, bond forming reaction using photoredox that led to the synthesis of diamycin C. I'm very happy that this came in the cover page, which is to dedicate the CRSI 25th anniversary. So these are the cover page came out of this research. Again, in 2024, what we have shown that if you take the monomer, this monomer can actually do site-selective oxidation leading to CC dimer. This is carbodiamycin C. This has been synthesized in 2024, and very recently we have shown that this kind of processes can be followed under mild condition using hypervalent iodine reagent. I'm not going to discuss this. So this is another area where actually our group is focusing. If you look at brevianamide A and B, this is isolated by Birch, 1969. And the first racemic synthesis came after 20 years by Robert Williams from Colorado State University. Williams is no more. Unfortunately, he passed away 4 years back. And then again, approximately after 50 years of his isolation, Andrew Lawrence showed that the first total synthesis of plus and minus plus brevianamides A and plus brevianamide B can be achieved following a chiral pool approach. So if you move ahead, you can see stefacidin. This is what is cytotoxic molecule, and even stefacidin B is nothing but a dimer of this particular molecule following oxidation and then dimerization process of stefacidin A that could lead to the complex structure like stefacidin B. In this particular area, Andrew Myers from Harvard University, he's the first person to show the first total synthesis where Seth Harjon, Yale, now he's a faculty at Yale and associate editor of JOC, he showed the first total synthesis in 2005, followed by Phil Barron in the same year, and then my postdoctoral advisor from UC Berkeley, Richmond Sarpong, showed a recent strategy. Robert Williams also showed another racemic approach to this kind of alkaloids. So what we thought, can we still contribute in this particular area? So my student Monosij and Apurva got excited, said that yes, we should able to do if we can make this kind of intermediate. If you look at this kind of pentacyclic intermediate, which can be obtained using MCPBA oxidation, if you have, there are two double bond, but indole double bond will be more reactive towards this oxidant, therefore you can make epoxide. Now this epoxide can open it up, therefore it can undergo a 5-endotric cyclization leading to the formation of this kind of pentacyclic mesylate. And fortunately, the student could able to get the mesylate X-ray, and with that in hand, we followed a 6-step cascade, and that actually shows us how nature might be following this kind of cascade, inverse demand disorder. What exactly going on here? Let's look at, in the presence of a base, it can make alkoxide. Now once it come back, then it can open it up. Now what it can do is that it can form a diene required for the inverse demand Diels-Solder reaction. So in this particular ring, what happening is that elimination followed by isomerization and followed by the formation of this kind of diene, which is responsible for inverse demand Diels-Solder to give us the 9:1 diastereomeric ratio of bavianamide A and B. Of course, in the process, we could also get plus minus pre-balchiamide. That also tells us what kind of mechanism it might be operating in the nature. So this is what is the 3 crystal structure through that we have confirmed that actually it is the structure of bavianamide, and very recently we have published this work. Now going to tetraterpenes, where Professor Jadhav and other people have contributed from our country. So those are all interesting synthesis, what Professor Binod Singh also showed in his lectures, that actually gives us a lot of energy to from India how to do total synthesis research. We really need to appreciate their effort. Even Professor Binod Singh showed one strategy by Govardhan Mehta, if you recall, is a 4+2 followed by a 2+2. That leads to the synthesis of hirsutin and other triquinone type of natural products. Those has been fascinating. So these are the molecules have been isolated in 2018, and they have some biological activities reported. So we thought, can we give the first total synthesis of these kind of complex natural products? Now, if you do the Diels-Solder reaction, one can think of doing a normal Diels-Solder reaction where diene, electron-rich diene, can react with an electron-poor dienophile. Therefore, one can envision an endoselective Diels-Solder, normal demand Diels-Solder, leading to this kind of endoproduct. However, one could also think that can I do an inverse demand Diels-Solder where diene is already attached with electron-poor carbonyl group, and here allylic, particularly this allylic olefin, can undergo a Diels-Solder leading to an inverse demand Diels-Solder. Now, if you look at the energy required for normal demand versus inverse demand Diels-Solder, it would always prefer a normal demand Diels-Solder, and that is what exactly happened. So basically from this process, we are asking only one endoselective product, and that is what happened. So when we tried to make the LUMO from this kind of dienol, so under oxidation, we could get this. However, we could not able to stop there, and it undergo a spontaneous Diels-Solder in one step, leading to the formation of this kind of heptacyclic structure that was confirmed through X-ray. And Professor Ayan Datta from ISCS, cultivation science, actually showed the DFT calculation, and actually it says that as soon as you generate this kind of dienophile, it is immediately reacting with this. So therefore, as soon as the 50% conversion of this kind of dienol happened to this dienone, the reaction is completed, and then further oxidation using DMP gives us this kind of enone, and that has been actually characterized, and this led to the first total synthesis of silazindorphone B, and that we have reported in last year. So this is just synthetic manipulation we have followed. In the presence of 4-normal HCl, one can deprotect this acetate as well as this ketone protection, and then demethylation using sodium ethanthioate lead to the first total synthesis of silazine B. Now moving ahead, if you look at thionide adduct J, this is one of the complex natural product isolated from, by looking at this name, you can think that from which country it has been isolated. These are isolated from Taiwan, and a species called Taiwania cryptomyridialis. And now 1996 and 1997, a number of complex tetraterpenoids have been isolated from this species. So this is having some biological activities, and we thought that this could be a very nice structure where actually we could contribute and show our capabilities. Now looking at this particular molecule, you will see that there will be 12 stereogenic center. Now this kind of butane ring actually gives us the retrosynthetic hint what kind of strategy we should follow. So what we followed a nature-inspired 2+2 cycloaddition strategy. So therefore, we broke this particular cyclobutane, and that gives us the simple structure like this. Now, if you look at the cyclohexene, that would gives us the another strategy, that is 4+2 cycloaddition. Now, if you break that also, you will see two simple unit. One is abioabutane as well as another is transoxic acid. Now, we thought, can we synthesize this from this quinol? Yes, it is possible because quinols are very easy, easily oxidizable to quinone. But what we thought, can we develop a common strategy for this kind of, both kind of diterpenoids? And so what we thought, this abioabutane type, skeleton can be obtained from abutane type of skeleton. So you can see that this particular carbon is actually here. Now one can do this ring contraction to get this carbon down, and one can think about the Wolff rearrangement to do so. And also one can do this oxidative cleavage of this aromatic ring to get this. So in practice, what we do is, we have seen that by taking these two substrate and using europium FOD, in fact, we can get a pair of regioisomers in the ratio of 1.5:1. These were not separable. However, DMP oxidation gives us the separable these two regioisomer in column chromatography, and from there we could able to do the first total synthesis of thionide adduct I and H, just demethylation from this enol ether as well as methyl ester. So with that in hand, the last step was the 2+2 cycloaddition, and in fact we could see that 93% yield could be obtained from this reaction to get a single isomer, and which has been characterized through X-ray analysis. In 1 minute, I'll just show that thionide adducts as the another class of natural product, but the only difference here is that earlier case, this was a 5-member ring, and where one carbon was appended, but in this case, it is 6-member ring. So by looking at this structure, now one can think that if you take this kind of dienone, it can act as a dienophile, which can do a Diels holder with this electron-rich diene, gives rise to the synthesis of this. Very recently, what we have seen that by taking this and this, now we can hit, in this case, actually negative 78 degrees centigrade, what you could see that it could give only one regioisomer, and now this kind of alpha hydroxy ketone is actually in tautomeric form. So this is the only regioisomer we are getting, and this is not formed. So this work is still under completion, but what we could see that an X-ray crystal of this complex structure also showed that all bond connections are actually in the right place. So with that, what I'm trying to conclude is that before we start a synthesis, we try to follow the biosynthesis of such complex target, and we try to always follow nature, because nature has been the inspiration for everything, not only science, engineering, everywhere. So therefore, we should look at nature with respect. Not only chemists, biologists, everybody must understand what are the biological processes happening in the biological system. Of course, asymmetric catalysis is very important. Nature-inspired pericyclic cascade has been, the inspiration for many discoveries. With that, I would like to thank Director of IISER Kolkata as well as IISER Bhopal, because few of the projects started actually IISER Bhopal. And I'm also thankful to ACRC, ACRB in the form of CRG, SCP, STAR, CSIR, DAE, MOES, STAR MOE, and of course, Industrial University for the nice opportunity. I'm thankful to all of my students who have completed degree as well as who are continuing their research. Thankful to all of my collaborators, and many thanks for your patient listening. I will be happy to take any, if you have any question. Chairperson, please.
Yeah, thank you so much for finishing it on time and for this amazing chemistry that he has presented. Any questions? May I ask you a question?
Yes, please.
So, in all your synthesis, you've been so lucky, or it's just serendipity that you've got the structure?
I'm not lucky. What I'm showing is that only the fruitful result. Because you can see that I said, I thank IISER Bhopal. I left IISER Bhopal 6 years back. But still, there are some projects actually started in IISER Bhopal. So, in some places we are happy, but majority place we are, we are not. We are like ordinary people, like everyone. But positive result I'm showing here.
Because the crystal structure of these products are very, very key, right, for your confirmation and everything. So, yeah, amazing.
So that is the key point for our synthesis. Everywhere we try to end with X-ray. Otherwise, it's very tough to actually convince the reviewer, because this kind of complex molecule, this NMR data will be, many times there will be exchangeable proton that will make your life hell. So therefore, the X-ray is the best way to move forward.
Absolutely.
Yes, Professor Mahapatra, please.
Regarding atropisomers.
Yes.
So, like, if you get a like diastomers or enantiomers, if you get atropisomers, how it easy to separate out in the through silica gel chromatography or through HPLC, how to find out?
Yeah. So basically, it's a very nice question. Basically, the question is that in this particular case, like, this NN, these are atropodiastomers. Now, look at their NMR. The NMR will be almost similar. They will have very, very little change in the NMR. It's a very good question. Thank you. Now, the question is then how to confirm they are two different compounds. So, because I said this is only, NN, bond restriction leading to a pair of atropodiastomers. So, what we do is that these are atropodiastomers. So, therefore, they have different spot on the TLC. However, they are very, very close. So we have to do at least at least 2-3 times column chromatography so that we purify them. But after that, what we do? We run in HPLC. We use C18 HPLC because we start with enantiomer-rich compound. So therefore, these are enantiomers, there is no doubt. But we take C18 column and we run them, and they show two different peaks in the HPLC. Also with MS, also you can confirm they are having similar mass, but they are two different compounds.
So how do you assign the structures? So by XRD or?
The monomer has been characterized by XRD. Monomer.
Okay. That's good.
Professor Gharpure. Sir.
I was curious to know, how do you control NN versus CN and CC in this dimerization?
Yeah, thank you very much. So basically, if you look at now, monomer is doing NN versus CN and CC. So basically, the point he's asking is that if it is NN, probably they should be the kinetically controlled one. Now, they should again break and then lead to either CN or CC bond forming reaction. The CC would be probably the thermodynamically more stable one than compared to this. Now, when we have seen now copper mediated oxidative, aerobic oxidation, we could selectively get this kind of, NN bond formation. And there we could get only, 5 to 6% of digamycin C. Now, when we develop digamycin C, in that case, we have used Buchwald-Hartwig type thing. So there is no other possibilities. Now, in the case of CC dimer, like in the case of CC dimer, in this case, what we have seen, if you use PIDA and PIFA, hypervalent iodine reagent, 28 degrees centigrade itself, it gives only CC. So basically, 3 different conditions we have utilized to get them. Of course, the biomimetic approach must involve the formation of not only NN but also CN as well as CC. Currently, we are using riboflavin catalyzed photoredox processes, and where we can see that we are getting mixture of compound, and we are trying to control them. Thank you for this question. Thank you very much.
Professor Yadav.
No, I have no question to ask. Only thing is, I can tell for young people. You have seen since the morning, Dr. Tapas Kundu, then he's there, then Mahapatra, then Tantra. Those, fantastic amount of work, lot of efforts has been done. Only one thing I can tell you, my own experience for so many years. Difference between Ramchandran and Khurana. Both molecule made the same. One was collagen, one was that, and one gets Nobel Prize, one doesn't get Nobel Prize because the difference was only the kind of problem here chosen. Both are making chemistry wise same, but one gets Nobel Prize, one doesn't get Nobel Prize. That is one. Another is so much efforts gone on the natural product for home synthesize. So my advice, I'll tell you a couple of our role. There is a company called L'Oreal, okay? It a lot of nail polish, hair dye, all kind of personal sophisticated skin thing, lipstick, all kind of things. But they found that they were adding one compound which was isolated from the sea, okay? But they don't know what is the structure. But only difference was, by the way, don't take, I'm advertising L'Oreal. The only difference was, we use the L'Oreal product. It was homologating. It is there. That compound is there in our hair. It is nail, also in the skin. Everywhere it is there. And they were only using that, but it was homologation. So it was far better result. Not your hair it doesn't get thin, your nail polish doesn't get nail spoiled. All these things. So they did not know why it is happening. So they posed us the problem. We actually synthesized, that Gita. We synthesized the compound, we knew that exactly we know what it is. And I'm talking around 99, right? 99. We charged 2 crore rupees in 99. You imagine how much it will be today. And they paid 2 crores. Okay. And we gave the structure, we make the process inside. Now today, it is synthetically available. And I can also share with you, my wife never is for her nail polish, lipstick, nothing she gets. It's only L'Oreal one. So they are lifelong they provide to her. So that job is done. So that is one thing. It was a fundamental basic work. We total, like Mahapatra said, so many isomers synthesized. Like that we synthesized and we came out exact structure what it is. But fortunately we were lucky. Only we made 2 out of 8 possible isomers. And in those 2, one was there. So we don't have to make another extra 6. And similar, I'll tell you one last evening, I'll give example. One camptothecin. Nobody will dare to go for total synthesis. Camptothecin is isolated from a plant, Camptothecia. When it is almost 60, 70 years old, you have to cut the plant, you have to take up the hardwood. And once the person works, they get a lot of allergic reaction and all. It is a complex molecule, it's not easy. But we made 16-step synthesis, and Dr. Amaros makes it, and it is cheaper than the natural product. And he makes a lot of money, around 100 crore. And since a monopoly, it's almost 60 crore is net profit out of that 100 crore. So what happens if we are so much efforts you are putting? Okay, I'm talking to you. There are natural product. Directly they are using our industry with paying very heavy money. Can you think of, instead of this kind of synthesizing complex molecule, other than your PhD, no use? Can you put your PhD problem on those kind of molecules? And I'll give you example. There are many molecules. which are our alkaloid, opium derived from opium alkaloid. If you can synthesize and commercialize some of them, it will be very good, because there is a lot of leakage goes out, drug abuse and all kind of. Can you make it in the lab, and can we? So there are many natural products which can be synthesized and made cheaper than this. Similarly, there are, one example I said prostaglandin. If anybody will say it is very difficult to do, but it is not difficult. Mahapatra himself is here. We did together transfer the technology, and today it is being used. Misoprostol is done. Like RU486, we are looking at China. It's a semi-synthetic one. Can you take that challenge, make it commercially available, cheaper rate, so that our India is a amount of demand it is there. And you'll get money. So why my thing is, why I want to say, please choose proper molecule. You will become rich. If nobody, you start after retirement, Agramar, I started, can be done, okay? So this is kind of things I like to suggest to younger. Look for a proper problem, proper goal, what you want to do, okay? There are many, nature makes so many compounds. It's not that take any natural product and start making it. Make for a purpose. That's what I wanted to say. So that, keep in mind. Thank you very much.
Yeah, thank you,
No,
sir.
there is no criticism, by the way. Don't take it negative.
नहीं नहीं वो बहुत अच्छा सर आप टारगेट दीजिए सबको थैंक यू सो मच इट्स वेरी वेरी नाइस थैंक यू campus is one of the target right thank you sir
Thank you, sir. I request Professor Sivapriya Kirumbakaran to felicitate Professor Alakesh Desai. Now I request Dr. Sudipta Basu, Associate Professor from IIT Gandhinagar, to felicitate the session chairs, Dr. Vinay Kumar Srivastava and Professor Sivapriya Kirumbakaran. We'll begin with session 7 after a short tea break, and everybody is requested to be back by 12:20 sharply.
Sending later only, but I want to check whether everything working or not. Shall I keep it here? Is it okay?
We request everyone, there are exhibits by Cadila Pharmaceuticals. Kindly make it convenient to visit those exhibits as well. Thank you.
Welcome back, everyone. Now we will begin with the next session. For this, I would like to invite Professor Devendra Mahopatra and Professor K. Srinath Tiwari to chair the session.
Look like this. Hello. Volume. Hello. It's looking good. Can you hear? मैं अभी बंद कर देती हूँ उसको.
Good afternoon to all of you. So this is the seventh session, and our first speaker will be Dr. Sivapriya Kiruvakaran, which is a professor at IIT Gandhinagar. And she earned her PhD in 2007 from IIC Bangalore under the guidance of Professor S. Chandrasekaran. And she has been having more than 20 years of research experience and bestowed with a lot of awards. I can read out few. And first one is the Alumni Excellence Award in 2024, CRSI Silver Medal 2024, Dr. R. Gopalan Endowment Prizes 2022, Future Leaders in Chemical Biology 2023 by the Royal Society of Chemistry. And she is the founder and chairperson of Propulsive Therapeutics, a startup for innovative drug discovery. With a small introductions, I'd like to invite Dr. Sivapriya to deliver a lecture.
Yeah, thank you, Dr. Mahapatra. So I'm going to put my timer too. So today I'm going to talk about, I would request all the students not to have WhatsApp for next 20 minutes because you're not going to hear a brilliant lecture, but you might get something from my talk because as a student, what I have learned is what I am actually doing right now current in my lab. So first thing is that this is probably the most successful story of the lab, but this has happened because of lot of observation that Professor Santosh was mentioning in his talk, right? How my PhD students, the first batch of PhD students, were able to observe things that was happening or that was not going the way they want. So here I'm going to talk about a kindness, which was a new kid on the block when we started the lab 10 years ago. But now this is one of the major projects in the lab. I'm not going to talk about the other projects which we are doing. We are basically a kinase drug discovery lab, and we work on DNA damaging kinases, which comes to play a major role in cancer therapeutics as well as cancer progression. So once your DNA gets damaged by chemotherapy or radiotherapy, what is the repair mechanism that happen biologically, and how that repair mechanism can progress the cancer as well as to stop the cancer. That is the layman words that I can put it. So I'm going to talk about this particular kinase, Tosyl-like kinase, how we started this project and what is the importance of that for the next 18 minutes. So as you can see here, many of you know, and all of you probably know, the cancer progression happens in this particular manner. And once this progression happens, only at the several, advanced stages, it is diagnosed. That is also because that lack of awareness in cancer diagnostic also. But you can see there are several hallmarks of cancer. I have only few of them depicted in this picture. But what happens when a small one cell actually divides so fast and then becomes a cancerous cell, right? That there is not only one single reason that you can give for the progression of cancer, but there are several, like evading growth suppressors or signaling pathway malfunctions or mutations that happen. So if you see the global cancer incidence, all of us know that it's a huge potential for drug discovery, but it's been happening for several years. Any of the students, can you tell me when the first documentation of cancer was given? Any guess? Except sir. So, yeah, if nobody answers in the student side, I will ask her. Yes.
First detection of the cancer was found in the mummies.
Correct.
In the Egyptian mummies, which they detected the
Yes,
cancer bone.
absolutely.
Yeah.
So, but it was documented in one of the very old Greek literature.
Yes.
Yeah, in 300 BC. Can you imagine? So it's not that new disease, right? Now we are talking about cancer therapeutics and everything, but this is not a new disease that it has just come a few years ago, and we are all dealing with it. So, but you see the incidences. You see that it's probably the second or third number killers. Okay, in terms of the number of patients that we lose every day. Okay. So here I'm going to talk about something called kinases. If you want to do a phosphorylation reaction in the lab, and if you want to take ATP, you take ATP in a round bottom flask, you set up a reaction, you leave it, nothing will happen. Okay. So, but this particular protein, I think majority of your chemists here, like me. Okay. So that's why I'm giving you a simple introduction and some introduction about kinases. It takes one nice phosphate from ATP, and then the kinase actually takes it and puts it on another substrate. This could be another kinase, it could be another protein, it could be a small peptide, it could be anything. But this is a simple reaction that it does. Okay, so a kinase is a protein which takes a phosphate from ATP. If you want to do this simple reaction in your lab, in chemistry lab, it's very, very, very difficult, right? But it does it with so much of ease. That is why, okay, please, you don't change. I'll change by myself. Okay, it's not happening. Okay. So we have about 518 kinases in human kinome. So that means all of these kinases does the same reaction, right? It takes either, so sometimes it could be from GTP. So it takes a phosphate from ATP or GTP and puts it on another substrate. But by which what it does is it makes a cellular cascade for signaling. If you see, anybody has seen New York Metro, how complicated that system is. If one train stops for a few seconds, then all the trains will stop. Okay, there will be major accidents can happen. That's why New York Metro system is supposed to be the most efficient metro system. So similarly, when you compare the complexity of kinase, which is part of this whole hallmarks of cancer. several cancers are reported that the kinase is overexpressed. That's what you will read. Okay, so this particular kinase is overexpressed, that particular kinase is overexpressed. You know why? Because if you see that kinome tree, which will come in the next slide, this is a network of all these 518 kinases in a very close knit tree. So if you see something is happening here, and that will affect here. Okay, so one kinase, if it fails to do some job, the other pathways will get affected. So it starts from the beginning of the cell. EGFR, how many of you have heard EGFR? Though it's a receptor, it's also one of the kinases. Okay, so it starts from that membrane of the cell till the nucleus that is present. It also presents in the DNA. So you can see from top to bottom, all this cellular signaling pathway is kind of nicely organized by the cellular signaling of kinases. So this makes the kinases more potential target for drugs wherever they are overexpressed. That means in many of the cancers, from lung cancer, breast cancer, skin cancer, or glioblastoma, one of these kinases is actually overly expressed. So TTK is one of the kinases that you have, is that kinase that, because the introduction section I missed, but TTK, I assume that there is also a kinase called TTK. But that is one of the kinases that after the TLK story, I'm going to tell you that slightly we are going towards glioblastoma. So the entire kinome, if you see, is closely knitted. That means if you want to do a drug discovery for kinase, this particular protein will bind to ATP or GTP. So when this binds, the active site of this kinase is going to be very, very similar, right? So if you want to target the active site of a kinase, it's going to have a lot of mutations in future, a lot of drug resistance in the future. So I'm going to show you how we have been able to figure out an allosteric inhibitor for this particular kinase called TLK. So this serine threonine kinase, when I mean, that means that serine or threonine in the next substrate gets phosphorylated by one kinase. So there are several types of kinases, tyrosine kinases. So these are the classification. So this particular tosyl-like kinase was first discovered in Arabidopsis thaliana, which is a plant. You can see the plant many places, and this is also a very important plant to study. Plant biologists actually like this. So this is discovered in human is called tosyl-like kinase. So this is there are two homologs, TLK1 and TLK2, and they are found to be very, very important in chromatin assembly, DNA repair mechanisms. Okay, and its activity is maximum in the S phase of the cell cycle. So as you can see here, once the DNA gets damaged in a cell, which could be single strand DNA or double strand DNA, these kinases come to repair this pathway, and TLK comes here where it gets it's phosphorylates. There are three important interacting partners: ASF1A, DAD9, NEC1, histone, and MK5. So I'm going to talk about these three substrates, the latest slides of mine. So it's a very important mediator for DNA damage and response pathway. And this inhibition, if you see this slide, so if you inhibit TLK, along with the DNA damage, okay, so if you do chemotherapy or radiotherapy, along with that, if you also inhibit TLK, probably that could be a nice combinatorial therapy for futuristic cancer therapy. So that is how, this is the background of why we started targeting TLK1. And TLK1 was kind of studied only from 2013, and Professor Benedetti is the first one that he has actually published a lot of papers on based on this, this strategy that I'm saying. So I'm going to talk about that, how we developed inhibitors for TLK1. And if you make inhibitors, if you don't have the protein, because I said 2013 only it got the limelight. So if you don't have the protein, then you will not be able to study that. So when I started the lab, I was very adamant, I can say that we wanted to develop this biochemical assays by ourselves. Otherwise, it would take ages for us to collaborate with a lot of people, and TLK was not very well studied protein, so you cannot get everything commercially available. So we have developed strategies for recombinantly purified TLK1 protein and purified both the full length as well as the kinase domain. And we have also developed cellular as well as, I'm going to talk about some animal studies that we have done, and now how the molecule is in the preclinical trial. So if you see from the bench to bedside, we say, right? So for last 6 years, we were being able to make the molecules which were potentially working with the purified protein, as well as that we could take it to animal level. Okay, so when we started this, we didn't have the structure of TLK. We didn't know how TLK is expressed. We don't know how the purified TLK will behave. We don't know anything about TLK1, but we know the importance of TLK obviously from brilliant biologists. And when we started this, there are 3 molecules that Professor Benedetti has published in this paper, where the particular molecule showed some kind of a phenothiazine scaffold, right? So this molecule attracted us. Then we thought, okay, so right now this is the molecule, but anybody know all of this molecule promazine is all these molecules are used as antipsychotic. But apparently all these molecules only has shown some inhibition towards TLK. So we took one of the scaffold and we thought we'll study how this molecule is going to behave in TLK. As I told you, we didn't know the structure. But what we did is we did an analysis and saw that this S-methyl group is important for the antipsychotic effect. So we thought, okay, so let's remove this and then extend this to have something like this improve the binding affinity. So we wanted to put something like this. Okay, so we just made this molecule. It might look very, very simple molecule, right? And then we made some varieties of this molecule. So my second student, Javina, when she joined the lab, she was given this because first year student, she wanted to make simpler molecules. Before that, we were making ATR, ATM molecules, which are pretty much complex. So when we made only first series of few molecules, and by the time we also had, okay, so you can just think about having a morpholin here. It's very simple chemistry. You don't need a lot of complex steps to make these molecules. And meanwhile, Siddhant, another PhD student from biology, joined. We said that we had several strategies tried for several DNA damaging kinases. Many of them were biggest failure. But one of the strategy which was failure for ATR purification or expression in E. coli, because you remember all of this protein are within our body. It's a mammalian protein, and you have to clone them in E. coli, which is not so easy sometimes. So many of the mammalian kinases we cloned before TLK was a huge drawback and a lot of challenges. We were not able to get even nanograms of protein. But for some reasons that when we cloned one of the strategies, TLK1B in E. coli, we were amazed to see that we've got really, really good amount of TLK1B, which is about 5 milligram per ml. So which is like, for a molecular biologist, this is really, really huge, right? But I'm not a molecular biologist. The people who are working with me, especially Siddhant, was extremely happy. And then we could able to do the biological activity for TLK1, because that's also very important. You make a kinase, as I told, the kinases can also get phosphorylated. That is what we call as PTM. So they can get phosphorylated, they can get any post-translational modification. So we wanted to see whether it's active or not, what we expressed in E. coli. So the kinase was very much active. And then we immediately, what we did a ATP hunter assay. While doing this assay, we could figure out this J54 and J56. These are the 2 molecules which showed really nice potent binding to this kinase. This is with the purified kinase. Okay, so we wanted to see. At that point, we submitted a couple of papers, and Professor Benedetti contacted us because we were probably one of the working with the chemistry of making these molecules for TLK inhibitors. So at that point, he had a strategy, or he proposed a strategy that androgen deprivation therapy is a frontline treatment for prostate cancer. Okay, and most of this prostate cancer deaths, okay, result from progressive failure in this standard therapy. So this ADT results in overexpression of TLK. So when overexpression of TLK is there, and when we can inhibit TLK at that point, okay, then that could be a combinatorial therapy, nice therapy for frontline treatment for prostate cancer. This is what the plan that we had. This is the axis that we wanted to follow. So we sent that molecule. So we had 2 molecules, as you see from the last slide. But one of the molecule, we have not taken it further, because you can see that instead of morpholin, we just had a methyl there. That is a very, very small molecule to be developed as a drug. So we took that morpholin molecule and sent it to Veridity's lab. As you can see here, in 51 days, okay, this is a control. This is the biclotamide, which is a FDA drug for prostate cancer. And this is a molecule that Javina made. We name as J54 with her name. And the combination, you can see that the complete, this is in the bulby mice. Okay, so the prostate cancer is induced and then we studied it. So in 51 days, as you can see here, the combination works really, really well. Which was extremely rewarding for us because it is just 3 or 4 years we could be able to do such a study in animal. So this was very, very, very much encouraging. But in drug discovery, not everything is your molecule. The molecule that you made might not have the pharmacokinetics data that you want to have. It might not have the proper toxicology profile. It might not have proper barrier. Okay, so it might not have metabolic stability. So we wanted to be assured that we are ready with the next generation of molecules. So the next student, when she joined Delna, so she started actually synthesizing or strategically design the molecules which could have better metabolic profile. So it's not J54 didn't have metabolic profile. It had, but it is not enough to take the molecule to clinical trial or preclinical trial. So it had some drawback. So we wanted to be ready with the next generation of molecules. So, but we didn't change anything in the phenothiazine scaffold till for the next generation. So she made a variety of molecules with, again, with simple chemistry. But what we could able to find out is by the time we could able to build a molecular simulated structure of TLK1 kinase domain. So that helped us a lot to figure out where the molecule could be binding. Okay, so in this is actually not the crystal structure, but most probably when I talk next time, we will probably have the crystal structure. So as you can see here, the most important binding sites for these molecules, we kind of theoretically predicted what could be the binding sites. And as you can see here that this particular portions are very, very important for the binding. That we can also see that in our cellular data, that when you have the interaction with the Hnd region with the oxygen like this, any place, but if you want to change sulfur to nitrogen, that completely lost its activity. So we didn't know that what was the importance of sulfur. Probably while having the crystal structure, we will figure it out. Nitrogen, it lost activity. Oxygen, it lost activity. So we did a lot of SAR from this scaffold because we wanted to make sure what is actually is needed for a drug-like molecule. Okay. So then Delna started synthesizing a new scaffold. By the time there are several other inhibitors which has come. So right now we have a molecule which is based on indole scaffold. And then we have made several series of the molecule. But now you can see that we are studying with NEC1, ASF1, histone. So for all the substrates that we have in the lab, and actually is a peptide, and ASF1A is a small protein where TLK goes and phosphorylate. So we have this also cloned. So when you have this molecular biology in-house and cell biology in-house, I think that gives a lot of importance for your work also. It's not just anti-cancer, right? You are proving that your mechanism by studying it in your lab itself. So this is the last bit of slides that you can see that this molecule 5N, which is indole-based scaffold, is kind of acting on this particular cell lines. LN-cap cell lines are the prostate cancer cell line in a really good manner. So it has, you can see that the next slide, that the apoptosis assays that we actually perform, that this molecule is very potent to TLK. So you can also ask whether this molecule is very specific to only TLK or it is also inhibiting 500 plus kinases. So we also do something called a kinome assay where 400 plus kinases are plated, and J54 actually inhibited only 2 kinases in the entire kinome, which was TLK and then TTK. So now we are progressing this particular work towards the next generation of TLK inhibitors for animal model. And also some of these molecules is showing real good potency with a lot of modification, which is structure guided for us in the lab for TTK also. So we made a library of small molecules. If anybody is interested to study for TLK, because from the morning we heard brilliant chemistry So if you think that any of your scaffold probably be a kinase inhibitor, please reach out to us. And then J54 was the first lead molecule from the lab, and now we have 2 generation of molecules with a better pharmacokinetic profile. And we also have a preclinical candidate now for prostate cancer, which we will probably be able to take it to for phase 1 clinical trial. And the structure is in process. And not only TLK role in cancer, by studying TLK role, we are also able to develop really nice mechanism and cellular assays for other kinases, which gives us a lot of challenging kinase drug discovery into a cellular level or the biochemical level understanding, much more better understanding. So I thank my collaborators, main thank to Professor Benedetti because he directly reached out, and 2 of my students who are postdocs with him, and then they did very well. And thanks to IIT Gandhinagar, Ramanujan Fellowship for bringing me back from US and all these funding agencies. And big for the startup grant that was mentioned. So Javina Siddhant are the ones who started this work, and they have been extremely lucky to be on the shoulders of my giants of my first batch of students. So I thank them a lot because they built the lab from scratch. And these are the stars right now in the lab. Gaurav, Deepika is here, I think, Delna, and then Banu. So they are the ones that actually on their shoulders I'm standing and presenting this talk.
You were aiming to have an allosteric inhibitor.
Correct.
So since you can model it, do you have any idea other than active site where it is binding?
Yeah, so when we do the docking, the model structure.
Docking what you showed, that is with the active site.
With the active site.
So
What
my
we-
question is, your aim was to go for, and your kinome analysis, 400 kinome analysis suggests that it is not an active site binder.
Correct, correct.
So you might be knowing where in the protein it is going. You have an.
So we have a, I'm not 100% sure about it, but with the model structure, we also have an idea that where particularly that allosteric site could be in TLK. So we have the model of KD, kinase domain, as well as the full protein. Maybe in my next couple of months or few months after, I can talk about that. But we have a rough idea where it binds.
So actually,
which is not
if
very
you have a
close to
point
the HB.
mutation in, since
Yes.
you have such a wonderful
Yes.
system,
Yes.
if you just based on that, if you have a one couple of point mutation, which is really touching the
Yes,
compound,
yes.
and if the inhibition goes,
Yes.
then much before the structure comes, it will be
Absolutely
ready.
correct. So that's what I'm expecting my the PhD students who are doing in biology to do.
Very
it.
good. Very nice work.
Due to pursuit of time, so let us thank Dr. Sivapriya for a fascinating lecture.
Thank you so much, ma'am. I request Professor Devendra Mahopatra to felicitate Professor Sivapriya Kirubakaran.
So our next speaker is Dr. Raji Reddy. So he's my good friend, so another eminent organic chemist from CSR ISCT Hyderabad. If I'm not wrong, actually, I know him from 1997, and he's earned his PhD from CSR ISCT in 2002, followed by postdoctoral research from USA. And he is then joined as a principal scientist at SIDD Life Sciences before joining CSR ISCT in 2006. And his research focuses on organic synthesis and API process developments. He is, I can say, is a technocrat, and he has developed many process for drug molecules like favipiravir during COVID time, remdesivir, pregabalin, eribulin mesylate, and TLR7
agonist used for covaxins. And he has a lot of awards and rewards, and he is the fellow of NASI and EFTAS. EFTAS is our Telangana Academy of Sciences fellow, and he has received the CSR Technology Award 2020-21. 2 times he has got, and NASI Reliance Platinum Jubilee Award 2020, CRSI Bronze Medal, CDRA Drug Research Award, and he is, to his credit, having more than patents, and he has so far given more than 35 PhD students. And with these small introductions, I would like to invite Dr. Raji Reddy to deliver his talk.
So very good afternoon to you all. Thank you, Dr. Mahapatra, for your kind introduction. So I would like to thank the organizers, particularly Professor Yadav sir, and also Vishnumaya and other team members for inviting me to for this conference to share some of our work, what we are doing at CSIR IICT. So before going to work, just I would like to thank Dr. J.S. Yadav sir. You know, for, when I joined student, also he was the head of the department signing on my application, and again when I joined as a scientist, he was the director, and he gave me opportunity as a student as well as to start my independent career at CSIR IICT. So from there, we started our journey. So what, before going to today's topic, this functionalization of alkynyl precursors, how they are helpful, particularly in promoting radical functionalization reactions to bring the molecular architectures in a one-part manner, in a cascade manner. So before that, just I would like to show what we do at CSIR IICT for the sake of interest of students. So as we are CSIR is I is one component is majorly for industrial collaborative research, where we develop some synthesis of new chemical entities toward drug discovery as well as process development for the existing drugs to make it more efficient manner with respect to cost as well as some other issues if there are any complex issues. So after this achievement, recently we have made the process for bempedoic acid, which is already in the market drug, and where the key challenge is to introduce one of the carbonyl moiety. The present process involves the usage of TOSPIC, which not only cost contributor, but also it generates a lot of waste. To just introduce one carbon, we are using this precursor as a reagent. So here we developed this, and presently we are in the process of transferring, and recently also we have accomplished one of the carba sugar in ticagrelor tablet, which is again a key intermediate and also major cost contributor in a stereoselective manner we have prepared. So in addition, we also do some of the basic research with respect to bioactive natural product synthesis, and also by looking at these scaffolds, we also we do revise the stereochemistry, particularly the hematopolite and other natural products. And but today I'm going to talk about dearomatization reactions, particularly how we arrived at that particular project. So initially we started the simple starting material propargylic alcohol as a precursor, because which is very versatile handle to generate the nucleophilic substitution, and this alkyne is the handle for annulation reaction, so that we can generate a large variety of heterocyclics and carbocyclic related compounds. Here if you look at it, these are some of the selected publications, but here if you look at it, the red portion is always being a part of aromatic ring. So aromatization is the driving force in this particular project. So inspired by this project, later we started a different precursor. It's a Morita Belisilmanadax, again which is a propargylic alcohol. But when we looked at the literature at that point of time in 2011, so we could not find an example of this particular Belisilmanadact generated from propylaldehyde, although the reaction was discovered in 1972. So we ventured into this, making this particular adduct, and from there again it gave a nice handle because it has a multiple functionalities: hydroxy, olefin, ester, and alkyne. Having these functionalities, we used this as a nice precursor to generate a large variety of heterocycles as well as carbocyclic molecular frameworks, and this is became again a good project for us continuously for last 10 years we are working on this. So having these two projects in mostly aromatization driven projects. So we thought, can we do some de-aromatization reactions, particularly by starting from aromatic ring, and where we started in 2017, we searched complete literature, thorough literature search, and made a review article, and then we most of the times either this methoxyphenyl ring is more susceptible for de-aromatization reactions, and we started from this N-propyolamides and biaralanones. But today what I am going to speak is not this particular methoxyphenyl, but the methoxybenzyl and also indole dearomatization, how these two aromatic rings are playing key role generating the quaternary carbon centered molecular architectures via dearomatization cascade acylation. So if you look at this particular framework, there are few examples well studied towards a monocyclic ring formation leading to spirocyclohexadienone. So this by either a electrophilic cyclization or a radical promoted annulation will leads in the presence of oxidant, leads to dearomatization and gives this particular reaction. But what we are interested is, since this is a symmetric molecule having two double bonds, can we make it in one part reaction to use these particular handles and do a selective cyclization which can lead to a tricyclic moiety with a nice quaternary carbons. So when we looked into literature into this particular aspect, not from this exactly moiety, but the generated from propylamide, and already there is an N-alkylated functionality which undergoes further Michael addition kind of reaction, and there are a couple of papers reported in a symmetric fashion. But this is, please remember that this is a pre-installed functionality on the nitrogen. What our interest is, since we identified this as a key precursor alkynyl, since it is generating an introductory through a radical or other electrophilic reagent, and our interest is to make from this linear molecule to a completely tricyclic molecule in a one part manner, where the incoming group is participating in the Michael addition rather than the pre-installed group. So here we could identify nicely this 1,3-dicarbonyl as a radical precursor, and in the presence of steric ammonium nitrate it generates a nice radical and then reacts in a cascade manner. It forms 3 carbon-carbon bond formations, and these are the key structural frameworks in various bioactive natural products. And these are the structures we can generate the framework. We are not going to complete total synthesis, but it generates the real frameworks with multiple handles. So how it works? So whenever we start a method, initially we need to identify the optimal suitable reaction conditions. Again, we've screened few oxidants and solvent systems and found that steric ammonium nitrate in methanol at 0 degrees was the optimal condition to generate. Interestingly, this reaction completely happened in one part. Initially, we thought we need to add some base for Michael addition reaction, but we directly obtain this tricyclic product, and we also verified that what is the incoming, the source of oxygen, because we have already a methoxy group, whether this oxygen is from this methoxy or external oxidant, and this again through O18 labeled experiment, we could confirm that it is from water, and again it is a radical reaction. By adding some of the scavengers, the reaction got inhibited. So if you look at the mechanism, the proposed, it generates the radical here, and it regioselectively attacks on this particular olefin and generates again a tertiary radical here, and which undergoes into the ipsocyclization reactions from this activated methoxyphenyl ring, and then addition of water molecule onto this oxonium ion, and then elimination of methanol gives to this symmetric molecule. And from here, it is a within the same reaction, one part it happened this very selectively, stereoselectively Michael addition type of addition, leading to this particular tricyclic molecule. So having these established conditions, we'd like to see the selectivity as well as the scope of substitutions from a different variety of substrate perspective. So initially we explored 1,3-dicarbonyls where various ketones were studied starting from alkyl to aryl and substituted aryl, and later we changed to ester and also further extended ester carbon skeleton also was found to be participate through radical generation. And again, simple 2 diketones, not only ketoester, but even diketones, as well as the substitution effect, this particular on the olefin, this R3, not only methyl, it can also be hydrogen or it can also be aryl group. And again, we also some of the substituents on the nitrogen as well as on the methoxyphenyl ring were also nicely studied. So in all the cases, we could get the product, although it's a racemic, but it is a single diastereomer. So we also explored whether only methoxy group works or any other. When we have hydrogen, it gave very little yield, 12%, but when it is a fluorine or OAC group, we could get a reasonably good amount of yield. But when it is electron withdrawing nitro, only trace amounts were we could get. So having the tricyclic moiety to show just the synthetic feasibility, a gram scale reaction followed by post synthetic transformations of this scaffold, whether this scaffold is stable to when we do further functional group modifications like reduction of the double bond as well as keto reduction, and also boron Michael addition onto this, and the skeleton found to be very stable. So now this concept we are further extending to a different radical source. This is carboxylic acids, where through decarboxylation the carbamyl radical will be generated, and then after the ipsoannulation reaction, now nitrogen is acting as a nucleophile for aza-Michael addition reactions. Now this again the similar manner, it generates a different scaffold having a nitrogen in on the ring, other ring also it is again it's 6-membered ring formation. So it is exactly like the previous work, but in this case particularly one thing we should note that the DBU addition is mandatory for the Michael addition reactions. Like in a previous case, it directly led to the tricyclic product, but here we need to add DBU, but again it is a one-part reaction. So this particular work is presently being continued to final substrate scope exploration, but we already explored some of these different carboxylic acids having different allyl groups and as well as alkyl groups, even the furyl. And in one particular case, especially when you have a substitution on this methoxy phenyl ring, it gave a mixture of isomers, where if you have this particular diketo here, the Michael addition participates, but when it is a ortho side oxidation, then there was no further Michael addition. So we isolated this particular product, and again further the R2 group on this particular alkene, again either hydrogen or phenyl group was also possible in the same manner. And further we found that the substitution effect on the nitrogen, either alkyl or benzyl groups or sulfonyl is good. But when we have, of course, this is expected if you block this nitrogen with another aryl ring, there is no further cyclization. So it goes through this intermediate involving a desymmetrization reaction, one part. So further, having this particular skeleton, we just try to extend by taking this ketomethylene as advantage. Can we build a few heterocyclic on this particular ring so that we can further make a complex molecules with including inserting the nitrogen, and we made one analog now with a pyridine ring construction on this particular moiety using this method. So little bit the next one, so far we have studied methoxyphenyl dearomatization, how it can generate the complex molecular scaffolds. And now with respect to indole, so there are a lot of indole natural products having this particular ring is dearomatized, 5-membered ring, and plenty of natural products. Morning, Professor Santosh also showed one of the similar kind of indole dearomatized natural product. And here, if you look at the literature, most of the time the dearomatization, either you use directly oxidation in the presence of various oxidants, or there is one 4+2 annulation from this lithiated indole to generate this particular moiety. So what we designed to have a aza enriched frameworks, indole linked with this benzimidazole ring, and again we made a handle of acrylyl ring so that this alkene is a handle for radical addition, and then it promotes the further cyclization reactions. So here we use the radical precursor as a carboxylic acid in the presence of oxidant, and what we observed is there are two different products based on the substitution present on the C2 position of the indole. If you have a alkyl group, especially methyl group at C2 position, it underwent a dearomatization reaction with a hydroxy as a nucleophile addition incoming group. But whereas if you have a aryl group, if X is equal to aryl group, this group migrated to the third position through again involving dearomatization reaction. So just to show this initially, again as usual, we did some experiments to optimize the conditions. We found that this potassium persulfate in the presence of silver carbonate in acetonitrile water gave the this product in 85% yield. And again, we proved that the source of oxygen is again from water, this incoming hydroxy group here. So having this hydroxy at this quaternary center, you can see that there are two quaternary centers and one particularly hydroxy. There are a good number of natural products with this hydroxy at this position. So that made us to further explore. Before that, we proposed this mechanism. After decarboxylation, the tertiary alkyl radical will be generated, which regioselectively attacks here again the stable radical here, and this which participate indole dearomatization reaction with this particular ring and generates this radical. So from here, the single electron transfer in the presence of oxidant generates this carbocation benzylic. So from here, the reaction takes two different pathways based on the substitution. If you have a methyl group at this particular position, which the nucleophile from water hydroxy addition here generates this type of product. But if you have a phenyl through this Bach deprotection, maybe because of steric hindrance, and this aryl group migrates before water addition, hydroxy addition at this place, the phenyl group migrates to this position, leading to this product via decarboxylative of this Bach deprotection. So having this proposed mechanism, we explored a few different radicals, including tertiary carboxylic acids, as well as secondary carboxylic acids, cyclic, as well as acyclic radicals, and also effect on this particular, some of the substitutions on the indole ring as well as benzimidazole ring were revealed that they are nicely suitable for this annulation reaction. The nitrogen substitution, whether it is tosyl or acetylated compound, even this particular ring, when you don't have any substitution at this SIDD position, obviously it is simply annulation without any dearomatization reaction participates in the C2 cyclization reaction. So this is the scope for arylated C2 arylated substrate leading to this migration based product formation. In this case also tertiary carboxylic acids as well as secondary carboxylic acids, including acyclic and cyclic, as well as the few different aryl groups were also studied about the suitability of that migration. So having this hydroxy handle, again, you know, to show this stability as well as the reactivity of this particular molecule, how we can do further transformations on this. So we did initially alkylation, proporgylation in the presence of base, and later having this benzylic hydroxy group in the presence of Lewis acid, the carbocation generation, a variety of hydrogen, azide and indole nucleophiles we could substitute, and in all these cases we found that the single diastereomer formation. So here little bit switch from dearomatization reaction. This is one of the recent project, still it is ongoing. So having these alkynyl precursors are very nicely helping for the radical promoted reactions. So this one of the reaction got our attention, Dowd-Beckwith reaction, which is a radical generation from the bromo followed by this having alpha carbonyl group leads and cyclopropionation, and ring opening gives the ring expansion products. So by looking at this, Durga Prasad from IISc Bangalore also published a nice review by extending this bromo to various other activators here and how the ring expansion takes place through this kind of reaction. But what we thought, having this particular precursor, whether can we regioselectively add here a radical and then it generates a stable radical here, and whether this is possible through this mechanism for ring expansion. And when we look at the literature through hydrogen radical, there is one method reported, but our interest is to add the additional radical. In the case of aldehyde, we interestingly got a very nicely bicyclic product. Apart from the ring expansion, we also got further cyclization having this keto, because it is a acyl radical from aldehyde. It underwent cascade ring expansion as well as another annulation reaction under metal free conditions. So we could able to make these functionalized tetrahydrobenzofuranones. Again, I am not going into the optimization details, but here what is happening here, once you have a acyl radical, it adds on to its stable radical generation and cyclopropionation. Once the ring expansion takes place, we have a radical here again which takes the lone pair of this leads to further cyclization through this oxonium ion. So again, the substrate scope is well here, a variety of aromatic aldehydes, including donating and withdrawing groups, and various heteroarylic aldehydes underwent this both ring expansion followed by annulation reactions. We also studied the substitution effect on this alkenyl group, further ring expansion, not only from 5 to 6, but from 6 to 7 with different groups were studied, and the fused cyclopentanone ring, indanone, also underwent to give this particular chromone products. Further again, this chromone to one more ring expansion, 7-membered, and of course further 7 to 8, we could not get this ring expansion. So here again, having this scaffold, we showed some of the synthetic transformations having this particular ketomethylene further constructed indole, as well as the reduction of this double bond, and also reduction of other functionalities were demonstrated on this particular product. So I hope I would able to show that how this alkene functionality can be utilized through radical promoted reactions, further cascades annulation reactions either via dearomatization reaction, and one last recent work as a ring expansion product through Dowd-Buckworth reaction. So with this, I must thank all my students. Because of their hard work, I could present the results here, some of the results, and today I presented Sai Prakash and Arti Kumari and Srinivas and Ajay Kumar's work. I also thank all the funding agencies for their constant support to do this basic research, and I thank you one and all.
Only one question will be allowed, so.
Great talk, Raj, as usual. I was just curious to know that did you try Favorsky on those angularly fused system? You had a 5-5-6. If you do a Favorsky rearrangement with the ketone product, you will do the ring contraction and you will get the
last
but one dearomatization
Okay.
with the cyclization happening. You had a nitrogen 5, carbon 5, and then the 6-membered ring coming from your paramethoxy
Yes.
phenyl ring.
No, I did not look into that. Maybe
because that could be a good possibility for total synthesis with the nitrogen in one of the heterocycles is what I thought.
Okay,
right.
Thank
And
you.
one quick last
Yeah.
one, the radical cyclization that you showed for the dihydrofuran last part.
Yeah.
So in the mechanism, what I noticed is that radical is reacting with the carbonyl and then generating a carbocation. But it will be generating first a radical and that will have to undergo oxidation.
Oh, okay, this mechanism.
Mechanism.
Yeah. See, I think still we have to look into that. Yeah, this one, right?
Yeah.
Yeah, here, yes.
D to E.
Yeah.
There is oxidation there.
Yes. Further.
Okay, so big round of applause actually for Dr. Rajesh Reddy for excellent lecture.
Thank you, sir. I request
Now the session will be chaired by Professor Tiwari. Next, two people, okay, speakers.
Professor Devendra Mahapatra to felicitate Dr. C.S. Raji Reddy.
Good afternoon. Our next speaker is Professor Chandrakumar Appaya. He is a professor, Department of Chemistry, Indian Institute of Technology, Gandhinagar. Dr. Chandrakumar Appaya is working in the area of asymmetric organocatalysis and asymmetric synthesis of bioactive natural products and drugs. He began his career as assistant professor at IIT Gandhinagar in 2013, later promoted to associate professor, and now he is full professor. Before joining IIT Gandhinagar, he pursued his postdoctoral research with Professor Ronald Breslow at Columbia University, USA, and Professor Brenner Moyer at Brooklyn College at the City University of New York. Professor Take Naka at University of Miami. He did his PhD from famous Indian Institute of Science, Bangalore, under the supervision of Professor K.R. Prasad. With this, I invite Professor Appaiah sir to deliver his lecture on the title Development of Chiral Organocatalyst and Their Applications. Please round of applause for Professor Appaiah.
Thank you for the kind introduction. Good afternoon, all. I thank Professor Vishnu Maya and Professor Yadavji for the kind invitation to present our very recent interest on the development of chiral organocatalyst and their application for the asymmetric synthesis of bioactive molecules, including pharmaceutical drugs. To introduce chirality, there are many speakers already spoke about that. I just want to remind this morning, Professor Mahapatra mentioned the thalidomide stories, and in principle, enantiomers have no difference in laboratory techniques, but a lot of difference in biological system. So there are very much urge in the pharmaceutical industry to make chiral compounds. And to make chiral compound, there are so many methods known. I would prefer to say catalysis is the best method because of the use of small amount of chiral source for this catalysis, for the synthesis of chiral compounds. Organocatalysis kind of established, I would say, in the recent time. And this has, you can use chiral amines or NHC or hydrogen bond donor or even mixed of either of this would give a very good scaffold to catalyze the reaction. It's after all, organic molecules still can do better work to make organic molecule as a product. And if you look at the activation mode of asymmetric amine catalyst with the aldehyde or alpha, beta, gamma, delta, or unsaturated aldehydes, they are usually condensed to form iminium ions, which are actually electrophile, and it could actually react with the nucleophile. But in situ, it can lose the proton at the terminal end that can convert into nucleophilic species that is a enamine intermediate by the loss of water. So this all species exist in the solution. Depends upon what reagent you are throwing at it, you could actually react either electrophile or as a nucleophile. So if you take this trienamine intermediate and you just throw electrophile to it, it in principle can go to this epsilon position, and suddenly it become an electrophilic intermediate. So you could add nucleophile to it. It can be, again, nucleophilic intermediate. You keep adding electrophile and nucleophile in a cascade fashion, you should be able to generate all these 5 centers of the unsaturated aldehyde with a chiral entity in a one-part reaction with a single catalyst. Apart from that, there is a very good advantage about this catalyst. You can do this catalysis in water, and it is environmentally very friendly. There are multiple chiral reactive centers exist in the system. You could actually do multi-component reaction. Of course, they are cost-effective as compared to precious metals. Despite it has numerous advantages, industry is not almost picking it. And there are very few examples here and there that industry using this catalysis. One of the reasons is that higher catalytic loading of this catalyst. Catalyst decomposes during the reaction, and it's very difficult to separate after the catalysis is done because catalyst is also an organic molecule. You have to find a way to separate them, and it is advantage we have several reactive centers, disadvantage that you will get a regioselectivity issue during the process. So usually in this iminium-enamine catalysis, this catalyst is very much established. That is a Hayashi-Jorgensen catalyst. People use a lot. We used it a lot during our earlier projects. This silyloxy group is very, very crucial to make this enantiomer selectivity in the product. However, during the reaction, this group may be falling off to form this inactive aminol. Even commercially available, this Hayashi-Jorgensen catalyst has some percentage of this inactive impurity in it. Apart from that, this amine directly could react with the electrophile to form this intermediate that can undergo graph type fragmentation that completely lose its catalytic activity. So we thought to develop a catalyst without silyl group, but almost the pyrrolidine system. So we actually come up with the two aim in one particular process. That is, we actually could do a click chemistry, and also we are removing silyl group from our catalyst, and we could attach this to polystyrene support so that we can reuse this catalyst. So having these two ideas in one shot, we actually synthesized this catalyst from commercially available this diaryl prolinol. We did acid formation at this position and clicked with this propargyl ether supported with polystyrene. We are able to make a catalyst with the different AR group and different chain length over there. This propargyl ether in principle can be accessed using alkylation chemistry. So in principle, we made in 3 steps with a polymer supported catalyst, which is probably the shortest method so far known in the literature to make polymer supported catalyst. And we wanted to test this catalyst in a very established reaction, that is amination of aldehyde. This product, you could oxidize to get amino acids after deproduction of this group, and you could access any unnatural amino acids using this method. There are many catalysts known for this reaction. We want to test our catalyst for this reaction. We screened different catalysts and conditions, and we figured out that this phenyl group over here, the catalyst works very good. Within 1.5 hours, we could get 85% yield and 91% enantiomeric excess of this product. So we tested these conditions in a different substrate in almost all cases, aromatic or aliphatic system. It works really good, and we are able to get a very high yield and enantio selectivity. We want to test the recyclability of this catalyst. The whole purpose of making this, one of the purpose of making this is to recycle the catalyst for multiple cycle. We tested in this particular substrate. We are able to make up to 4 cycles without losing any enantio selectivity, but very small decrease in the yield as observed with a different cycle. We also tested with the other substrate for the same system to make sure that it is compatible and it can be recycled in the catalysis, and we are able to show in this substrate the standard enantio selectivity in all the cycle. We have tried this catalyst for beta reaction of alpha beta unsaturated. We were not getting a high enantio selectivity. It's around 80s. So we were wondering about this CC bond rotation is the real reason that the enantio selectivity is not coming for beta reaction. However, the silyl oxy group in Hayashi-Jorgensen catalyst could do that with ease. But we do not have that big group, and we have only 3 aryl group, and one of them is heteroaryl. So we wanted to make this particular CC bond rotation arrested so that we should be able to make a better selectivity. For that reason, we were searching in the literature. Lombard developed this catalyst where this CC bond rotation is arrested, but still he doesn't want to lose silyl group because silyl groups are believed to be giving higher enantio selectivity. He's stuck with this siloxane bridge. You know, many of you might know that siloxane is much more labile than silyl ether. And even at room temperature, it could actually decompose if you keep longer time. So we do not want to have a silyl group here either. We want to make a non-silyl bicyclic system. So we want to have a linker that could not, have any silyl group. So for that reason, again, we actually started from commercially available, naturally occurring hydroxyproline. And we did in 2 steps, we are able to make this diol. We try to cyclize this 2 diols unit with several non-silyl linkers. So many failures have been seen, but only this case we are able to cyclize. That is with the iodochloromethane, we are able to make this dioxolane system. So just deprotected this acyl group using methyl lithium, and we are able to make the catalyst in just 4 steps from commercially and naturally available starting material. And we wanted to test this catalyst in a very useful reaction, that asymmetric transfer hydrogenation of alpha beta unsaturated aldehydes. There are many catalysts known in the reaction. Almost all of them goes with 10 to 20 mole% catalytic loading. We want to test our catalyst in this particular system and where we stand with respect to this existing catalyst. We screened this several conditions with a few catalysts we synthesized. And you could see this entry where you are able to even make a 2 mole% of the catalyst could do the job with 3 days approximately to get a 78% yield, 95% E. However, we stick to 5 mole% catalytic loading. Within a day, we are able to complete this reaction with 96% enantiomeric excess. We consider that as a standard conditions. We want to test various substrate for this reaction. And in almost all the cases, we get a very high yield and enantio selectivity. Even we are able to make phenoxanol, cetronolol in one part, which are actually commercially available fragrance reagents. We synthesized this intermediate starting from this enol using 5 mole% of our catalyst with 99% enantiomeric excess. Just in one step, we are able to convert that into Ramilton, which is a commercially available drug for insomnia. We also synthesized this particular intermediate using our ATH reaction with the 5 mole% of the catalytic loading with 98%. Even though it is known in the literature for the aromatic electrophilic substitution at this diaryl prolinol carbon with the arene ring, it intramolecularly didn't work for us. We tried several conditions and few I'm reporting here. There are many different methods, did not go in our hand. Also, we used this ether formation known in the literature. We want to test our alcohol and that didn't work either. So we want to drop at this stage. Suddenly my student said, okay, anyway, we made first time 2,5 disubstituted system. Can we use this as a catalyst rather than leaving it here? That turned to be very useful because we just protected them as a TBS ether using this method. And then we got a different RL group, different R groups with the 4 different catalysts. We wanted to test this catalyst in a very useful reaction that is nitromethane addition to alpha beta unsaturated to make this adduct, which is very useful because you can make several bioactive commercially available GABA drugs. All of them can be accessed in just 2 or 3 steps from that intermediate oxidation followed by reduction. And we looked at the literature. There are so many catalysts known this purpose, and we want to test our catalyst for this reaction. So when we tested our catalyst, irrespective of any conditions we studied, everywhere we got above 97% NHO selectivity. Particularly in these conditions with ethanol at 0 degree, we are able to get a 91% yield and greater than 99% enantiomeric excess for this compound, which is actually useful for making one of the GABA drug. So we tested this particular conditions in a different aromatic group. In all the cases, we are able to get 99% enantiomeric excess. And only this heteroaromatic system gave 98% enantiomeric excess. And we've also tested a aliphatic system for this reaction. We are able to make a moderative with the excellent enantiomeric excess for this substrate as well. And we utilized this method for the synthesis of R-baclofen, which is a spasticity drug. With 92% yield and 99% enantiomeric excess, we are able to achieve this intermediate. From here in 2 steps, it is known in the literature to make the drug. So apart from this, we actually would like to compare our results with the other reported reactions in this intermediate synthesis. Hayashi-Jorgensen catalyst gives 92% enantiomeric excess for this intermediate. Even this enzyme, 4-oxylocorotinate tautomerase, gives only 96% enantiomeric excess. In principle, we made a catalytic system that works for this useful reaction that gives the 99% enantiomeric excess that could be useful for making a commercially available drug. So if you look at the reaction mechanism, here we have a purposefully made, one is bulky, other one is less bulky. If you make both are bulky, it is going to be racemic product you will get. So we believe that this bulky group will make this iminium ion as a E iminium ion. And then front side, now you have 2 groups to take care of it. That nucleophile cannot approach the front face of the system. It has to come from the back face. A nitro group come from the back face and lead to the product and by regenerating the catalyst. Apart from that, we also got interested in using this catalyst for a trienamine catalysis. It's very challenging because of you have to do all the way reaction over here, which is very remote in terms of distance with respect to the catalyst sitting over here. And if you look at the literature, there are people use with either gamma or delta substituted substrate only being used a lot. Without that, it is very hard to work. There are very few reports known in a linear system. And if you look at this work by Chen, he tried to do the reaction with the nitrostyrene, and he uses always this 2 functional group present in the substrate. Linear one, he clearly said it is not working. Despite increasing the temperature or changing the additives, linear dienal is inactive in his case. We didn't take that much out of that. We thought that our catalyst might in principle do that because we have a 5 position another arm. So we actually did this reaction, invariably all conditions, we are able to get the product. And particularly optimized conditions, we are able to get a 10-to-1 DR and a 94% E with a moderate yield. We tested this condition in all the substrates. It works very well. Yields are moderate to high and NHO selectivity is high. I'm going to finish soon. Okay. So we wanted to show that our catalyst is not just complementary to the existing catalyst. It also can do that catalytic reaction what the other catalyst could do. That we did the substituted dienal as well. In all the cases, we are able to make the product. We actually would like to apply this methodology for the synthesis of this alkaloid, naturally occurring molecule. It's a beta-lycorine. And if you look at the literature, there are many methods known to make this compound. Chiral version goes more than 10 steps to 16 steps. And if you look at the system, the cyclohexyl group has a 3 contiguous chiral centered trans-trans fashion and multiple cycle and even heterocycle present in the system. It is really complex molecule to make. There are many groups have tried. We want to show our method to make this in a concise way. So since we are able to access the linear dienal for with our catalyst, and we made this substrate with 10-to-1 DR and 93% enantiomeric excess, in just 1 step, we did a 4 transformation. We reduced this double bond, reduced the nitro group, made the imine, reduced the imine as well. All happen in 1 shot. With the nickel hydrogenation, we are able to get this product. Simply formaldehyde, TFA condition, we are able to make this bridge as well. So we are able to synthesize this particular natural product in just 3 steps and with a commercially available starting material. And atom economy is amazing. That is 78%. The only side product present in this total synthesis is water. That is remaining 22%. So very delighted to see this result. We are currently writing a manuscript on that. And to summarize my work, I'm on time, I believe. To summarize the work, we actually synthesized a recyclable non-silane polymer supported catalyst and applied this for the synthesis of several amino acid derivatives. And we also synthesized bicyclic non-silane catalyst. All these catalysts works in a 5 mole% catalytic loading. And we are able to do the asymmetric transfer hydrogenation with excellent enantiomeric excess and showed the application to the synthesis of Ramilton and tericalent. And also, while we are trying to make a bridgehead system at the adjacent to the nitrogen, we failed, but we have done standard silyl chemistry, and that actually turned to be very useful to make a bioactive molecules such as baclofen and even phenibut. So we are able to make an excellent enantiomeric excess for this nitromethane addition reaction and applied for the formal synthesis of R-baclofen. Also, we tried a tri-element catalysis using this catalyst where we have this arm. I'm sure it is doing amazing job here that we are able to make a otherwise unachievable, that we are able to make this compound in 11 example I'm showing here. We made many more. We are writing the manuscript now. And we applied to the concise total synthesis of beta-lycorine using our methodology. With this, I thank all my present students and past group members. And I thank IIT Gandhinagar, ANRF, SCRB, and PMRF for the financial support. I thank you all for your kind attention. I'm happy to take any questions.
Thank you, Professor Appaiah. Now session is open for question, and due to shortage of time, we can take one question, I guess.
Nice talk, Appaji. I was just wondering, you showed that at the 5 position when you are having the diaryl with the ethereal 2, you are kind of jipping together. There, did you try to make them on the solid support, because that can become another version of taking that?
We did.
So what was the problem?
Here, E was good. It's not 99, it was 98. But the problem is that this is cis.
Yeah.
It is very fast, it's undergoing decomposition. If you look at the catalytic loading, these are all 5 mole%, but this takes 20 mole%.
Okay.
10 to 20 mole% is minimum needed because the catalyst is decomposing. Of course, it is at 55 degrees centigrade. So what I mean is that when you attach to polymer, recyclability is a problem. Second cycle itself, you get a slow time. Third cycle, fourth cycle, we are having 50% yield one.
Another, prolinols are well known that they, even if the reaction time increases, silyl ether gets decomposed. And that's what you can say Hayashi version and, Jorgensen versions are having the different silyl ethers.
Yes.
So you don't feel that, jipping them together only will solve this problem, are converting into another kind of less hydrolyzable substituent.
You're talking about this case?
Yes.
Yes, we tried so many methods. I have deleted few slides just before coming today. That we made at least 10 different conditions to cyclize, and it didn't work. I can tell you that we did intramolecular click, intramolecular.
What about having some solid support on that, where you are having methylene group? Yes, yes.
This hydroxyl group with the solid support, could be done, but if you open this out, there is a problem that you need to again make silyl protection. Without silyl group, if you open it out, E is going drastically down around 60% to 70% only you will get without silyl group. If you met O methoxy here, methoxy, 60-70% E only. The silyl is very, very crucial. So we want to replace that, but we put phenyl group properly assigned over there, and E is 96. But
so not only the bulk, something else is also responsible. You are saying that silyl group, because you substituted with the triazoles and those kind of things, which was very much complementary, that whatever the E is received by the others actually.
We actually properly connected this to polymer support. Otherwise, this E also will be lesser. You see that among this, it is giving 96 for some reaction. It never gives that. It gives 86, 80. So in other words, triaryl might not make much more bulky when the CC bond rotates. Sometimes you can bring between these two R group, right? So if you lock it, then this RL group is fully available to block the front face. So I think we got a success over here, but still it is not the best one. we are still working on it. Still, we are not done
We
with
can
cyclocity.
continue our
I'm
discussion
sorry
during
about
lunch
that.
hours because now we are running short of time. So please give a big round of applause to Professor.
Thank you.
Thank you, sir. I request Professor Kesrinath Tiwari to felicitate Dr. C.H. Raji Reddy.
Our next speaker is Dr. Jankiram Baitala. He is Associate Professor, IIT Delhi. He completed his PhD from CSIR National Chemical Laboratory, Pune, under supervision of Professor Ganesh Pandey. He pursued his postdoctoral research at University of Tromsø, Norway, with Professor Hoffman and Professor Bayer. In 2019, he joined Emory University, USA, as a visiting researcher with Professor HML Davis Group. His current research interests include allied-derived carbene-mediated organic synthesis and total synthesis of natural products. He has recently received Teaching Excellence Award from IIT Delhi, Theme Chemistry General Award, FRIPRO Mobility Grant Award from Norway, and RCN Fellowship for Researchers Norway. So with this, Dr. Jankiram is going to deliver his lecture on switchable divergent benzonylation using vinyl sulfoxonium millets and electron deficient alkynes.
Thank you. Thank you very much, Tiwari. Thank you, Yadav sir, and thanks, Dr. Vishmaya, for inviting me here. So today I would like to discuss about a divergent benzonylation using these two reagents, vinyl sulfoxamylate and electron deficient alkynes. We know the importance of that arene ring. If you look at the commercially selling drug molecules, it's around 63% of the drug molecules contain this substituted arene ring. And that's why approximately 63% it contains drug molecules of the benzene ring. So that's why many of the pharmaceutical companies are focusing the developing that arene libraries to develop new drug molecules. Consequently, the developing new methods for synthesizing this drug molecules, the substituted benzenes is attracted attention of the synthetic chemists. So if you look at common methods for synthesis of the substituted ring, this one of the classical method for synthesizing the substituted arene is a electrophilic substitution on the pre-existing arene ring. So this is many of the methods, many of the groups still using this method. But the only issue with this substituted arene is if any function group is present on this benzene ring, installation of another function group is really challenging because it need to encounter residual selectivity issues. So these two methods somewhat can overcome these issues. One is a metal-mediated coupling reaction. Another one is a directed metallation reaction. Another interesting approach for synthesizing this substituted arene is a benzonylation reaction. In this strategy, acyclic precursors can combine together either via 4+2, 3+3, or 2+2+2 to give this substituted benzene ring. If you look at nature's approach, this is a shikimate pathway. In this approach, this erythrose 4-phosphate can club together with the phosphoenol pyruvate it will generate a substituted arenes. By using this reaction is regulated by enzyme catalysts. So various kind of enzyme catalyst, it will give a various type of substituted arenes. So these arene scaffolds can be utilized as a building block for synthesis of various bioactive molecules. So we have devised our method, this one. It's not exactly like a biosynthetic approach, but somewhat like this approach that in this case, we would like to utilize this vinyl sulfoxamylate and electron deficient alkyne to synthesize this substituted arenes. So in this case, we tune the reaction conditions and reagents to get diverse kind of arene scaffolds. So there is a particular purpose to use this particular kind of scaffolds, because if you look at this template, it has that electron-rich ylide unit in one side and electron-deficient carbonyl unit in another side. Both these valuable function groups are located on the both ends of this alkene unit in one particular geometry. So that's why this template can provide a rich and versatile chemistry. that can be utilized for various synthetic transformations like various kind of rearrangement reactions and various kind of annihilation reactions and olefination, insertion kind of reaction and coupling reactions. In this today's topic, I'm going to discuss about carbocyclation, in that particularly the benzannulation reactions. So one of the interesting property of this vinyl sulfoxylamide is it can generate carbene in the presence of metals. So if we treat this vinyl sulfoxylamide in the presence of ruthenium catalyzed condition, if you use electron deficient alkynes, you will get this substituted arenes with the para position OH with respect to OIT. And if you use electron deficient alkenes, we will get a substituted arene. Here we will not have a OH here. So the mechanism is just simple. That one, as I said, that one, it can generate a carbene in the presence of metals. So if we treat this vinyl sulfoxylamide with a ruthenium catalyst, it will generate a ruthenium vinyl carbene. It will undergo the intramolecular carbene transfer reaction. It will generate a furan, 2-methoxyfuran. So if you look at this one, this 2-methoxyfuran can act as an excellent diene in a Diel cell reaction. So when it clubbed together with this electron deficient alkynes, it will undergo nicely the 4+2 cycloaddition. It can generate this oxanarbonadiene compound. So because of the strain in this oxanarbonadiene, and also this is like a ketol unit, this one, in the presence of metal catalyst, it will open this ring, it will generate this biaryl compound. So then next our thought is that one, can we able to perform this reaction without using metals? Because we can consider this vinyl sulfoxylamide as a safer alternative to vinyl diazo compounds in a metal mediated transformation. So then can it be an alternative in under photochemical conditions also? Because this vinyl diazo compound, in the presence of light, it will knock off this N2 and it can generate this carbene. It will undergo the rearrangement to generate this cyclopropene units. So then can we able to do the similar kind of transformation? Because in the presence of metal, the chemistry is almost same. In the presence of light also, can we able to generate this carbene so that we no need to use a metal in this particular kind of transformation? Suppose if this transformation works, then whatever the previous, the type 1 annihilation that what we have done, that one we can perform similar reaction, similar chemistry without using the metal. But surprisingly, when we keep these two reactant in the presence of light, instead of reacting this electron deficient alkane in the right hand side, it reacted on the left hand side. It gave very nicely that naphthalene kind of scaffolds. We searched in the literature, is there any such kind of reactions are known? Yes, it is a known, this kind of reactions. It is called styrene alkyne 4+2 cycloaddition reactions. And are also there another name is the dihydrodeal reaction. It is a pericyclic reaction. It is thermally allowed, but not photochemically. According to the pericyclic rules, it should allow only under thermal conditions. Moreover, in this case, it can give possibly it can give two different kind of isoisomers. So to the best of our knowledge, there is no intermolecular addition is reported. Only intramolecular styrene alkyne cycloadditions are known. That too, it requires specific condition, either micro irradiation or maybe 180 degrees centigrade, sometimes both together. So what is the reason why intermolecular reaction cannot be performed? This styrene alkane 4+2 cycloaddition is because one of the alkene unit is present inside the benzene ring, so that if you want to perform this 4+2 cycloaddition, it requires very high amount of energy. Very strong heating conditions are required. Under this high energetic condition, this styrene can undergo the polymerization instead of the 4+2 cycloaddition. But our reaction, it's not exactly the same, but it's somewhat like a similar conditions, this one. So this styrene unit and this alkene unit in the presence of light, it will react with this electron deficient alkyne, just 427 nanometer ethanol room temperature, and it open air atmosphere very nicely gave this naphthalene scaffold. And another thing is, in this case, we'll get exclusively but only one isoisomer, and does not require any catalyst or any reagent, and it can proceed under air also. That means the inert conditions are also not required. We have synthesized various kind of naphthalene scaffold, but here I would like to show some of the representative examples of this naphthalenes, and we can utilize this method to synthesize this benzothiophene also. So it is a very interesting method, that one, and because this trimethyl sulfoxylamide, we can get it around 1 gram in 5 rupees. Just if we add this one in the sodium hydroxide and DMSO, we can get vinyl sulfoxylamide. And to that, if we add another alkyne, then in the 427 nanometer, we can get very nicely this naphthalene scaffold. So for this reaction, just it does not require any fancy light setup also. Just if we take these two reagents and if we dissolve it in ethanol and if we keep it in oil and if we keep it in sunlight, that one after 3 hours it will form an yellow precipitate. First it will be when we dissolve it, these two reagents, it will be a clear solution. After 3 hours, it will become like a yellow precipitate. I will discuss what is this yellow precipitate in the next slide. And after 2 days, again, it will become like a clear solution. So then this clear solution, it contains 56% of this naphthalene scaffold, this one. So this is a kind of dihydrodeal reaction, but not exactly the same. But that our mechanistic studies reveals that one, it is not a actually Diel reaction. It is not a pericyclic reaction at all. Because first what will happen, these two reactant in the absence of light, it will generate this dienyl sulfoxylamide. So it is just what is happening, this sp2ch is functionation is happening. If you club these two together, this whatever I have shown, the previous, that yellow precipitate is nothing but this dienyl sulfoxylamide. For this formal CH insertion does not require any reagent or base, anything at all. Just if you mix together and it will form this one. We have analyzed this compound by X-ray crystallography. So this is actually key component in a photochemical transformation. Under photochemical condition, it will go to the excited state. Then excited divinyl sulfoxylamide can reduce the oxygen. It will convert it to superoxide, and later it will convert it into radical cationic species. It will undergo the subsequent cyclation and hydrogen atom transfer. Then subsequent aromatization. It will give a naphthalene scaffold. This one. So then our next concept is, instead of by replacing this winoate, instead of this ester, if we keep the carbonyl compound, means winone or electron deficient alkyne containing the carbonyl compound, what will happen? So we thought that one, in this case also, we were expected the same thing. Instead of ester, if we replace with the ketone, we expected that we will get this naphthalene scaffold, but we did not get this one. Interestingly, it underwent another kind of a benzanylation. So here, and what we observed that one during optimization, for this transformation, light is not necessary. Just if we mix these two components in acetonitrol room temperature, within 12 hours, we can get this metaterpineal kind of compound. So these are some of the examples. So we have done various metaterpineals. So these are some of the representative examples. And we can use heteroatoms or aliphatic also, we can use it, this one. So by using this one method, we have done some application. This is one of the applications. So if we treat with the methane sulfonic acid, it will undergo the intramolecular Friedel Crafts acylation kind of reaction. Within 2 steps, we can make this pyro compound. This is the host material for organic light emitting diodes, this one. So the mechanism is somewhat like a similar, like a previous type 2 annihilation. If we mix these two together in a dark at 10 degrees centigrade, we can get this dienyl sulfoxylamide. It was confirmed by Proton and C13 NMR. Then to reveal the mechanism, this one, then we have done some DFT calculation. So what happens first, after clubbing these two together, it will generate this dienyl sulfoxylamide, and it will exist in this deuterionic form also, because the SILAD, I can write it negative and positive charge here. This negative charge can propagate till this carbon and so till this oxygen. So it can exist in a different kind of conformation. In one of the particular conformation, when this negative charge is close to this carbonyl, it can undergo the Corey-Czechosky kind of reaction. It can make this annihilation product. But what happens in a Corey-Czechosky reaction? What happens in a Corey-Czechosky reaction? After attack of this sulfoxylamide on carbonyl compound, then this first it will form a syn-butane. Then due to the less oxophilicity of this sulfur as well as good leaving ability of this DMSO, this O- will attack here in an antiposition and it will generate an epoxide. But in our case, there is no chance of the bond rotation. Then there is no other option. This O- has to attack this sulfur. So it is attacked here and it made this 4-membered ring, and later it will undergo the fragmentation to generate this metaterpineal compound. So this is like, we can also say this one, it's like a Wittig kind of reaction. So it is just, both components are reacted when it will generating oxosulfitane ring, and this is generating the alkene and a DMSO. Sorry, dimethyl sulfone. This dimethyl sulfone, we confirmed it by GCMS. So because GCMS, we did not observe this DMSO, we observed only dimethyl sulfone. Next, to further prove this mechanism, what we have done, suppose if we block this O-, then if we block this O-, there is no chance of this oxosulfitane formation. Then to perform this reaction, we have added triplet, TMS OTF, this one. So if we do not add this one, we are getting this metaterphenyl, but if we add this TMS triplet, we got it a para-hydroxybenzoate. The commercial name for this one is a paraben, so we have synthesized various kind of parabens here. So the mechanism is the same that what I have shown in the previous case. So here that once if the O- attacks, then before O- C- attacks, this carbonyl is activated by the TMS. Then once the C- attacks, then here it will form the OTMS. But here the oxygen O- is not present here. So oxosulfate formation is not possible. So in this case, what will happen? This phenyl group is the opposite to this DMSO group. So it will undergo the NGP kind of reaction. Then it will form this keto and it will undergo the aromatization to generate this 4-hydroxy benzoate compounds. Sorry. So we have seen in the type 1 benzynylation, these two compounds in the presence of metal catalyst, it will generate this this oxanarbonadiene complex, and it will undergoing the fragmentation to give this 4-hydroxy benzoates, this one. Now, we want to develop another kind of new benzynylation by focusing only on this oxanarbonadiene. So it is known in the literature that when this oxanarbonadiene, in the presence of light, it will go to the excited state. Then it will generate this oxaquadricycline. This is a multicyclic scaffold. So this one is a very strange compound. So this one, if we heat it, then again it will undergo the cycloreversion to generate this narbonadiene scaffold. So this is in morning we can say that when we can store this solar energy, and evening time then we can use this strained energy to the narbonadiene. So this is in physical chemistry, it is regarded as a photo switch, this one. So our idea is that can we bring this strained scaffold into the organic synthesis? So what we can, what we wanted to do is, if we perform the 2+2 cycloaddition, we can generate this oxanarbonadiene. Then this scaffold we wanted to use a substituted benzene synthesis, this one. So for that then why we wanted to focus on this oxanarbonadiene is, recently if you look at in the literature, there are various strained molecules like a BCB, ABB, TCP and Hausen. They are performing interesting, they are doing, by using this one, lot of interesting chemistries are originating. So by using this one, valuable transformations are originated. So now our idea is that the strain energy of this quadricycline was not explored. So we wanted to introduce this one into the organic synthesis, this one. So, but there are some potential challenges are there here. For example, if you look at this oxanarbonadienes compound, this is a highly strained compound. So if we want to perform this light mediated condition, but this molecule wants to relieve the strain to become like a arene ring instead of that increasing the strain here. But even if it forms this an oxanarbonadiene, there are like a multiple donor acceptor systems are there in this one. So selectively cleaving one particular donor acceptor cyclopropane system is highly challenging, this one. So we have done a lot of optimization for this transformation. So these are the optimized condition to obtain this benzynolated compound. So in the presence of iridium catalyzed condition, we got this oxanarbonadiene. Then to this one, so we have added specifically this polyoxometallate. This was, we have collaborated with one of the inorganic chemists in IIT Delhi. 1 mole% of this catalyst in the presence of blue LED, we got this benzene scaffold as well as dihydrophenanthine scaffold. This scaffold does not require any bronzed acid as an additive, but this scaffold, it requires bronzed acid as an additive. So these two are nothing but fragments of this aromatic steroids. I do not want to discuss much about the substrate scope of this one. We have done a lot of substrate scope by varying the different positions this arene ring and this esters as well as the electron deficient alkyne, this one. So this is another interesting approach that we can synthesize the aromatic steroids in a very shorter approach. Previously, if you look at this aromatic steroids, it requires many steps. So just if we take this trimethyl sulfoxamide, it is commercially available. This one, if we add this one to this electron deficient alkane, very quickly we can generate this dinyl sulfoxamide. Vinyl sulfoxamide. To this one, if we add a methyl propylate toluene at 0 degree centigrade, it will generate dinyl sulfoxamide. So to this one, if we add this dimethyl ethylene dicarboxylate, this one under optimized condition, we can get very quickly this aromatic steroids. Of course, there is another isomer also can be possible in this case. So this is the mechanism for this transformation. So we have done a DFT and experimental investigation. I'm not going to show all the things. I'm just last minute. This is a so here first it will club together and it will generate this hexanorbornadiene, and under photochemical condition it will generate this quadricycline scaffold. So under this is the highly strained compound, then this carbon-carbon bond it will selectively fragment, it will generate two radical species. So there are two chances that one this radical undergo the fragmentation to the left hand side as well as the right hand side. So we have done the computation and the right hand side is the more feasible for this fragmentation. It can open up this one, and as Professor as Dr. Raja Reddy sir said that on this one, it's this kind of radical species are highly labile species and it can undergo fragmentation to generate the cyclopentadiene scaffold. And again, this one under this photocatalyzed condition further undergo 6,5 electrocyclation followed by oxidative aromatization to generate this bench-Indian scaffold. So in conclusion, I would like to say that one, if I numbering this one and vinyl sulfoxamide 1, 2, 3, 4, and 5, 6, and 7, 8, 9 and this electron deficient alkane, if we put it under metal catalyzed condition, 1, 2, 3, 4 of this carbon and 7, 8 of this vinyl, it will club together, it will form this 4+2 annulation, this one biaryl compound. And in this, if you put it in a blue LED, this 3, 2, 5, 6 of this halide and 7, 8 of this alkane, it will give this naphthalene scaffold. And in just if you keep the room temperature and instead of this R2 is a white, if we keep this is a carbonyl compound, it will undergo nicely 3+3 cyclation. It will give this metaterpineal compound. And if we do the similar kind of reaction in the presence of TMS or triplet, it will generate this parabens derivative. So actually today I did not discuss because of the time restrictions. This is also another type of annulation. So if we treat with the PHSH and AlCl3, it will give another naphthalene scaffold. This is nothing but exactly the isomer of this type 2 annulation. In the presence of a polyoxometalate and iridium catalyzed condition, it will give another 4+2 annulation. This is a bench-Indian scaffold. So finally, I would like to say I need to acknowledge my students. So this is the whole bench annulation, whatever I have shown. This is a contribution from the one particular student, the Dakshin Gawas, and Dinesh Kumar has provided some computational studies for this experimental support. And these are my current collaboration. And I would like to thank this funding agencies, IIT Delhi, DST, CSR, and Norwegian Research Grant. And finally, I would like to thank to all of you and as well as organizing team.
We can have one questions.
Excellent talk. You have just demonstrated how the sulfoxamine can be maneuvered for different kind of extended. How you showed in the last? Can you go to the last result? Yeah, there you showed that it is breaking down. Not the conclusion. Last project. Yeah. So what are the condition will extend to that 5-membered one and other one you were saying that it is making the 6-membered one?
Okay. In that case, what will happen? In this case, if I do not add a Bronsted acid, it will give this process. It will undergo this this transformation. If I use a Bronsted acid, what it will do? That one it will kill it. The protonation it will do this one. Then this OIT, it is a strained ketone moiety. It will undergo the fragmentation here. So then OH it will come. I will. Yeah. So like this fragmentation, it will happen and it will OH, it will come. Now, due to the presence of this additional that alkene unit here, it will undergo the 6,5 electrocyclation. Just opening of this ring instead in the presence of Bronsted acid, then a 6,5 electrocyclation here by using this photocatalyst.
Okay.
Yeah. Yeah. So if there are no more questions, please give a big round of applause. I feel very happy because Janki Ram and we work together. He's my junior actually. We work on the same PhD project that I started and he continued the same PhD project. So I'm very happy to see his progress at IIT Delhi. He's creating a new kind of chemistry in our India. It's sulfoxonium amide, which is coming very nice. So thank you.
Thank you, sir. I request Professor Kesrinath Tiwari to felicitate Professor Janaki Ram Vaitla. Now I request Professor Ravi Singh, speaker from IIT Delhi, felicitate the session chairs, Devendra Mahapatra and Professor K. Srinath Tiwari. We can now proceed for the lunch break, but everybody is requested to please be back in 30 minutes after lunch. That is at 2:40. Please be on time.
Conference in Chemistry, Sustainable Innovations in Drug Development. I hope you all had a great time networking and recharging for an exciting session ahead. We now move on to session 8. It is my honor to introduce you all to our esteemed session chairs, Professor Chandrakumar Apai, IIT Gandhinagar, and Dr. Ankit Srivastava. Welcome, Professor.
Sure. It is his computer. Hello, Professor Rene. We have to open here.
Et quand vous voulez entendre ce qui se passe, ça sort d'où? Ici. Soit vous partagez l'écran, soit vous partagez uniquement ça. On le fait tout de suite? Je le mets en diaporama.
Good afternoon, Professor Gray. Are you able to hear us?
Le présentateur?
Hello, Professor. Can you please unmute yourself? He's already unmuted.
Il y a le laser. Le laser.
On va vous aider. Qui voit bien? Je suis pas sûre qu'il voit. Il voit le diaporama là.
Sir, are you able to share the slides?
I listen to you.
Say it again. No. Is it possible to share the slide and access? We're able to see, no? Yeah, we are able to see your slides.
Okay, very good. That's fine.
Okay, I'll give a brief introduction about you. Then you may start your presentation.
Okay. Parfait. Merci, tu finis là, tu fermes.
Okay, good afternoon all. So nice to gather again after lunch. So we are having four speakers here in this session, and the first speaker is Professor Rene Gray. He was a scientific director for Indo-French Joint Laboratory, and he was a CNRS director. And He is going to talk to us on discovery of new potent and selective inhibitors for CLK kinases. Professor Renegri, please start your. Please start your presentation. Are you able to hear us?
Yes, I'm okay, no problem. Is it okay for you? I can start?
Yes, please.
Okay. First of all, thanks a lot for your very kind invitation to participate to this conference. And today I will speak to you about some of our medicinal chemistry research. And more precisely, I will speak about inhibitors of a special family of kinases called CLK kinases. Okay, what's going on? No, next here. Okay. What are kinases? Kinases are enzymes which transfer phosphate group from ATP to a target molecule, and then in most cases, when the target molecule gets phosphorylated, it becomes active for some activity in the human body. And therefore, kinase inhibition has become a very, very important topic in medicinal chemistry since the last 30 years or so. In fact, there are 538 kinases identified in human genome, and in less than 30 years, there are already more than 50 drugs approved by the FDA in this area. Around 200 inhibitors in clinical trials, and this is a very, very important target for big pharma with a huge market. Yeah. So this is a very important topic. In fact, if we check more carefully on the human kinome here, this is kind of a tree where the kinases have been organized by families. What do we see is that some part of the kinome have been very well studied with a good number of drugs around here. However, there are some part of the kinome which are almost empty or completely empty, which means that there are a lot of work, a lot of discovery to be made in this area, in spite of the good number of drugs already on the market. Kinases which are of interest to us today are called the CLK kinases, CDC2-like kinases, which is a recently target member of this family here of kinases. There are 4 isoforms, 1, 2, 3, 4 known, and all these kinases are well known in terms of structure, and you get the structure in the PDB data bank. Why do they are important? They are important because they are involved in the splicing problem. What is splicing? In fact, we know that from DNA to mRNA, there is a step intermediate, which is called pre-mRNA. At this stage, splicing occurs. That means that you disconnect and reconnect part of the pre-mRNA, which means that from one pre-mRNA, you can get different mRNAs and then different isoform of the protein. So this splicing is a key factor in gene regulation, and by some route which is indicated here, this kinase plays a major role in this aspect here. and they have been identified as disturbed in more than 30 types of cancer, and they are becoming very attractive target, which is proved by 4 inhibitors in clinical trials and 7 inhibitors in preclinical studies. So what I will explain to you first is the discovery by pure serendipity of a compound which is a potent and selective inhibitor of 3 of the kinases. In fact, this happened during the PhD thesis of Mr. Bramaya here. The topic, the choice of the topic was made that we work on the quinazoline type of structure. In fact, in the literature, you have already alkaloids, which are known with such a structure, and there are some anti-hypertensive drugs which have quinazoline with 2 amino group in position 2 and 4. In fact, there are also a good number of, I'm sorry? In fact, there are already a good number of quinazoline, which are inhibitor of kinases, which are drugs on the market. You see here those molecules, but all those molecules have an anilino group in position 4. So our design was to try to see what happened if you put some substituent now in position 2 of the quinazoline, either oxygen or nitrogen here. Okay. First, we make molecule with oxygen atom, and we synthesize a focused library, 10 molecules like this, in a very straightforward manner, starting from nitroaldehyde first. I'm sorry, what? You have a problem?
Yes. No, it's okay. Please go ahead.
It's okay. Please go ahead.
Vous voulez que je mette le sous-titrage? Yeah. Is it okay now?
Yes, we are fine.
Okay, so we start from this nitrobenzaldehyde, reduction of the nitro to amino, then reaction with urate makes the quinazoline, then substitution with chlorine, and then substitution with propargylic alcohol. Final step will be a click type reaction with azide to give this type of structure. When we got those molecules, these were sorry. Yes, Raji.
Yeah, you can go ahead, Rene. It's a very small, some network issue. You please proceed now. Now it's okay.
Okay. Okay. So we got these molecules, and we screened those molecules in 2 assays. First, cytotoxicity against the 9 cancer cell lines, and there absolutely no cytotoxicity effect on this molecule. Furthermore, we screened against a panel of representative kinases, and again, no inhibition of kinases. So those molecules with the oxygen here are absolutely non-active, non-toxic, no activity on it. Next, we move to the molecule with the nitrogen, that means anilino-quinazoline core structure. Similar type of reaction. First, palladium catalyzed Burwell-Harvik reaction to introduce the aniline, then deprotection here, connected, and then introduction of propyl bromide, and finally
Il y en a un qui doit avoir un micro allumé.
a final three-type reaction gives the target molecules. So we have made a library of 12 molecules like this, and I would like to draw your attention on this one, the 7H, which is called DB18. Yeah, with this molecule, very unusual molecule with a orthonitro tolyl system here. When we screen this molecule against kinase, 8 different selected kinases at 10 micromolar, what do we see is the following. This is the residual kinase activity at 10 micromolar. When you have a huge number on blue, that means that the compound is not active at all. We have 100% residual activity. On the contrary, when the number is low, and in red, it means that the molecule is quite active, is less residual activity of the kinase. So what it means is that there are two interesting compounds here. The 7L, which is quite active, but on a number of kinases, which is non-selective. On the contrary, this molecule DB18 here, 11H, is quite active and selective on this CLK1. So this is made on this type of kinase, musculus here, and we show also that there are still 20% activity at 1 micromolar, and this translates into IC50 of 120 micromolar, the 0.12 micromolar. Next, we shifted to human kinases here, and we see that this molecule is has outstanding activity because it is now active at 11 nanomolar on this kinase CLK1, 27 at CLK2, much less in CLK3, and still again very good at CLK4. So this is a very excellent activity, very outstanding activity. However, now we check again the neighbor, the DIRK kinase here, and we found absolutely no activity of this molecule at 100 micromolar on the neighbor here. This is a remarkable result, very, very unusual result for such series of molecules. So altogether, we have made with nitrogen here, dozens of molecules, low cytotoxicity except little bit of DB18. You have screened again mammalian kinase, all of them, one of them exhibit a very potent selective inhibition with no action on the neighbor. On human kinases, this has been confirmed even better with very, very high activity against CLK1, 2, and 4, and no activity on the neighbors, which is very, very unusual profile. Next, we make another series of analogs of this by changing this part of the molecule here, aromatic group here, different system. We change also the substituent here in position, terminal position here, by more polar system. And we change also the middle part of the molecule with this system. Altogether, we had many active molecules and useful structural activity relationship. One compound was more interesting, this one with an acid group here, which proved to be extremely active against CLK, even better than the DB18, with very important activity. Here, 4 nanomolar. However, here, what we see is that we have now some activity against DIRK1A, which means that this molecule has lost a little bit of selectivity against DIRK. And therefore, This molecule has been selected for the kinome scan assay because it has the best activity-selectivity relationship here. So this kinome scan performed at 1 micromolar. What it means is that this molecule has been screened against 468 human kinases from the human kinome here. And what we see here is that this molecule has really outstanding selectivity. You see here, it reacts on CLK1, 2, and 4, little bit on the neighbor here, HIPK, and one strange result here, which may be artifact. In any case, this molecule is very unique with a fantastic selectivity in this area. Now the question is, why did that? And therefore we made docking studies of this molecule in the CLK kinase here. And what do we see is that how this molecule enters very well into the ATP binding site here. You have a molecule here, another view, and another view here. It enters very, very well. And if we go into more detail, what do we see is that this 2-anilinoquinazoline is again fitting very perfectly to interact with this leucine 244, which is the key to the ATP binding site. Further, this very crazy, unusual nitro group here has also strong interaction with this lysine here, which explain why it has a key role in this reaction, in this study. Now, if we look in full detail of this, what we see is that this molecule is incredibly well suited for the CLK. Every part of the molecule has an interaction and explain why its activity is so high. This is interaction with the ATP binding site. There's a pi docking here. There's also halogen bond interaction between the chlorine here and the triazole here, at this stage. I told you about the nitro group already, and there is further a pi interaction of this aromatic group. So this molecule is very, very well suited for the CLK binding site, which explains why we have 11 nanomolar interaction. Now the next question, complicated question, is why it is selective for CLK compared to the very neighbor DIRC1A. So in that case, it's a little more complicated problem, but what the analysis shows is that the DIRC1A kinase here has a lot more of intramolecular interaction between the different parts of the kinase, some van der Waals interaction, some salt bridges, and pi stacking, which means that this DIRC1A kinase is very much more rigid than the CLK1 in red here. And therefore, what it means is that CLK is more flexible, and therefore the our molecule can enter into the CLK and adjust for the binding. While the DIRC1A is much more rigid and the molecule cannot enter into that, even if they are very close kinases. So the lesson from that is that we have to remember that kinases or protein are flexible molecules. They are not rock, they are flexible and can move. So we have been very good. We have, by serendipity, we found a fantastic molecule. It's in terms of activity, selectivity here. We have discovered this molecule, very potent inhibitor with outstanding selectivity. This molecule can become very useful pharmacological tool to explore the role of these kinases in many biological study. And we have first example, we cooperated with Dr. Salvetti in Lyon for some problems related to hepatitis B virus. We have been able to explain the activity and selectivity of this molecule, and this paves the way for next step for this in this family of kinase. So this is good. This is good. Very happy with that. Now, can we tackle the next challenge? Having that in hand, how would it be possible now by design, no more serendipity, to make molecule acting also on cell cat 3? As I told you, DB18 is very active on 1, 2, and 4, but not active, very less active on cell cat 3. And this is a general tendency. This is very common with most of the known CL-cat inhibitors. So what to do? How to obtain cell-cat3 selective or active inhibitor? First to do is to perform the analysis of the amino acid sequence for the 4 CLK here. That's what we have done by Nicolas Levoix. And what do we see here is that there is a major difference in this amino acid sequence, which is at position 241, there is a lysine instead of the leucine for the others. Moreover, if we check the structure of the CLK3 now, what do we see? That this lysine is here, the lysine 241 is here, very nearby the ATP binding pocket here, very nearby. And in fact, if we dock DB18 in this CLK3, what do we see is that we are very nearby of this lysine. And therefore, our design was to put an acid group here, which could interact by hydrogen bonding with the lysine, linked by appropriate linker to this aromatic group here. In such a way, we can have a molecule which has a stronger interaction with CLK3 due to this hydrogen bonding. In fact, after a little molecular docking study, the solution was looking very easy. A simple aromatic group here with acid group here, either in meta or para position, should be quite sufficient for that. Synthesis of this core of this molecule, similar reaction as before, palladium catalyzed amination, and then deprotection of the methoxy, introduction of a propagelic system, and click reaction to introduce this upper chain here. Now we have the bromine in meta position here, and therefore we have just to perform palladium catalyzed reaction to get the ester and saponification to get the acid, either in para or in meta. We also took care to keep the chlorine or to remove the chlorine in case there could be a little adjustment for the conformation here. There's more flexibility with hydrogen in case it could be required. So we have made those molecules and screened those molecules. Now what's the result? In fact, the result is quite interesting. Again, you see here DB18, the ratio for CLK3 toward the other 1, 2, and 4 is between 50 to more than 100, the selectivity. Now, when we perform the biological studies of this molecule against those kinases, what do we see is now that we have a ratio around 3 to 4, 3 to 5 or so, which means that we have a very much good gain in selectivity. It means now our molecules here are much more active against CLK3 than before. In fact, we have now here molecule which can be called pan-CLK inhibitors here. So we are again very happy with that. We are proud of our AI, which allowed us to get the desired result. Okay. But sometimes nature is very crazy, and make something unexpected. Just to finish it, in fact, when we dock this molecule into the cell country, we see that the position is perfect, and we have a very strong hydrogen bonding interaction between the acid and the targeted light. So it's good. So again, we are very proud of this. Our AI allowed us to get whatever we want. But nature reserves a lot of surprise to us. In fact, when we screen those molecules against Dirk kinase, as usual, what we found is that those molecules are now active again. This DB18, again, remember no activity at 100 micromolar. Now this molecule has activity against Dirk. On the contrary. So nature gives a lot of surprise. Now, when you try to understand this, why is it like this? In fact, it proved to be very simple. When we dock this new molecule, now in Dirk, what do we see is that nature has introduced a lysine in position 175 here, very close to our acid. So nature has invented well before us what our AI has designed for this molecule here. When we come back here, we have put here in order to do that, to do this interaction. But nature has done this before with the Dirk to put this interaction directly. So altogether, we have designed new structure with a possible selective inhibition of the cell cat tree. We have obtained a new inhibitor of the 4-CLK kinase with lower but significant action of their neighbor. These are novel molecules with a new interesting profile, and this paved the way to a new selective inhibitor of the cell cat tree and to explore the role of this still underexplored kinases in living system. To finish, I would like to dedicate this lecture to my friend, Dr. J.S. Yadav, who is not only an outstanding chemist in organic synthesis, but also we recognize his strong and constant support for the Indo-French cooperation with ISD Hyderabad during the last 25 plus years. Of course, I didn't do all this alone. I would like to acknowledge all the people here, chemists, biologists, molecular modeling specialists who have made this chemistry possible. I'm feeling that I'm just like an orchestra conductor trying to make the best music, the best possible music with all these artists. So now, thank you for your attention, and see some of you in Brittany sometime. Thank you.
Thank you, Professor Gray, for a wonderful talk and insight into serendipity and planned approach to make kinase inhibitors. It's a time to have a few questions. I will.
Okay. No, not that way. Oh my God, it's getting Professor Reni, good morning.
Hello.
Hey, can you hear me? This is Mahapatra.
His
voice is not there.
He's hearing.
Yeah, he is able to hear. But why
you ask your question? No problem.
Okay, so Reni, so my first question is, so what is the driving force of designing DB18?
What is the
What is the driving force to design DB18?
Okay. These kinases are very less studied. Okay. And therefore, we have to understand how they work. Hopefully, it will translate later on into a good tool to study different part of the living system and maybe some drug later on, but this long-term study.
Well, there are a lot of connections are there. That's why. Otherwise, it will never. It will never. All these connections. Lot of open. Okay, Renny, nice. So my second question is that so you have mine is not there. Okay. So instead of metachloro, if you replace with a fluoro group, so what will be the impact?
We have tried a few times, but fluoro is the best.
Okay, you can mute after asking question.
No. I'm one question. One more question is there.
Yeah, please go ahead.
Yeah, Renny, my third question is if you go to the second chapter,
yes.
So where you have tried to introduce a acid group, so there is a fast coupling reaction is there. Can you please go to the slide? Book world coupling. But
I'm sorry, I missed it.
Anyway, so let us. Okay, Renny, I think there is some internet problem is there.
Yes, connection is not easy.
Yeah, it's a very
Thanks a lot anyway.
Okay, elaborative lecture, very impressive. Thank you very much. Yeah.
Okay, Professor Green, I'm sorry for this.
All the best.
In the internet. Your presentation is really wonderful. Thank you.
Okay, thank you.
Okay, after this complex first presentation, and now we have a next speaker, my friend Balraman Yekamram, Associate Professor and Chair of Department of Chemistry at IISER Tirupati, and hello, yeah, it is echoing. You have to zoom. Oh, he has to stop sharing. No. To stop here. Okay, he was earlier senior scientist in CSIR NCL Pune, and he's going to. He had numerous awards, few to say MRSI medals, Swarnajayanti fellowship, CRSI bronze medal, and many more. To reduce the time, I just stop here and then call him to present his excellent research on sustainable chemistry, chemical production by catalytic dehydrogenation of alcohols. Polysols.
Thank you, Chandra, for your kind words. At the outset, I would like to thank Vishnu for setting up this platform to showcase some of our ongoing research at ISA Tirupati. Thank you for your invitation and wonderful hospitality provided by Dressel University also is highly appreciated. And today, I just give you an overview of what we are doing at ISA Tirupati. And basically, we are a catalyst person. So we make catalysts for energy and environmental applications, particularly homogeneous and heterogeneous catalytic system. And homogeneous catalytic system is mainly based on non-precious metal catalysts. And morning, yesterday, there are two of our speakers. They emphasize the importance of 3D, particularly non-precious metal catalysts. And also, the heterogeneous based catalytic systems mainly based on n-dope, transition metal anchored on n-dope catalytic material. Using these two catalytic systems, we work on green and sustainable catalysis for affordable and sustainable chemical synthesis, and then hydrogen generation from feedstock chemicals, and then CO2 value-added chemical. Just in each of these verticals, I'll give you what are our impacts or what is our contribution in all these three domains. And then I'll go into today's interest of the topic. And theme one, the value added to carbon dioxide. We developed a new type of catalytic system for the conversion of carbon dioxide into functional polymer, particularly polycarbonate diol, which is used a potential starting material or monomer for the polyurethane production. And then conversion of carbon dioxide to carbonaceous material via tandem methanation followed by dehydrogenation strategy that also we have developed on both the processes, not only the process development as well as catalyst development also very important. We developed both catalysts as well as the process on this direction. And then team two is hydrogen generation from feedstock chemicals. And very recently we developed a core cell structure based ion-based catalytic system, heterogeneous catalytic system used for dehydrogenation of alcohol as well as the amines. And wherein you see water, hydrogen is the co-product. This was very well highlighted by various articles and then very well received by the scientific community as well. Another very important in this line is hydrogen generation, and wherein we use formaldehyde and methanol as a redox couple to generate a C1 source free, particularly carbon monoxide free as well as carbon dioxide free generation of hydrogen was developed, and use this strategy for chemocyclic hydrogenation or triple bond to development. This again is a very well received and highlighted by the DST. And then another very important work on hydrogen generation is dehydrogenation of aqueous hydrogen, and using that hydrogen one can do the several chemical synthesis. Not only that, selective depletion also is possible. This again is highlighted by the TST portal. And now I'm coming to a third topic that's today's interest of the thing. And we use alcohol as a another feedstock chemical, that is alcohol as a reagent for various chemical reaction, for example, olefination, acylation, dehydrogenation, alkylation reaction, and then tandem CC and CN bond coupling reaction to make an interesting heterocycle. Wherein we see all the reaction is based on non-precious metal-based catalytic system, and also if you use dehydrogenation strategy, all the reaction, either water or hydrogen is the only byproduct. And this beauty of this particular reaction. And also, if we look at it, atom and highly step economical processes. And then not only that, we applied our strategy for synthesis of very interesting or life-saving pharmaceutically important compound, and few representative examples are shown over here. And also, again, a few more example I'm showing over here. And for the interest of the topic or the conference, I'm going to focus only one particular system, that is using alcohol as a acylating agent, particularly for carbon-carbon bond forming reaction. Not only in the pharmaceutical, and what we used is our C alkylation strategy, one of our nickel-based catalytic system. We use our nickel-based catalytic system for a synthesis of a trade secret donor, which is used by this particular company. Presently, they import from China, but using our strategy, we could make this molecule in a bulk scale. And because of this IP thing, I'm not showing the correct structure, but we develop a non-infringing route, and then it reduces the cost of this molecule. They import this molecule in almost $380 per kg, but using our strategy, we can reduce the cost up to $100 per kg. That's
our small contribution on this particular system as well. So now I'll show you how alcohol can be used as a acylating agent. You know, alcohol we have used as an alkylating agent. This very well explored, and we have developed so many systems. But for the first time, we are showing this alcohol can be used as a potential acylating agent. So it means that, so what is the strategy we are going to use it? First, see there is alcohol in the presence of transition metal catalyst. It undergoes the dehydrogenation reaction. So it means that it liberates hydrogen and you get highly reactive carbonyl compound. And then it undergoes a kind of condensation reaction with amine nucleophile or carbon nucleophile. You get alpha beta unsaturated system, or that's called chalcone or imine. And then whatever hydrogen is liberated, that is used to reduce that in situ liberated double bond. Then you get a saturated system. This is a typical reaction. This is known as borrowing hydrogenation or hydrogen transfer reaction. But in this case, what we observed was, instead of a kind of in a nucleophile, wherein we can use a electrophile, the alkane is using here act as a electrophile, and you get unsaturated system, and then that undergoes selective hydrogenation and lead to the saturated system. So typically, you take alkyne, and then in the presence of nickel catalyst, the alcohol undergoes a dehydrogenation, and you get aldehyde, and then this alkyne undergoes acylation reaction, and C2 you generate alpha beta unsaturated compound, and then selective reduction of this particular double bond and leads to a branched ketone, beta ketone. This is very important synthon in organic chemistry synthesis, and I'll explain to you how one can use this particular system for synthesis of various molecules. Okay, and when you devise this particular system, always one can face or encounter several challenges. It's not able, the reaction looks very simple, but you know one can expect several mode of reactivity or a different kind of product one can expect. So for example, whatever hydrogen is liberated, that can be used to reduce the triple bond, and you get such kind of molecules, or it can undergo, both the things can undergo the hydrogenation, you get alcohol, or it can stay at this stage also. There are several modes of reactivity or intermediate or possible, or these are the final product. So, but under our catalytic condition, and selectively we could get this particular alpha disubstituted ketone, right? And we optimize the reaction condition in a systematic manner. And initially we have used a nickel Temada catalyst. This is Temada tetramethylethionidiamine. It's commercially available. The catalyst derived from this particular ligand shows excellent reactivity. And also not only that, various other ligands also, bidentate ligands also we have used, and we found that this is the best catalytic system. And not only that, and other reagents, or for example, effect of catalyst, mole percentage of the catalytic system, and base and solvent, all the things were systematically studied. We found that this reaction of tetramethylethionidiamine derived nickel catalyst 10 mole percentage, and then toluene as a solvent and tetra potassium tetrahydroxide as a base is the optimal condition to achieve this alpha branched ketone synthesis. Okay, after having this particular system in our hand, next we showed the generality of this particular reaction. Initially, I've taken a symmetrical alkyne, and then weighing various alcohol, it leads to a kind of expected product in moderate to good yield. And when you switch alcohol, keeping as a constant, and then when you change alkyne from symmetrical alkyne to unsymmetrical alkyne, as we expected, you get a regioisomer, and regioisomers mostly so 1:1 ratio. So though the reactions work very nicely, and first time we are showing this acylating agent in a carbon-carbon bond forming reaction, but there are some limitations. What are the limitations? The limitations are as follows. And regioselectivity issue is there, and this is of course one cannot avoid that. Another thing is all the reaction, if you look at it, a moderate yield, almost the yield is 60 to 80%, not more than that. This is another drawback of this particular method. And also the aliphatic alkyne as well as the terminal alkyne are not reactive or unreactive in this particular system. And if you look at very carefully, and here you are generating a chiral center over. And presently, we have developed a new type of catalytic system to bring a also. And then this is the synthetic application. This is a well-known molecule, is a estrone receptor modulator. For that, this is a particular molecule, and alpha branched ketone is used as a starting material to make this molecule. And synthetically, or this is the reported route, you can see that under palladium catalytic condition, you have to use the carbon monoxide and to get this particular molecule. But in our system, just simple alkyne with the corresponding alcohol under our catalytic condition, we can get this in a decent yield, and then this can be further used for making this molecule. Not only that, we can also derivatize this particular system to various other molecules also by using a known synthetic strategy. And also having this strategy in our hand, next we know that deuterium incorporation always in a molecule is very interesting thing. It always increases the metabolic stability of the molecules. So in that context, instead of using a simple alcohol, we use a deuterated alcohol. And then in this particular case, we could get a selective beta deuterated compound. You know, the selective beta deuteration in a carbonyl compound is always very challenging one. Alpha deuteration is very simple. Just take this corresponding carbonyl compound and react with some kind of base and then throw it in a D2O, you will get this alpha deuteration. But this beta deuteration is extremely difficult. And we could show this particular strategy can be used for the synthesis varieties of beta deuterated branched ketones. And not only that, once we have this particular compound, and we have used this molecule as a starting material for synthesis of varieties of compound, for example, isoxazolate via hydroxyl intermediate, as well as reduction followed by cyclization and leading to indene derivatives. Very important thing here is, and upon reacting with corresponding aryl magnesium bromide grignard, and followed by a treatment with dehydration catalyst, the solid beta catalyst which developed in our laboratory, and that leads to a tetrasubstituted olefin. You can see these R4 molecules are groups are different. This again is a very interesting chemistry over here. And with respect to the mechanistic studies and the reaction proceeds via dehydrogenation strategy under radical scavengers, shows that it is not following a SET, there is single electron transfer mechanism, and as well as this particular compound, these are the intermediate we thought, and we found that these two produce via deactivation pathway, and these two are potential intermediate. Okay, that leads to a corresponding compound under our catalytic condition. So we believe that the reaction proceed via allyl alcohol intermediate, not via this particular strategy. Later, what we thought that okay, conversion of triple bond to double bond is another very interesting reaction. We exploited this strategy for the synthesis of this particular molecule. I will explain later. And then this formation of nickel hydride is a potential intermediate in this particular reaction. And under our catalytic condition, and there was no product formation, reaction of aldehyde and then alkyne, there is no formation of this corresponding compound. So then we added external, we added this alcohol, and then we could get this corresponding compound. This shows that the alcohol dehydrogenation by the nickel catalyst and then formation nickel hydride is a potential intermediate for this reaction. To further prove that, and you know, sodium triethyl borohydride is a very good hydride donor, and then makes the hydride, nickel hydride, and we generated nickel hydride in situ, and then that catalyzed this particular reaction. And as well as separately, we have synthesized a very stable nickel hydride that also proceeds this particular reaction. All these reactions or mechanic studies anonymously proves that there is a formation of nickel hydride this particular reaction. So based on experimental results as well as theoretical studies and spectroscopic evidences, based on all the things, XPS data, intermediate isolation by HRMS studies, all the things, we postulated the reaction mechanism. It proceeds via two different catalytic cycles. And first, there is a ligand metathesis followed by a dehydrogenation reaction, and then alkyne coordination, migratory insertion, and then migration insertion reaction that lead to allyl alcohol. Then allyl alcohol enter into the second cycle, and that undergoes isomerization reaction and lead to expected alpha substituted branched ketone. And these are the potential intermediates which are spectroscopically characterized. And also this is very well supported by DFT studies. And this insertion reaction is found to be the rate limiting step. Okay. And then, as I mentioned, then there is a formation of alkyne to alkene. We wanted to explore this strategy. Instead of using a alcohol, we use simple methanol as a iodide donor. But when you perform the reaction, the yield of the particular compound is very low. So what we thought that we added a formaldehyde into the system, then what we observed were there is a selective reduction happen. This shows that there is a formation of hydrogen gas. So means that, for example, if you take formaldehyde and methanol, you get methyl formate. This is a kind of redox type of coupling, and you get this hydrogen. And if you change either D or H here or versus D versus D, and accordingly you will get the product. Either HT or D2 or H2 you will get it. So this is the one it utilized to reduce a triple bond to double bond. Okay, and then again in this case, and there are several optimization, and general condition is 3 equivalent of formaldehyde and then in methanolic solvent under 110 degrees centigrade is a bath temperature, and then alkyne undergoes selective reduction to get a trans-olefin, and you get methyl formate as the only byproduct or co-product. And the reaction is very selective, chemoselective. If you see, there are several groups which are hydrogen or sensitive functional group, for example, bromo or carbonyl compound ester, all are very well preserved under our catalytic condition. And this is the superiority of our strategy. So under hydrogenation condition, that leads to mixture of product. For example, if you take keto here, as well as alkyne, that leads to varieties of product. And as well as under transfer hydrogenation sort, either conventional formic acid triethylamine or methyl formate and other or ABE, all the things that leads to mixture of product. But under our catalytic condition, and selectively it reduces the triple bond. So this reaction is highly chemoselective in nature. And this also is used for synthesis of several drug molecules, for example, ribositol and DMU12, and then this particular estrogen receptor via this intermediate. And again, the reaction, we have studied several mechanistic studies, particularly XPS, that shows that nickel 2 is present in your system. There is no formation of nickel 0 or higher oxidation state also. And this nickel methoxy or nickel hydride is the resting state of your catalytic system. And it involves 3 catalytic systems. First thing is the methyl formate formation, and then active nickel catalyst, and then nickel catalyst converts the triple bond into trans olefin, and then the trans self olefin enters another catalytic system that undergoes isomerization reaction and leads to cis olefin. Okay. And this is again supported by the DFT studies. And finally, I would like to thank my highly motivated young and talented students, and finally the organizer of this present conference. And I'm from ISA Tirupati, and this is located here. And this is one of the latest or very young institute. And this is a view of our campus. And there are some pictures. This is our research building, our academic building, and this is our collegium, that is conference hall. And then this is our library. And you can see a different scientist name over here. And inside it look like a elliptical structure. And then this is, I think I would say, it's the biggest periodic table, I would say, in our campus. And thank you for your kind patience as well as opportunity.
Very nice presentation. Within the time, still 1 minute left. We can have many more questions.
Yes.
If you have said first,
have you tried only with alkyne having aliphatic substituent? You have always told that alkyne has to be disubstituted with aride.
Correct.
Is this possible with alkyne substituent?
That's what I mentioned it. When you have a aliphatic alkyne, the reaction will not. Yes. That's one of the limitations of our strategy.
Oh. the reason that your double bond is not reduced by your metal hydride? It's staying.
That's what. So when you use, so initially we use the DPP system, diphenyl phosphinoethane system. Okay. In that case, there's acylation reaction takes place. But when you change the phosphine ligand to nitrogen ligand, right? The initial case is the tamada, right? So and then that we change from nitrogen to phosphine ligand. So when you use the phosphine ligand, there is a reduction is taking place. Okay, so there is a soft hard nature, it plays very important role. So phosphorus very soft in nature, nitrogen very hard in nature, that plays very important role, and that allows or that did not allowing that aldehyde to coordinate with your metal center. If the aldehyde is coordinating to your metal center, then there is a insertion reaction takes place. But if this nickel catalyst, because of the hard nature of nitrogen, nickel allows aldehyde to coordinate. But the moment you change nitrogen to phosphine, is a soft nature. So what happens is it's not allowing the aldehyde to coordinate. In that case, only this alkyne is intact with your molecule. There is a hydrogen transfer to take.
So I think he has covered almost all my questions. But one more question is that suppose now you are getting that selectivity in the reduction, double bond.
Yeah.
You're getting trans versus cis.
Mm.
So 98:2.
Yeah.
Now whatever metal you can use so that you can get exclusively cis, not trans. Now you want to reverse the selectivity.
Okay.
So is it possible using nickel, same nickel, this thing, or you have to change the metal catalyst?
We have to change the metal catalyst.
Under that condition,
No, under that
you have not. Okay. No other additives or combinations, not possible.
Yes.
Great. Good work. Yeah.
Yes.
Very good presentation.
Thank you.
The catalytic cycle, the second catalytic cycle, the first project, you are after hydride attacks on the generating the enol ether kind of process.
Right.
Again, how this is converting into the carbonyl compound proton source from pressure butanol. You are getting from the pressure butanol.
Yes. And then this undergoes isomerization. I mean, epimeri.
That hydrate, why it can't take a proton up from the pressure butanol? Because that is a more acidic. Nickel hydrate.
So what you're saying is this nickel hydride can take the proton from tertiary butanol, right? Correct. Then if it takes tertiary butanol, then again you will have this species. Then I mean, so you will have this particular species. This again there is alcohol is present, and you will have this species. Again you will undergo the thing. You will always at the end point of time. That's what I mentioned that this XPS shows the nickel hydride is potential resting state of your catalyst. At any point of time, the nickel hydride will be there in your system.
And this enol ether, whatever you got it, the first catalytic cycle you are generating the aldehyde.
You are talking about this, right?
Intermediate K. Intermediate
yeah, correct.
First catalytic cycle you are generating the aldehyde. Can't they both react and can give like a aldol kind of reaction?
This with what?
This one with the first catalytic cycle aldehyde. Aldehyde that will after intermediate C.
It is too bulky there, right? So if you look at it, no. So if I look at this, this is not that stable. So immediately take the thing, and you will have a enol, and that undergoes a kind of tautomerization reaction. You get this. Correct.
There it can possible to generate two isomers, exterior isomers. Like this one.
Yes.
The previous application of this one where you are putting a aryl magnesium bromide.
Yeah.
So I feel like different kind of isomer. That you are getting 78% of yield.
Correct.
Is there any particular reason? Substituent effect or something?
So
compound 11 can have other isomer. Compound 11.
Okay. That it's also possible. I know this is the combination of both the isomers. So what we want to show that one can make a tetrasubstituted olefin. Both the groups are all four groups are different. That's what we wanted to show here. Our aim is not to show only one particular isomer.
Any further questions? If not, let us thank the speaker for the excellent talk on the dehydrogenation process.
Now I request Professor Chandrakumar Rape to felicitate Dr. Ekembram Balaraman with a token of appreciation. Thank you so much, sir. Now I invite Dr. Navneeta Sadhu Khan, ICT Mumbai, as a co-chair for this session. Welcome, ma'am.
So in next session, we have two speakers. First speaker is Dr. Sudipto Basu and Dr. Ravi Pratap Barmal is the second speaker. So let me give you a brief introduction of Dr. Sudipto Basu. He is currently an associate professor at the Department of Chemistry, IIT Gandhinagar. He obtained his BSc from Presidency College, Kolkata, MSc from IIT Kanpur. He earned his PhD from Max Planck Institute, Dortmund, with International Max Planck Research Fellow, and he obtained his postdoctoral research from Harvard Medical School, where he received Charles A. King Trust Fellowship. Returning to India in 2012, he joined IISER Pune with DBT Ramalingam Swami Fellowship. Further, he joined IIT Gandhinagar in 2018. Today, he will be talking about powerhouse targeted small molecule mediated chemophotodynamic therapy induces apoptosis in cancer cells. I welcome Dr. Basu.
Good afternoon. Thank you, Navneeta, for your kind introduction. First of all, I'd like to thank the organizer, especially Professor Jadhav and Dr. Vishnu Maya for giving me an opportunity to present some of our latest research in front of you. So today, what I'm going to talk about powerhouse targeted small molecule mediated chemophotodynamic therapy for cancer cells. So afraid with this long jargon field title, I'll break it down slowly. So first, when we talk about powerhouse, this comes in our mind. So this is a thermal power plant in Gandhinagar, which provides electricity to the Gandhinagar Ahmedabad area. This is a man-made powerhouse. And in this particular picture, you will see that there is a natural powerhouse also, which is nothing but sun. But also, apart from these powerhouses, we also have very tiny, small powerhouses in our body, which are called nothing but mitochondria, which are very small packets full of many enzymes. And this mitochondria from our class 12 biochemistry courses or textbook, you will know that this mitochondria is involved in bioenergetics. They generate this ATP. Those are the energy currency of our cell. And they control the metabolism. But very recent studies in last 20, 30 years revealed much more than textbook chemistry or textbook biology, that they control the biosynthesis of small molecules. They are also, can you hear? Okay. They are also acting as signaling hubs. They are also controlling the cellular stress responses like autophagy and apoptosis. And most importantly, they are the hub of genomic material. For example, mitochondrial DNA, they have mitochondrial RNA, and they have whole translational machinery to generate some few proteins for oxidative phosphorylation. And you can understand that because of these so many functions, there is some kind of a problem, some kind of a disturbance, they will lead to different types of diseases. And as a result, mitochondria are implicated in diseases like neurodegenerations, Alzheimer's disease, Parkinson's diseases. They are also involved in metabolic disorders. Very recently, they are also implicated in aging. But what we are mainly interested is their role in cancer. So what we do particularly in our labs, it's called organelle chem lab. So we work in 4 different aspects. First thing is that we develop chemical biology tools. So either to visualize those mitochondria or other related organs, or we try to perturb those organelles as cancer, anti-cancer agent. We also try to understand the organelle trafficking, how these molecules end up inside the particular organelle like mitochondria. We also try to understand different organelle crosstalk. Mitochondria is not alone, so they are constantly crosstalking with other organelles like endoplasmic reticulum, like nucleus, and so on. So we try to understand that. And very recently, we are focusing on the light and matter interaction to develop phototherapy tools. Either it is a small molecule or nanotechnology-based material. Then this is very recent focus. And for doing this, we constantly use either chemical biology tools or nanotechnology-based tools. So today I'll be talking about one particular project which is unpublished yet, and that is development of a small molecule for targeting mitochondria. And here we are focusing on light matter interaction. So suddenly one day we realized that, okay, light has huge potential in changing the properties of a particular small molecule or nanomaterial. So now if we look into the textbook again, the Jablonski diagram, so if we see that in the ground state, if we irradiate the molecule with light, then the molecule goes into the excited state. And when this excited state, they're coming down to the ground state, there are multiple phenomena happening. So one phenomena is radiative decay, which I have not shown here, which is known as fluorescence. And the other part, which is very rare, is the non-radiative decay, which generates heat in response. So whatever the energy it is taking from light, it is dissipated by heat. And the other way, this excited state, singlet state, can actually, by intersystem crossing, can come to the triplet state. And when this triplet state, it's a little bit long-lived triplet state, when it comes back to the ground state, then multiple things happen. So one is the energy is converted or reacting with triplet oxygen, giving you the singlet oxygen. And this singlet oxygen can generate the reactive oxygen species and react with the cancer cells to kill the cancer cells. This is one way of killing cancer cells. And the other way is this electron in this triplet state can again react with different biomolecules to generate or to kill the cancer cells. So this is called photodynamic therapy. And when this heat is actually used to kill the cancer cells, this is mainly the photothermal therapy. So we actually developed last year, a dog bone type gold nano rod or nanoparticle, which can target mitochondria. And when we irradiate this cells with light, can generate photothermal effect, as well as chemotherapeutic drugs can be released, and it can generate this mitochondrial damage leading to cancer cell death. Also, very recently, we developed small molecules, which if we irradiate with light when they are inside the cell, mainly they are localizing in lysosomal compartment or lipid droplets. When we irradiate with light, that generates photothermal effect. And as well as this reactive oxygen species to leading to the cancer cell death. As well as we have developed recently a small molecule which can show only photodynamic effect, not photothermal effect, while localized into the lysosomal compartment. So when you irradiate these molecules inside the cell with light, that generates this photodynamic effect, which leads to the autophagy of the cells and finally leading to the apoptosis. But here, and you can see these are all very new data which we have not yet published, but we are not going to talk about these systems today. I'm going to talk about a different system today. So what is the system consist of? This system we have hypothesized that, okay, we'll have a system which is a long pi conjugated cyanine moiety, which will serve three different purposes. One is this will help in mitochondria homing because this is a positively charged molecule and mitochondria has highly negative potential. This molecule will also serve as a fluorescent probe, so we can track this molecule inside the cell. And third, if we irradiate this with light, then it can generate phototherapy, either photothermal or photodynamic. And with this, we have appended a drug molecule, which is called indomethacin, which is a non-steroidal anti-inflammatory drug used for cancer therapy. And what we hypothesize is that if this molecule can go inside the mitochondria because of its positive charge, we can irradiate with light, and that can lead to mitochondrial damage followed by apoptosis. So how we can validate our hypothesis? So we have to first synthesize this particular molecule, and we started our synthesis with this cyanine moiety, where we appended a small carboxylic acid linker. And at the same time, we generated this enamine kind of molecule. Now it is time to couple these two to generate this big molecule. So what we have done is that we have taken indomethacin. We use this carboxylic acid group and you appended a small linker, ethylene diamine linker, of course the protected one. We deprotected it to get the amine, and finally we reacted with this cyanine dye to get this amide. And finally, we dimerized this particular cyanine moiety with this molecule in 2:1 ratio. So finally, we got this particular molecule, though it's looking big, but it's a kind of a dimer. So now we have achieved our goal. So this is what we hypothesized and we have synthesized. But what we also have interestingly saw that this molecule is a kind of an amphiphilic molecule because the positive charge will be delocalized over this molecule, over these two moieties, and this is quite hydrophobic. So we wanted to see if that molecule is somehow self-assembling or not. So we checked the dynamic. Yeah, so it is kind of making some noise. Is it fine now? Okay, okay, yeah. So what we have seen is that we suspended this molecule in water and try to see the hydrodynamic diameter by dynamic light scattering. So we saw that this is actually forming a 284 nanometer structure, 284 nanometer hydrodynamic diameter, and most importantly, this is showing a zeta potential or the surface charge of very high, positive 41.5 millivolts. So that gave us some indication that, okay, this molecule might go into mitochondria. Also, we have visualized the morphology of this self-assembly, that it is actually showing a spherical structure. But very unfortunately, when we irradiated this molecule with light, 740 nanometer LED, that did not show us as expected photothermal therapy. So it did not give us any heat generation. But on the other hand, we checked, we irradiated this molecule under white light and reacted with this DPBF, which shows absorbance at 4 40 nanometer. So now if that molecule generates singlet oxygen or reactive oxygen species, then it will react with DPBF and form this DBB, which has no absorbance in this particular 440 nanometer. So if you monitor this 440 nanometer absorbance through UV with spectroscopy, then you will see a gradual decrease, which makes us confident that this molecule under white light is generating singlet oxygen and it is reducing the absorbance of DPBF. So in a nutshell, that this particular molecule is generating only reactive oxygen species, not heat under light irradiation. So that is why we can go for photodynamic therapy, not photothermal therapy. So now when we have this molecule in our hand, so the next step would be to see if it is localizing into mitochondria or not as expected, right? So what we have done is that we treated the cells. So this is HCT 116 colon cancer cells. So we treated the colon cancer cells with our molecule in a time-dependent manner. And the molecule is red fluorescence because of this long cyanine and conjugation. And we stained this mitochondria with commercially available MitoTracker green, and we saw under focal microscopy that what is the colocalization. So we see that this red and green fluorescence is giving merged yellow fluorescence, and we can actually calculate the overlapping constant, so which is called Pearson coefficient, correlation coefficient. If that is high, near 1, that means it's a very good overlapping. Okay, if it is lower, then it's very less overlapping. So you can see it's nearly 0.85 to 0.88 PCC value. That means this molecule is very nicely overlapping with the mitochondria. So it is localizing in mitochondria. Now, what happens to the other organelles? Is it going really in the other organelles or not? So that's what we also have checked. So we stained endoplasmic reticulum, Golgi apparatus, and lysosome. So you can see that again in 6-hour time point or 3-hour time points, the PCC value is quite low. So 0.45, 0.6, 0.6. So that means this is majorly going into the mitochondria. Now this molecule is going to mitochondria. What it is doing in mitochondria? Is it really damaging mitochondria after irradiating with light? So exactly what we have seen here is the real depolarization, membrane depolarization. So JC1 assay is JC1 is a dye which is red when it is aggregated, when it is in the healthy mitochondria. When mitochondria is damaged, when it comes out to cytosol, it is disaggregated, it is a monomer, and it gives a green fluorescence. So now what we have done, we treated the cells with control, no treatment, with only light, with only this molecule and this molecule as well as irradiation with light. So now you see that in the green panel, the green fluorescence is really, really high in the light irradiation, as well as the red fluorescence is gradually decreasing. So that means this mitochondria, healthy mitochondria is damaged, and the JC1 dye has come out into the cytosol. And that shows the green fluorescence increment, and that tells that this molecule induces mitochondrial outer membrane permeabilization when irradiated with 740 nanometer light irradiation. But that is not enough. So we have again checked the mitochondrial morphology, how the morphology is changing. So you can see that in control or in only light treatment or only this molecule treatment, you see that very nice filamentous mitochondrial structure. So this mitochondrial structure, filamentous mitochondrial structure tells you that these are healthy mitochondria. But when it is irradiated with light, you see that these all mitochondrial structures are broken down. They are punctated structure, and these mitochondrial fragments are all over the cells. So that clearly tells us this, okay, this molecule under light is really damaging mitochondria. Again, that is not enough. So we have done another assay to make sure. That is a TMRM. This is a red dye, cationic dye, which stains only mitochondria, healthy mitochondria. But when the mitochondria is damaged, this fluorescent molecule is effluxed from the cells, and you don't see any red fluorescence. So you can see again this in control. You see very nice filamentous structure, mitochondrial structure, or already treated cells are very nice filamentous structure. Also with this only this molecule treatment, but as soon as we irradiate it with light, the whole mitochondria is damaged. You can see that cells here in the bright field. But mind you, this light treatment is only for 10 minutes. It is not for a longer time. Okay, so we treat the cells with the molecule for 24 hours, and then we irradiate the cells with only 10 minutes, not more than that. Okay, so now mitochondria is damaged. Is it really showing subcellular reactive oxygen species generation? So we use this H2DCFDA assay. So H2DCFDA is a dye which reacts with reactive oxygen species and then gives a green fluorescence. Okay, so now again you can see that in control, LED treated and this small molecule treated cells are hardly showing any reactive oxygen species generation. But as soon as you irradiate it with light, then these cells are generating green fluorescence, which is coming from this H2DCFDA and reactive oxygen species. Further, we have seen that if there is a generation of mitochondrial superoxide or not. So this mitosox is actually showing reacting with mitochondrial superoxide species and generating this red fluorescence. So again, you see that in these control cells, you hardly see any red fluorescence, but as soon as you irradiate these cells with the molecule and light, you see this mitochondrial superoxide generation. That also tells us this molecule is going into mitochondria. Okay, so now mitochondria is damaged, reactive oxygen species is generated, as well as the superoxide is generated. What is the final effect? So that induces apoptosis or not. So how do we see that? This is a flow cytometry analysis. So we treat the cells with 2 minutes. Okay, this is the last slide. So we treat the cells with this molecule and irradiate it with light. So and then stain the cells with FITC labeled annexin V. So annexin V is binding with the early apoptotic and late apoptotic cells with the phosphatidyl serine, which is outside the cells when they are in early and late apoptotic late apoptotic stages. And propidium iodide is binding with the nuclear DNA when necrosis is induced. So you can see that you should look into these quadrants. So in control, LED treated, and this only small molecule treated cells, these two quadrants, this is a late apoptosis and this is necrosis. Very less necrosis is induced, but when we irradiate with light, you see that majority is going to early apoptosis and little bit of necrosis. We also validated the same experiment with confocal microscopy, stained the molecules, stained the cells with this FITC annexin V, and you see that in light induced, you see more and more early and late apoptotic cells and in necrotic cells. We also checked the pro-apoptotic proteins like BCL2. You can see that pro-apoptotic proteins are really decreased and cleaved, as well as COX2, because you know that we have this indomethacin, which is a COX2 inhibitor. So you can also see that COX2 is inhibited in mitochondria. Also, what is the final outcome? That this small molecule is toxic to the cells, but not that much, because after some time it becomes saturated. But when we irradiate with light, it kills almost majority of the cells in a higher concentration, though it is having IC50 value is around 3.5 micromolar. So we are trying to develop or improve this molecule to give you photothermal as well as photodynamic effects. So in summary, we have developed this molecule for photothermal photodynamic therapy, which self-assembles into nanoscale material, goes into mitochondria. When we irradiate with light, it damages mitochondria, generates ROS, and finally leads to the apoptosis. And finally, I'd like to thank my students, past and present students, but especially this work was done by Tripti and Phanindra and funding agencies. This is again this IIT Gandhinagar campus. Would be happy to host you. And finally, I'd like to thank you and happy to answer any question. Yeah. Yes, sir. Yes.
Hello.
You have used the HCT116 cell,
Yes,
right? So is it P50 plus or minus?
Sorry, what?
Is it P50 plus or minus?
I think it is P53 minus.
So the reason is, you are inducing finally apoptosis, right? So presence of P53 would be important for your.
Yes.
Maybe you are underscoring it.
Yes, quite possible.
Yes.
So we have to actually find out.
And that's the
P53
reason.
plus.
That's the
cell
reason
line.
it will be more effective if it is a P53 wild type cells rather than mutant. I have one technical question: is that did you check tubulin in your staining?
We have not checked tubulin.
Yeah.
So please.
Because you have checked all the organelle I saw.
Please
That's-
enlighten me, because I do not know how mitochondria is related to.
No,
tubulin.
because is your just to check whether your dye will be targeting tubulin as well. That needs to be looked into, I guess. And the other thing is, when you induce mitophagy, there are many ways you can do that. Did you check with your dye what happened?
Not with this.
You have one data which is close to that. No, you don't need to go back. Just I want to ask you, there is a conventional way you can induce the mitophagy. Like when it is the autophagy in mitochondria, we call mitophagy.
So what we have seen is that, so this is not for this molecule, but in a different molecule, when we try to target other organelles, like not mitochondria, say for example, endoplasmic reticulum or Golgi, we do not see immediate apoptosis. We see autophagy first. And when we use autophagy inhibitor, then we see apoptosis. So it's a two-way process. So, but for mitochondria, it directly goes to autophagy, because apoptosis, if I understand correctly, biology, I'm not a biologist, that it has a balance.
You know very good biology now.
No, don't.
So another, see, you need to check what's happened now with the, either xenograft or ortho.
Yes, that's where we are actually struggling and
No need to struggle, because your compound looks pretty easygoing. So you can take some of the liposomal vehicle. You can talk to Rajkumar Banerjee, IICT. See, he's a great guy, and he's very helpful and very knowledgeable.
In Kolkata, right? IICT?
IICT.
IICT. Okay.
Indian Institute of Chemical Technology.
Okay,
the director was here.
Okay.
And the stars also, several stars are here, and the big star from there. So, he has a very good liposomal system, and he can also modify the liposomal surface to get into certain tumor directly. It will be very useful.
Last time we met, probably, I think I can talk to him.
You talked to, he was there.
Yeah, he was.
Yes.
Last meeting.
If there is no more question, then let me thank Professor Bashu for the wonderful deliveration. Next, we have.
Excuse me, ma'am. Now I request Dr. Navneeta Sadhu Khan to felicitate to Dr. Sudipto Basu with a token of gratitude.
So our next speaker is Dr. Ravi Pratap Badwal from Department of Biophysics, Punjab University, Chandigarh. He is currently an assistant professor there. So his brief introduction. Dr. Banwal earned his PhD in chemical sciences from TIFR Mumbai. Then he obtained postdoctoral research at the University of Washington, Seattle, with specialization in structural biology, NMR spectroscopy, with contribution to TARP thesis and nucleopathies. So he has 85 plus publications, H-index 23 plus, with 2,000 citations. Some of his awards are visiting lecturer at Chonsei University and visiting fellow at Monash University. He obtained Professor DVS Jain Best Publication Award, ICMR, DHR Fellowships, and many more. With this brief introduction, I would like to welcome Dr. Ravi Pratap Banwal. His title of the talk is Drug Development for RNA Target from Neisseria Meningitis. So let us welcome Dr. Ravi Pratap.
So, very good afternoon. So let me start with, so before I start my presentation, I would like to thank the organizer, especially Vishnu Maya, for the invite and for the chair to give the detailed introduction. So in next 15 or 17 minutes, I will touch upon some of the recent work we have been doing in the lab and especially from Neisseria meningitis. So a part of my lab is working on a different RNA targets, and so we start with, we are working on a dengue virus. So there is a stem loop A RNA, and that is around 45 nucleotide long. And we are trying to get the drug developed for these RNA in the dengue virus. Apart from this, the influenza virus, it is also a promoter reason which we are trying to target in the lab. And then the famous target from HIV TAR, this is from the HIV virus. And for this case, we have two leads. And if you look at this thermal profile, so this is a free RNA. And if you add the other two molecules, see how well they sits along and the binding goes very high. It's around nanomolar range binding. And the other two projects we are trying to work with is, one is from RNA thermometer, that is from RNA Neisseria meningitis, and the microRNA work that is in collaboration with the PGIMER Chandigarh. So to look into the Neisseria meningitis, so if you look at the bacterium, this is one of the smallest bacterium which is reported, and it has several different stages and different mechanism, and that's how this bacterium goes through the host immune response. And there are basically three factors. So one is a factor H, second one is a lipopolysaccharide, and third one is capsule. So these are the three different factors, and using this bacteria is subverts the host immune response. And if you look at the literature from the WHO, it says that if you do not detect this bacteria for first 16 to 20 hours, it becomes lethal. And if you look at the literature, there are different ways these bacteria, the response from these bacteria can be increased. And temperature is one of those signals. So if you look at the nasopharynx, which is an upper air pathway, the temperature is around 30 to 32 degrees centigrade. But if you look at the bloodstream, the temperature is around 37 degrees centigrade. So that's how the bacterium goes in from the upper air pathway to the bloodstream. And if you look at the Western plot, so these are the three different factors. So from capsule, this is a RNA thermometer. This is called the CSSA. We basically name as a CSSA. Second one is factor H binding protein. We just name it FHBP. And also from lipopolysaccharide, we call it LST.
So if you look at the low temperature, there is no protein expression. But if you just increase the temperature, you see more and more protein expression is going on. So we said that there are 3 independent RNA thermometer regulating this 3 virulence factor in the bacteria. Moreover, our collaborator from Oxford, they showed that if there is a polymorphism, so basically if you look at the RNA, it is around 110 base pair. But once there is a polymorphism, basically there is a deletion of 8 base pair, this becomes lethal. And you can see from this plot, so this is Western blot. So for the wild type, this is acting as an RNA thermometer, but once there is an 8 base pair deletion, it is active even at low temperature. So that means all the clinical samples which had this deletion, the 8 base pair deletions, they become active even at the low temperature. And that was problematic. So they also looked at the persistent rate in the blood. So if you look at the input, so these are the inputs. The blue one is at 30 degrees centigrade and the pink one is at 37 degrees centigrade. So if you look at the survival rate in 10 minutes, so the survival rate at low temperature, at the lower temperature is around 40 to 45%, but at higher temperature, if you go, the survival rate is slightly different. And as you proceed in the time, the survival rate goes down and it goes rapidly. So we started the question. So if this is so important, what is the structure of this RNA, how it behaves? And once we looked into the structure and mechanism of this RNA thermometer, we wanted to look at different motions. So how each nucleotide open up and how the ribosome comes and interact with the cyanidal-Garneau sequence. So after completing these 2 objectives, we thought that why not to look into the drug development, because that is also a plan in the long run to look into the RNA project. And just to mention, this project is funded by the DBT as well as ICMR in my lab. So to start with, so if you look at the RNA in vitro transcription, so you start with a DNA template. So this is single-stranded DNA. There is a T7 promoter region which you complement together, and then use the T7 RNA polymerase and optimize the different condition of MTPs, magnesium, and different buffer conditions. So you get the RNA. But this comes with a price, and price you pay is the different aborts and polyphosphates you get. And this is what the page look like. So this is the denaturing gel, and you can see the different conditions of this RNA in vitro transcription, and you can see the intensity of the band, and based on the intensity you can decide which condition to take it further. The problem is here. So if you have a lot of aborted transcript, you have to somehow clean it up. And there are a few ways people have been using. One is the just excising the gel and then purifying the RNA, and the second one is using the column chromatography. And in most of the cases in our lab, we have been mostly using the column chromatography. And apart from column chromatography, we have also been using the denaturing gel just to make sure that the RNA which we are purifying is correct. So again, going back to this full length RNA, that is a CSS-A RNA that is from the capsule. So this is 110 base pair RNA. It is more like a stem structure. And if you look at the secondary structure, there are 2 flanking end. So this is a 5' end, this is at the 3' end. There are 2 8 base pair which are completely identical. So we call it 8 base pair copies. And there is a bulge on the top, is the apical lobe. There is a small bulge here, and there is a small bulge here as well. And then we again, as I said before, this works as RNA thermometer. So that means at low temperature, there is less protein expression. But if you go at a higher temperature, like 37 or 42 degrees centigrade, there is more protein expression. Why it doesn't move? It has to go. So, but then being a structural biologist, we wanted to solve the structure of this RNA. So when we did the just simple 1D NMR, so this is the immunos proton. So that's where you get the AU and GC base pair. So if you look at the immunos proton, this is completely broadened out. So there is no way you can solve the structure. using NMR spectroscopy. So what we did, we made a couple of the changes, and the first change we made just to remove the flanking end. So once you remove the flanking end, we again went back to the collaborator and we did this biology. So you can see that it is still active at the higher temperature, but the data doesn't look good. I mean, there is some improvement. So immunos proton has gone better up, but then it is still not doable to solve the whole structure. So we thought, why not to change this top apical lobe, because we have seen in different RNA that apical lobe, it start broadening up because there is a lot of flexibility at top. So we replace with this apical loop with something called tetraloop just to stabilize the RNA. But then the problem comes. So once you replace with a tetraloop, this RNA is no more active. And that was quite surprising because you try to solve the structure. So you got a better RNA sample to work with, but there is a different problem now because it's no more active. Then we decided that, okay, we will continue with this RNA just for the sake of solving the structure, and then we will try to back project just to look at how the biology works, and hopefully to get some model mechanism to understand how this RNA thermometer is active in samples. So to start with, so this is the second RNA I was showing in the previous slide. This is around 70 nucleotide long. And just to cut the story short, to make it more easy to solve the structure in a lesser number of days, we made 3 different samples. One is the top RNA, top part of the RNA, that is called stem, upper stem. Then we made another construct which covers the bottom part of the RNA, that is called a lower stem. We also thought that we will make another RNA which covers the middle region of this segment, and we call this as a middle segment. And this is a proton-proton nosy experiment, 2-dimensional NMR spectroscopy. And if you look at this data set, the data which is coming from this 70 nucleotide RNA, and these RNA, they completely overlay on top of each other. So this suggests that if we solve the structure of these ones, we can, it's like a jigsaw puzzle. So we can put the pieces together and we could look at the bigger picture. So individually we assign, so this is H1 prime S6 H8 walk, and that's how the nucleic acid NMR spectroscopy work. So we assign the top part of the RNA, we assign the lower part of the RNA, we also assign the middle part, though there are some sections we could not completely assign. And then we translated all the assignment back to the 70 nucleotide RNA. And so as I said, because we did not think of doing this RNA as such, because there are a lot of overlap, and that was another reason to make the small pieces of this RNA and to assign it. So this way we could completely assign the RNA, and then we went on to solve the structure. So this is the NMR structure for this RNA. It has around 1000 NOEs, around 500 different angle constraint. Also, it has a coupling, RDC, residual dipolar coupling, just to resolve the flexibility at the top or bottom of this RNA. And if you look at the structure, it's reasonably good as per the NMR goes. So then the second point is, once you solve the structure, there has to be some validation. And in case of RNA, we thought that why don't we do the SAGS? So it's a small angle X-ray scattering that gives the low resolution atomic model. So this blue copy is the SAGS image. So we did the SAGS for this RNA, and we thought that we will overlay just to see how the RNA structure look like. And this structure is completely sits well with the SAGS image. So that also tells the structure we solved is a good one to start with. So once you solve the structure, you can look at the different section or segment of the biomolecules, and that gives important information to continue further. So there is a one stretch. So there is something called UU base pair, and that is sort of very rarely reported in the literature. It's called the wobble pair, and we reported that there is something called UU base pair here. Apart from that, there is some AU base pairing, but this uridine 51 is flipping out or flipping in. So its NMR is mostly in symbol structure. So there are around 20 structure in that. And out of 20, 15 structures were just flipping in and out. And that was quite surprising. But it became very evident when we started doing dynamics studies. In dynamics, we could soon see that this is working at the microsecond scale motion. And that is also important because when you do the small molecule binding studies, most of the small molecule, they are binding in this segment. And then there are some other segment like GU rich segment, and then AC segment, and also the CC base pair, this one. So C42 and C43. So this one. So they are also playing roles. So it's kind of flipping in, flipping out. So in one case, it is going in. So it's like opposite transition is happening in this case. And these things, they club together and they make a very smart patches for the small molecule discoveries. So once we solved the structure, we thought that let us look at the mechanism, how the whole RNA unfolds and how the ribosome comes and interacts with the RNA. So, so we started doing something called DNA fingerprinting. So this is the radioactivity gel. So the low temperature one, then 30 degrees centigrade, 37 and 42. So in this case, what we do, we use a reagent called NMIA and we label at 2' hydroxyl position of the RNA. And so a strong band suggests that the regions are flexible, and the weaker intensity, they suggest that those are the base paired ones. So this segment is from the 5' end, this is from the 3' end, and this is from the apical region. So if you start looking at this one at 40 degrees centigrade data set, you will see that the 5' region and 3' region, they are completely flexible because of the intensity of this band. And you can see this is also intense. So you can see that this is also flexible. But as soon as you start increasing the temperature, it starts expanding. So this suggests that this RNA starts opening from the top and it goes all the way to the bottom. So very similar to rheostat mechanism. And we propose that at low temperature, which is in the case of nasopharynx, the RNA is more like a stem structure, but as soon as you increase the temperature, it starts unfolding from the top, and then the ribosome comes and interacts with the SIND and Delgado sequence, which is the case in the higher temperature in the blood. So this is just to give a brief summary what I spoke. So this is the first time we reported RNA of this length, especially from the RNA thermometer studies. And we also showed that the RNA structure, it acts as a rheostat mechanism. And there are several polymorphisms which I did not talk in at least this part of the story. But then we reported at least there are 7 to 8 polymorphism which is reported in the second 8 base pair regions. And once you solve the structure at the 37 degrees centigrade, we thought that we will do the snapshot of the structure at all 4 different temperatures. Can I play the movie? So this is the just a structure snapshot at different temperatures. You can see how it starts unfolding, and at a higher and higher temperature, it completely unfolds. And this is the SIND and Delgado sequence. This is the 8 base pair regions, so 2 8 base pair region, one in green and one in the yellow segment. So once you have the structure in hand, the second part is how to go to the drug development. But before you go to the drug development, we wanted to know how each nucleotide move along. So to look into the dynamics study, there are different ways and means to do. So NMR, one can do NMR, one can do the molecular dynamics, fluorescence, space, etc. to look at the different set of dynamics at different time scales. And here in our case, we looked at NMR as well as molecular dynamics at microsecond scale just to see how this unfolds. And then finally, that was very helpful to decide where exactly we can target the drug. So based on the secondary structure, we can say that there are 3 prominent reasons where P is basically a possible drug, where drug can bind. But when we completed this NMR and molecular dynamics, we realized that there is only one possibility for drug development. So this part is in collaboration with Bhardwaj Satyamurthy from IISER Bhopal. And we did the RNA dynamics at so this is spin lattice relaxation time, this is spin relaxation time. So we did at 3 different temperatures from 285 to 310 degrees centigrade. And just to cut the story short, we can pinpoint the residues, which I was talking the one of the slide. that in somewhere here, there is a very strong groove where the small molecule goes and sits. And I will just skip this slide. So, okay, so here is, so this is a 1 microsecond molecular dynamics studies. Here you see some of the base pair, they populate only in the one major region, but the other nucleotide like G6 or G19 in this case. So for example, this region of the RNA segment, that is very strong section where a small molecule is going to bind. So this was very helpful from the dynamics per se to develop the regions where we can pinpoint or where we can target the small molecule. And finally, so this is the last part of the studies. So we have a very small library. So we have around 300 different molecules. So there are at least 20 different groups, and each group has 10 to 12 different molecules. And there are a couple of molecules. You can see the binding is quite reasonably tight. It's around nanomolar region, and it binds in the segment where I was pointing out that was very, very stabilized based on the molecular dynamics and NMR studies. So then we also did the molecular dynamics just to showcase the data is pretty much flat. So there is no fluctuations in the free RNA as well as in the complex form. And these are the nucleotide which are involved in the binding. And this was very well corroborated from the thermal melting studies. So in the free form and complex samples, there is around 8 to 10 degrees jump. And this is the fluorescence data. We have also started doing something called the ITC as well as SPR just to get the more detailed kinetic studies about the binding constant. And I'm sorry for this. The part of the NMR data just got flipped. So this one suggests that the region I was pinpointing, there is a reasonable change. Basically, the binding is happening in the slow exchange regime. And that is where we started working with the second generation of the small molecules. So in summary, this is the first investigation for the structure and dynamics of a RNA thermometer. And this part, the relaxation experiment for the 3 different studies, is suggested the one segment where we can start working for the small molecule drug discoveries. And this is also first investigation to work with the small molecule binding for RNA thermometer. And similar approaches, as I mentioned in the second slide, we are also trying to look into the different RNA targets in the lab. And my lab has been funded by the sponsor agency, DBT, SERP, DST, CSR, and Department of Health Research. Last year, they funded one project in the lab, and obviously ICMR, which has been supporting throughout my career at Punjab University. The RNA work is mostly led by Hema and Akanksha, who just graduated. She's moving for the postdoc. Simran, she's working on the microRNA work. Poonam and Suraj, they started working on the different microRNA as well as the HIV TAR and RRE2B. And the collaborator, Dr. Bhardwaj Satyamurthy from ISET Bhopal, Dr. Gurpal Singh from Punjab University, Dr. Suresh Babu from Anna University, who's making all the small molecules, and Dr. Vikas Sood from Jamia and RCB, who is doing all the biology for the virus aspects. And there are some students from MSc and project trainees who helped with these people all along. And finally, the Yonsei University, which I completely forgot. So Yonsei has been supporting me for a while for my travel and all those in Korea. And thank you very much. And if you have any question, feel free to ask.
Excellent piece of work, Professor Ravi Brata.
So in your 2D NMR, mostly you are focusing on proton-proton.
Correct.
How about nitrogen and phosphorus?
Okay, so we also did a 3-dimensional NMR. So we did HCP and then a CN NMR, but those were only done to characterize us to validate the data sets. It was not like in case of proteins, people do the triple resonance experiment to solve the structure and dynamics. But in case of nucleic acid, most of the NMR is still done using the unlabeled sample where they use the 2D toxi or 2D noses. They do some 3D NMR where they use SCN or SCP, but that is just to validate some of the constraint.
I'm still sticking to the 2D NMR of nitrogen with the proton or nitrogen with the
So we did
phosphorus.
proton nitrogen HSQC. We also did proton nitrogen NOG HSQC just to add more number of constraint that we did. Yes.
Thank you.
Any more questions? If not, let us thank Professor Ravi Pratap.
Now I request Dr. Navneeta Sadhu Khan to felicitate Dr. Ravi Banwal with a token of appreciation. Thank you so much, sir. Now I request Dr. Janakiram Vaitla to officially commence this session by bestowing mementos upon our distinguished session chairs, Professor Chandrakumar Appay and Dr. Navneeta Sadhu Khan. Janakiram Vaitla, sir. Thank you so much, sir. That concludes this session. Thank you to all our speakers. Now let's take a short tea break before we return for the next session. See you all at 5 p.m. sharp. Welcome back, everyone. We now begin session 9, chaired by Dr. Sudipto Basu, IIT Gandhinagar, and Dr. Ikamram Balaraman, ISER Tirupati. Welcome, sir. Sudipto, sir.
Good afternoon. It gives me immense pleasure for introducing the first speaker of this session, Professor Ravi P. Singh. He's a professor in the Department of Chemistry, IIT Delhi. So Ravi P. Singh obtained his BSc from UP College Varanasi and followed by MSc from BHU and PhD from IIT Kanpur with Professor Vinod K. Singh. And then he joined as postdoctoral research in total synthesis and asymmetric catalysis at Harvard University with Nobel laureate Professor E.J. Kore and Brandeis University with Professor Li Deng. After coming back in India, he joined he started his independent career as senior scientist at CSIR NCL Pune, followed by joining in IIT Delhi, where he is currently serving as a professor. His research focuses on synthetic organic chemistry, particularly asymmetric catalysis, CH activation, and total synthesis. And his group develops cost-effective strategies for carbon-carbon and carbon-carbon hetero bond formation, aiming to synthesize biologically active and pharmaceutically relevant natural product. Today, he is giving a talk on expanding the repertoire of chiral fulvalin-based bioactive scaffolds. Please welcome Ravi P. Singh with lots of applause.
Good afternoon to all of you. Thank you, Professor Sudipta, for kind words. At the outset, I would like to thank Professor Vishnu Maya and Professor G.S. Yadav for giving me this opportunity to share some of our science, what we are exploring at IIT Delhi. Before going to details of today's presentation, I want to make this point that it is very clear because in the very first talk itself, Professor V.K. Singh mentioned that chirality is very important component for the life, you can say that. And in this presentation, what I'm going to showcase that how different types of chirality can be applied in synthesis, and that can lead to the induction of chirality for the small molecule synthesis. In the textbook, we have been taught that generally, what are the requirements for the point chirality, and there are other chiralities, such as you can think about like axial chirality, planar chirality, and helical chirality. In this presentation, I will showcase some of the observations which are related to the point chirality and axial chirality. To make you understand in a better way, we have been taught with this cartoon, you can see that chirality in prochiral substrate can be introduced either by the separation. What I mean to say that synthesize compound in racemic form and then resolve it. That way one can get chirality. Other that you start prochiral substrate and in presence of chiral catalyst one can introduce chirality. That way one can get. So these are the synthetic and resolution. There is another third way that you take the symmetric molecule and kill the symmetry, destroy the symmetry in presence of chiral environment. So what I'm going to showcase in another few minutes that how by destroying symmetry in chiral environment one can induce chirality. And to showcase that one, I have selected here vinylogous nucleophile. Why vinylogous nucleophile? As we all know that vinylogous reaction or Diels-Alder reaction, these are the two reactions which are very powerful. and they provide very complex structure with the very simpler, starting material. In the case of Diels-Alder reaction, you start with the diene dinophile, and same way in the vinylogous reaction, generally you get highly functionalized core. And to understand in better way, you can see here, I have accounted few of the vinylogous nucleophile, which provides opportunity to install either butanolide, lactam, or alpha beta unsaturated ester, and so on and so forth. You can see here even indole, which is very frequently observed in the pharmaceutical or natural products. If you do SciFinder search, almost more than 1 lakh, organic scaffolds are having these core. So that's make the very, important that how we can install these cores in the, already existing library. Similarly, if we talk about the coumarin is also frequently observed, and that in fact, introduces chirality, introduces bioactivity, and that's make them very, very important as a drug discovery, you can say the tool. To showcase that one, we thought of exploring 4+2 cycloaddition, applying basically 3-cyano-4-methylcoumarin and cyclopentanone, dienone. You can see here, our first objective was to do 4+2. So, and here is the another coupling partner. However, instead of getting corresponding aniline, if you see, we got here corresponding, aminofulvene. What is happening over here? That chirality in this operation is getting introduced over here, and at the same time, you can see that ring enlargement is taking place. That is the very important, you can say that synthetic phenomena which is getting observed, that 5-membered ring is getting converted to the 6-membered ring. How is it happening? And we have also, observed that chirality is very important for the bioimaging property. We have already, demonstrated that in place of racemic dye, if you use chiral dye, that has very strong impact. Their biological impact is a lot more better than the racemic one. So how is it happening? Why fulvene? Aminofulvene, if you look over, fulvene are very commonly observed in the pharmaceuticals. You can see here, acylfulvene, which is anti-inflammatory, and minidenol A, which is antiviral in nature, and nicotianoxasterpine, which is anti-cancer. Not only this pharmaceutical property, even fulvene, which is when it is sandwiched between the aryl core, that demonstrate very high electrochromic material property. So that is the importance of fulvene. And if we look over our synthetic accounts, that what are the procedure to synthesize basically the fulvene, there are very limited protocols. You can see I emphasize that what important synthetic phenomena is taking place during this transformation is the ring enlargement. If we look over the literature, basically there are so far only 3 reports to do ring enlargement in the chiral fashion. And the first was by Maruka et al., who desymmetrized cyclohexanone. He started with the cyclohexanone, and in presence of aluminum bienal complex, he did desymmetrization. Another by Feng, and he used basically chiral N oxide and scandium triflate complex, and he quinoxylene. He basically insert into quinoxylene he made. And third one is by Kingsbury. He used bisoxazolene scandium triflate complex to convert cyclobutanone to cyclopentanone. These are the 3, and all the 3 methodologies relies on basically using diazoalkane. We all know that limitation with the diazoalkane is that pyrophoric nature, and scaling of this protocol is very, very strenuous because of its very limited substrate scope and limited you can say that application. There is always demand for the new methodology how we can do that. When we treated basically 3-cyano-4-methylcoumarin with cyclopentanone, we observed that with the different kind of chiral organic molecule, it gave some selectivity, and further after optimization with the solvent and the temperature, as well as taking care of that, how is it responding with the chiral organic molecule. Basically, the reaction proceed with the bifunctional catalysis, and you can see here that tert leucine-derived dipeptide thiourea catalyst gave 97:3. Very high selectivity we observed. What is happening? How this, sudden, fusion of these two coupling partner taking place, and that gives the, aminofluin. If you look over, chirality get introduced in the first step itself. The Michael addition is taking place, chirality get introduced, and then after thrombate reaction takes place, which leads to the formation of ketimine, and this ketimine goes for intramolecular aldol reaction, followed by ring expansion, and then intramolecular manic reaction, ring expansion, ring opening, and isomerization of double bond, which gives cyclopentadiene, aminocyclopentadienyl core, and that aminocyclopentadienyl core goes for the aromatization in presence of silica and oxygen, and that give the aminofluin. At first hand, we were not expecting. We were we were hoping that it is giving aniline, corresponding aniline product. However, the nature of, if you look over, this nature of this, adduct was different. Very micromolar level of this molecule has very strong dye property, and that was the reason, we were, while optimization, we were very much expectable, but when we received the crystal structure, that made all the doubt clear. And because by the proton NMR and C13, we were not having any clue that how is it happening. You can look over that this optimized procedure is very robust, and it can bear all kind of substitution on the aryl core in 3-cyano-4-methylcoumarin, as well as substitution on the cyclopentadienone. What is happening, either you can think about aryl substitution, or you can think about aliphatic substitution. Slight deterioration we observed in the aliphatic substitution, here 85:15 with ester. In other way, I can say that it has very wide substrate scope, and it can be tried out with, even with the heteroaryls, there is no deviation in the selectivity, and it works perfectly fine. We also showcase that even small molecule T3-cyclopentadienone can go for corresponding lesson reaction, and that can give very high DR. You can see here with the palmitic acid 97:3, and for the indomethacin 97:3 DR, ER, and naproxen DR, and all these, were perfectly observed. What we observed that these polycyclic aminofluin can be further utilized. This is they are chiral in nature. They can be converted into another class of catalyst, organocatalyst, what I'm saying that. By converting into the corresponding thiourea, or corresponding to the amine, it will become primary amine, or corresponding vinyl bromide, through which one can install the aryl core or aliphatic core, and that can again, we found that even for, as morning, Apaji showed that how nitromethane addition, these all addition works perfectly fine for this catalyst. Now I will switch my gear for the another observation. As I told that, point chirality, you saw that how point chirality in this, desymmetrization of cyclic core works perfectly fine, and it gives very high selectivity. Now let's talk about that, axial chirality. As we know that allene, spirans, and biaryls, these all are having, axial chirality. But, allene is not heteropoisomeric. Basically, biaryls are heteropoisomeric because their chirality comes because of the hindrance rotation along the carbon-carbon bond on these two. And achieving that restricted rotation and installing them in the perfect geometry is very challenging. If the temperature increases, then rotation happens and you may lose the chirality. So what is challenge that at the particular temperature, how do you, in a specific orientation, how do you install both the, aryl core? And to account all the challenges limited with this axial chirality, you can see here, most of the research has been explored for the 6-6 moment biaryl, only for the carbon-carbon bond or carbon heteroatom bond. However, for the corresponding, aryl amide, as well as aryl styrene, our chiral styrene is very challenging because the rotational barrier is very slow, very less rotational barrier, and once temperatures, certain fluctuation in the temperature can lose the chirality. Not only this one, I'm talking about monoaxial chirality. However, when we think about diaxial chirality on any scaffold, what is challenged that how in specific orientation, you can install the two, aryl core on the same middle scaffold. And when they are ortho to each other, then you get certain assistance. When it goes ortho-meta or it goes ortho-para, then the steric assistance because of the substrate keeps on reducing. So you have to have a double prong approach that both the side you have to hold the scaffold in the transition state, and then in specific orientation you have to install. That's what we wanted to showcase. As you can see that, biaryl aldehyde we have utilized, and in first project you saw that how fulvene has been. Here we have installed benzofulvene to showcase that how 1,2-diaxial chirality can be obtained. You can see here in presence of taught-leucine, which is like transient directing group. What does it do? It forms with the biaryl aldehyde, it forms the LD-amine, and that LD-amine reacts with the metal state, which goes for oxidation, oxidative addition, follows by again chelation with the another coupling partner, and then reductive inhalation, so on and so forth, generate the how this whole process is happening. If we look over the timeline, only 5 reports are there for 1,2-diaxial installation. Very limited reports are there because of the challenges associated. You can see here that what is required, taught-leucine is required, not the amide. Corresponding study processing showed that how in his study amide catalyst has worked perfectly fine. Amide catalyst for the aldol reaction, but it is not always true that whatever the organic molecule you have observed, that will become like super duper for all the transformation. You can see here that taught-leucine is giving 95%, but whereas for other the same the amide nature catalyst, they are not giving selectivity. So what is detrimental? How this reaction is proceeding? How to learn about it? You can see that we can take a look at the mechanism how this is proceeding. Basically here kinetic resolution is happening. It is not going with a dynamic kinetic resolution. How the kinetic resolution or dynamic kinetic resolution? If dynamic kinetic resolution will take place, then you will observe that yield is going more than the 50%, close to 75%, 80%, something like that. But here we observe kinetic resolution. What is happening here? Basically, you see that at certain temperature, when we inject the starting material, we saw clearly like 2 peaks. And when after the reaction, when we had the leftover component, and when we injected that one, we got some selectivity, which clearly indicates that at certain temperature, only one enantiomer is reacting and leading to the product, whereas the other one is not reacting. Okay, as I told about the mechanism, you can see here that mechanism basically that aldehyde, biaryl aldehyde reacts with the taught-leucine and forms the corresponding LD-amine. Now LD-amine reacts with the palladium acetate and forms the corresponding amino palladium acetate, and that goes for oxidative addition, chelates with the fulvene, and that goes for the corresponding favored major product, and that goes syn adduct basically goes for isomerization to go for anti-adduct, and that anti-adduct goes for the beta hydride elimination. It's going with the 7-membered transition state, not the 5-membered. The reason here is that 5-member transition state with electronically not that rich, whereas the 7-membered is like electronically rich, and that proceeds further. It gets hydrolyzed and regenerated taught-leucine for the next catalytic cycle. After having this observation, we thought that can we, as we were using earlier, you can say that biaryls, can we expand this methodology to the coumarins, because coumarin are very important as a pharmaceutics. And you can see here that same observation when we did, we were able to do this alkylation, unprecedented alkylation of the furin. What is important here, that biaryls are robust in comparison like coumarin, which is having lactonic ring. At high temperature, it gets hydrolyzed and forms the corresponding hydroxycarboxylic acid. How does it happen? At high temperature, you need to find out a balance that what the procedures I optimized earlier, and there those steps has to be retained, and that's what we are able to do. You can see here that not only ethyl acrylate, but acrylamide, vinyl sulfones, acryl phosphate, and acrylonitrile, these all can be amide. Very vast alkylation can be observed by applying this methodology. We have also supported the absolute configuration by having the X-ray structure. You saw that how alkylation of the coumarin can be achieved. It can also further process for the group transformation chemistry. One can reduce, or even from the axial chirality to point chirality you can do. How you can do that? You again similarities and do the group transformation chemistry, and on those groups, if you do reaction, you can achieve the point chirality on this score. What did we see? That generally six-membered biaryl cores are having axial chirality, and when we reduce the size of this aryl core, then the energy required for the rotation keeps on reducing. Once you can see that when we go from 5-6 biaryl, then a rotational barrier is slightly better because of the ortho substitution you can see here. But when we have mono substitution, then the rotation barrier gets decreased. And having those kind of biaryls in the molecular library becomes challenging. As you can see here, that if we see the most of the bioactive pharmaceuticals, they are having like 5-HTC receptor antagonist, MRT6X 1719, and GNJ 4355. All these are having, you can see that only mono-substituted pyrazole and biaryl core, pyrazole and aryl core, pyrazole and aryl core. How to synthesize this one? To answer this problem, we used silver chiral phosphoric acid catalysis. And what is happening over here, that we activated the alkyne system, and then that nucleophilic attack can lead to the indodic cyclization of this diazole, and that can lead to the product. You can see here that very champion, you can say that chiral phosphoric acid, bienal phosphoric acid did not succeed actually. We succeeded with spinal phosphoric acid, which gave like 80 to 20 selectivity, and then further optimization leads to in the chlorobenzene, it gives 99:1, very high selectivity. How is it happening? Basically, you can see here the substrate. Here at the ortho position, isopropyl ether group is required. Why is it so? Because that hinders the free rotation of these two aryl link. If rotation is not hindered, then you may lose. When we reduce this core with the like methyl ether or ethyl ether, then again the selectivity get reduced. But when we have MOM, then this selectivity remains. That's what we wanted. That okay, find out the limitation that at which this will have the axial chirality, and with that we got 95:5. So case is that not only this, we can have even silyl substitution, box substitution, all this work for this one. How reaction proceeds? Reaction basically vinylidene quinone methide intermediate forms in this one, and through that it reacts, because generally it is reported that alpha naphthyl alkyne system goes for the orthovinylidene quinone methide intermediate, and that goes for the again cyclization by the AJA attack, AJA-Michael reaction. No, it is not happening. And the same way, if you have ortho-naphthyl alkyne system, phenolic system, that one also in the same optimized condition when we have silver acetate and chiral phosphoric acid, that also did not give the product. So how is it happening? Spinal phosphoric acid reacts with the silver acetate, forms the corresponding silver phosphate, and that silver phosphate, that supramolecular assembly, that does the catalysis. It reacts with the corresponding alkene, and it activates it, and then it goes for the indodic cyclization further. We have also experimental proof that this is the vinyl silver complex is forming, which gets hydrolyzed. If we exchange with the silyl chloride, it forms the corresponding vinyl silane, and which we have tried with the G, and that goes for the again further hydrolysis to take care to the next catalytic cycle. We saw that how biaryls and similarly can we do that is for the axial chiral styrene, which has less energy barrier. It's very challenging. What is required? That how to control EJ selectivity, how to control this axial chirality, and you can see why it is required, because biologically relevant molecules, butysol, secanol, luminol, veronal, all these are having this corresponding barbiturate with the substitution, aliphatic or aromatic substitution. And they are very important for the sedative as well as hypnotic property. What is challenge over here? If you start with the barbiturate, it can go for the C alkylation or O alkylation. That's the challenge over here. So how to have selective C alkylation in presence of the electrophile. And that's what, that's what we optimize by taking barbiturate, substitute, alkyl substitute barbiturate. We again treated it with the orthoalkyl, naphthol, and that basically reaction proceed with the vinylidene quinone methyl intermediate, and there it reacts and forms the very, high selectivity with the 9-amine quinone thiourea catalyst. You can see over here that gives very high selectivity in chloro form. Reaction, scope is very broad. I will not bore you. And you can see here not only heteroaromatic, carbon aliphatic substitution. However, we slightly lower, selectivity observed, but DR is very high for this one. Whereas in the case of, other aromatic substitution one, it has very high selectivity in both one. Reaction works perfectly fine for the both the, changes in the substrate. So how is it happening? As I told you, vinylidene quinone methyl intermediate, that's what forming, and that reacts basically with the bifunctional catalyst. Bifunctional catalyst means two core in the same molecule which are participating at the same time. Nucleophile attacks. So nucleophile is getting activated with the tertiary amine part, and thiourea holds the, vinylidene quinone methyl, and that attacks forms the corresponding, chiral styrene. We can easily scale up, and we can also convert it into different analogs. The second order, molecular library, third order molecular library can be formed. Also, you can see here, it has axial chirality. This axial chirality can be converted into the point chirality just by doing epoxidation, very high. This is like typo, it has 90:10 DR as well as be the same. So we showcase that how axial chirality can be translated to the point chirality also, and with the it can be even the barbiturate core can be hydrolyzed, and that is scaffold can be further processed for the group transformation chemistry. To conclude, today I shared 5 short stories. We started with the point chirality, that how by the desymmetrization in presence of the chiral environment, one can install the point chirality. Then we moved to axial chirality. There we saw that how by using tangent directing group, leucine derived tangent directing group, one can have 1,2 diaxial chirality in presence of fulvene. I also showcase that, how coumarines can be, converted to the axially chiral alkenes. You can see here all type of substitution one can have. I also showcase that, biaryl, axial chirality how can be achieved by taking the diazo compound in presence of spinal phosphoric acid. And in the last, I showcase that how in presence of barbiturate, by using, bifunctional catalyst, quinine-derived 9-amine thiourea catalyst, one can install the, chiral styrene. That's the beauty of this chemistry. And in the last, I would like to thank my students. Because of their hard work, I'm able to share this chemistry, especially the desymmetrization work is done by Sanjay, and Abhijit did the corresponding that coumarine alkynation. And finally, for the I'm really thankful to funding agency CSIR, SERB, and BRNS for their generous support. Without the money, there is no science. And I thank you all for listening me presently. I'm ready to take some questions.
Thanks, Ravi, for this energetic talk. Maybe in terms of time constraint.
Yeah.
I have no question. I have just 2 short comments.
Yeah.
Number 1 comments is I understand little chemistry, but I felt like I'm listening Kishore Kumar's concert.
Oh,
you are amazing.
Oh,
you are
thank
amazing.
You.
And
Yeah.
I mean, it's really, chemistry can be so interesting and so emotionally it can be spoken. That is really amazing. Second comment I have, I feel with your expertise, your lab, even if in a small corner, 2 things you should explore. One is protract technology and the second thing is antibody drug conjugates.
Okay.
I mean, whomever you want to collaborate, you do that, because protract is coming in a big way for a targeted destruction of the particular enzymes. It could be useful for many neural diseases, autism, etc. And it can also be for very devastating cancers, especially when the cancer cell divides, the drug resistance. And antibody drug conjugates is one step forward for the biotherapeutics. So you have fantastic chemistry with you. These two lines, if you introduce, I think that will be great.
Thank you. I will do that.
Thanks, Ravi. So
I request Dr. Sudipto Basu to felicitate Professor Ravi P. Singh with a token of appreciation. Thank you so much, sir.
okay, our next speaker is Dr. Namrata Rastogi. She is a principal scientist in medicinal and process chemistry division in CSIR CDRI. Dr. Rastogi has made significant contribution in synthetic organic chemistry, transitioning between academia, industry, and research institutes. Her expertise is in drug discovery and development and leadership at CSIR CDRI. And she has often received many accolades like roles in editorial roles in Tetrahedron and Asian Journal of Organic Chemistry, as well as a recipient of CRSI Bronze Medal and DST SERB Power Fellowship. And her talk is on sustainable photocatalytic synthesis of carbo and heterocycles. The floor is yours.
Okay, thank you, Dr. Basu, for the introduction. And before starting, I would like to convey my sincere thanks to Dr. Yadav and Dr. Vishnu Maya Desai for giving me this opportunity to come to this wonderful university. And so today I'll be talking about sustainable photocatalytic synthesis of some of the carbocyclic and heterocyclic scaffold which we synthesized in a laboratory. So what is more sustainable, which when we talk about sustainable organic synthesis, what is more sustainable than the nature, okay? And the most basic natural reaction, that is photosynthesis, where only using water and carbon dioxide in the presence of sunlight, plants make complex organic molecules. But the main challenge, and scientists have been trying for more than a century, scientists have been trying to mimic nature. But it is not that easy, because most of the organic compounds do not absorb in the visible light region. They are not able to absorb sunlight, okay? And therefore, a molecule known as photocatalyst or photosensitizer is required, which can absorb visible light, get excited, and transfer that energy or electron, single electron or energy, to organic molecules for further relevant functionalization or organic synthesis. Okay, so this concept was started taking momentum in around 2009, which Professor Macmillan and Corey Stephenson's groups independently published some of the some of this photoredox catalysis. But at that time, the photocatalyst, which efficient photocatalysts were mostly iridium and ruthenium based compounds, mostly polypyridine complexes of iridium and ruthenium. So now, the whole concept of sustainability and then using iridium and ruthenium based catalyst, it is kind of counterproductive, because iridium and ruthenium both are these rare earth metals. So later people started using, okay, it's not working. Okay. So other than ruthenium or iridium photocatalyst, then people started using some organic dyes like rose Bengal, eosin Y, methylene blue, and so on. However, the redox potential window of these organic dyes is not very huge. This is a limited window. So in recent years, mostly say after 2015, around 2015, lot of simple organic molecules too have been utilized as or photocatalysts. So those photocatalysts, we call them organophotocatalysts. In today's talk, I'll be concentrating on two photocatalysts, which is one is methyl acridinium dye or Fukizumi catalyst, and another is a simple dialkyl dicarboxylate pyridine molecule commonly known as Hans ester. If you look at the redox potential values of methyl acridinium, it is highly positive, and Hans ester it is highly negative. So the redox window of these two is this one is a strong oxidant and this one is a strong reductant. So in next few slides, I'll discuss selected reactions of these two catalysts in our, and some related ones in our lab. Because the major research area in our lab is diazo compound chemistry. So in around 10 years back, when we started working in the field of visible light photoredox catalysis, diazo compounds were our first choice as substrates. This was not the only reason. Another reason was that photoredox behavior of diazo compound is very less explored. The reason being, these compounds, depending on the group flanking diazo carbon, this chemistry changes, okay? And mostly because all other than donor acceptor diazo compounds, other diazo compounds do not absorb strongly in visible light region. Okay, and that's the reason donor acceptor compounds, most of the chemistry, photoredox chemistry of diazo compound revolves around donor acceptor diazo compound by photolysis leads to the formation of singlet carbenes. Okay, so now in order to make other diazo compound, especially acceptor ones, it is first thing, they do not absorb strongly in visible light region, and second thing, the photocatalyst required for their activation has to be strongly reducing, because if you look at their redox potential value, it is highly negative. And that was the reason until very recently only iridium based catalyst, FAC iridium or other iridium catalyst have been used for reducing these molecules. Not only for single electron transfer, even for energy transfer, only iridium-based catalysts have been used. So when we started working in the field of organophotoredox catalysis, we looked at this molecule, Hans ester. Its ground state potential is not very high, but in activated mode, it is a strong reductant. Until our paper came out in 2019, Hans ester have been used as a terminal reductant or a co-reductant with other iridium or ruthenium photocatalyst. But we noticed that this molecule can now not only transfer single electron, it can also transfer single hydrogen atom as well as functional group, a single functional group present at 4 position. And looking at its highly negative redox potential value, we thought that transfer of single electron from Hans ester or reduction of these acceptor diazo compounds using Hans ester as a single photocatalyst should be thermodynamically feasible. To test this hypothesis, we used alpha keto, beta diazo, this phosphonates, carboxylate as well as sulfone as substrates, and using Hans ester and TBAB and blue light irradiation, we generated this vinyl radical intermediate, and then these vinyl radical intermediate were trapped by alkynes and in intermolecular as well as intramolecular fashion for the synthesis of hydroxynaphthalenes or hydroxyphenanthrenes. This proposed vinyl radical was also trapped by allyl tributyl-tin, and further formation of donor acceptor complex between Hans ester and diazophosphonate was also ruled out because the UV visible spectrum, there was no bathochromic shift noticed in the UV visible spectrum of Hans ester. So based on these observations, we propose that Hans ester activated by blue light irradiation transfers of an electron. This photoinduced electron transfer to diazoenolate followed by nitrogen elimination leads to the formation of this vinyl radical. Now this vinyl radical, when trapped by alkyne, forms this benzylic radical. This undergoes intramolecular cyclization. On the other hand, this Hans ester, after single electron transfer to diazo compound, forms diazo radical cation. Now at this step, if you recall, Hans ester can also transfer single hydrogen atom. So now a single hydrogen atom transfer from diazo ester and hydrogen atom elimination leads to the formation of this enolated product, which upon workup gives the final product. And the only byproduct in the reaction is dialkyl dicarboxylate pyridine. So this substrate is very wide. Reaction works with terminal alkenes as well as internal alkynes. And in case of unsymmetrical internal alkynes, we got a separable mixture of regioisomers. Reaction works with diazosulfones, diazoesters, diazophosphonates, and in intramolecular fashion, phenanthrene, hydroxyphenanthrenes were isolated. Okay, so now we thought that we have solved a big problem. We could replace iridium catalyst in the photoredox chemistry of acceptor diazo compounds. But when we tried to trap this vinyl radical with the vinyl azides, instead of getting this anticipated phenanthrene product, we got D-diazotized product. That is, this radical got reduced. Okay, so because Hans ester, instead of this radical, instead of getting trapped by this double bond, undergoes hydrogen atom transfer by Hans ester. So there's a competition. Okay, Hans ester was giving only 18% yield, whereas iridium was giving 56% yield. So the concept is working. This radical is generated, and once it is trapped, this molecule will be generated. But again, in this competition of reduction of this radical or trapping of this radical by vinyl azide, there's a competition between these two reactions. So at this step, we came across a very wonderful reaction proposed in 2016 by Professor Koenig's group, where he proposed the concept of CONPET. CONPET is called consecutive photoinduced electron transfer. Now, this consecutive photoelectron transfer was exemplified by rhodamine 6G. This is another dye. Rhodamine 6G is unique because it absorbs in green light as well as in blue light region. But it is not strongly reducing. Its redox potential is only minus 0.8 volts. But in presence of a base, when rhodamine 6G becomes rhodamine 6G radical anion, it takes a single electron from a base, and rhodamine 6G radical anion, now this does not absorb in green light region. But rhodamine 6G radical anion again gets activated strongly, strong absorption in blue light region, and now its redox potential goes to minus 2.4 volt. That is, it becomes very strongly reducing molecule. This consecutive photoelectron transfer was demonstrated very well in our case when we use rhodamine 6G without any base light irradiation. In presence of Unix base, green light irradiation again lead to the same amount of product, only 34% yield, because the species at this point was rhodamine 6G radical anion, again not strongly reducing. But in presence of base, when reaction mixture irradiated with blue LED, we got 68% yield because the working species here is rhodamine 6G radical anion, which is strongly absorbed in blue light region. So after extensive optimization, we ended up getting triethylamine, best reaction conditions with triethylamine. Yeah. This mechanistically, we could show that the standard photolysis of vinyl azide, which leads to the formation of 2H-azirine, is actually not the pathway, because 2H-azirine does not provide enough amount of the product. So formation of 2H-azirine in the reaction was ruled out. A strong fluorescence quenching of rhodamine 6G was observed with TBAB, tetrabutyl ammonia bromide. The reason we could not yet, we are not sure how TBAB quenches the fluorescence of rhodamine 6G, but the anticipated quenching of rhodamine 6G fluorescence with triethylamine was also observed in increasing amount of triethylamine. So the mechanism is rhodamine 6G, once activated by blue light, undergoes single electron reduction by the base triethylamine, and now rhodamine 6G radical anion, which strongly absorbs in blue light region, becomes a strong reductant. It transfers single electron to the diazoenolate compound, which eliminates nitrogen to form this vinyl radical. Now, unlike Hans ester case, there is no species which can quench this radical. No hydrogen atom transfer, hydrogen atom providing a species present. So unlike in that case, this radical survives, and this attacks on the vinyl vinyl azide, and which undergoes nitrogen elimination to form this imines radical. So imines radical undergoes intramolecular cyclization followed by oxidation by triethylamine radical cation, and dehydration leads to the dehydration, and workup gives you the final product. But Hans ester story is not over yet. Okay, this is just substrate scope. I won't go in detail of substrate scope. Okay, another very interesting reaction which always required iridium-based photocatalyst is photocatalytic denitration of benzotriazole. So benzotriazole under photolytic conditions can be termed as equivalent to orthoamino diazonium, arene diazonium compounds, because it leads to the formation of aryl radical. However, and then this aryl radical has been trapped with several species by Gloria cell group. However, the catch in this one is that this benzotriazole has to be a benzoyl group specifically for the stabilization of this negative charge. So if there is no benzoyl group, this benzotriazole chemistry under photoredox catalysis does not work. At this point, we thought two things. One, we thought that if we prepare a chalcone, if we attach a chalcone moiety at the benzotriazole, we will not only stabilize the negative charge form, we will also be providing a ready template for intramolecular cyclization of this radical if generated, which will lead to the formation of enanthridine molecule. When we calculated the redox potential value of this chalcone, benzotriazolyl chalcone, we realized that there is possibility, the thermodynamic feasibility of single electron transfer by Hans ester to chalcone, benzotriazole chalcone is quite possible. When we tried this reaction, we did actually isolate this product in ketoenamine form, which was confirmed by proton NMR as well as this crystal structure. Again, very white substrate score. Okay. And in this reaction, we used various substitutions on Hans ester, and clearly all kind of R1 and R2 group works, but R has to be H. Without H, this reaction, with H, this reaction gives all kind of Hans ester are successful, but without H, when R is methyl, this reaction does not work. Means hydrogen atom transfer in Hans ester is required. Secondly, the fluorescence of Hans ester was strongly quenched by benzotriazolyl chalcone. 97.18% of fluorescence quenching was noticed with it. And also as a routine, this formation of donor acceptor complex between benzotriazolyl chalcone and Hans ester was also ruled out. Further, when we took dye-substituted product and two substitutions at ortho position to to prevent intramolecular cyclization of the radical, we could trap this radical. We could trap this radical, aryl radical generated with tributyl allyl 10. So the mechanism is again for this activated Hans ester transfers a single electron to benzotriazolyl chalcone, which eliminates nitrogen to form this radical species B. Now this radical undergoes intramolecular cyclization as well as protonation, followed by hydrogen atom transfer from Hans ester radical cation, and this molecule transfers another hydrogen atom radical to release hydrogen gas. The final product is formed this way. So until now, I was talking about the strongly reducing organophotocatalyst, that is Hans ester, and rhodamine 6G was one of them. Now I'll be talking about chemistry of highly oxidizing photoredox catalyst. So the visible light mediated reactions of 2H azerines, depending on R1 or R2 group, if either of them is H, reaction goes through cleavage of C-N bond, and if none of these is H, reaction goes through formation of acylenyl radical cation. But in any of these cases, given the highly positive oxidation potential, redox potential of 2H azerine, very strong, strong oxidizing photocatalyst, that is fucosomic catalyst or mesylacridinium dye is required. So we generated this acylenyl radical cation and trapped it with various radical, with various substrates for the synthesis of triazoles, this imidazolines, and this pyrols, triazoles, imidazolines, etc. I'll quickly go through all these reactions because the mechanism is almost same. This reaction, this mesylacridinium catalyst undergoes reductive quenching and by a single electron transfer from this 2H azerine, where this azerenyl radical cation undergoes CC bond cleavage to form acylenyl radical intermediate. Now this acylenyl radical intermediate, when tried by nitroalkene, forms this forms this radical cation intermediate. Now this one further oxidizes this reduced photocatalyst to complete the catalytic cycle, and this intermediate D undergoes intramolecular cyclization followed by base catalyzed this aromatization in this case. Now this mechanism is same, and in this case a tri-substituted pyrrole is formed. When you take, When you take Morita-Belli-Selman products or are substituted as bromine, either through debromination or denitration, nitration, you end up getting this tetra-substituted pyrrole molecule, where either nitro group, you can also introduce nitro group, as well as various these substitution where alcohol, ketones, carboxylate substituted pyrroles could be synthesized. The same chemistry applied for the synthesis of these tetrazoline molecules by taking this imines as radical acceptors. There's this interesting reaction where we took disonium salts as radical acceptors. The interesting point here is this disonium salts under visible light activation, they undergo nitrogen elimination to generate arene radical. But in this case, due to a strongly oxidizing dye, reaction works the other way. And instead of generation of aryl radical, this disonium salt works as radical acceptor by accepting this acyl-anenyl radical intermediate. So acyl-anenyl radical intermediate now, either it can attack on terminal nitrogen or center nitrogen. Depending on this attack, two regioisomers were formed in case of different R1 and R2 groups. At this point, I would like to mention one interesting reaction. At the same time, we used this pyrrolium salt as dipolarophiles in a cycloaddition reaction with diazo compound. So at this point, we realized that this pyrrolium salts are also strongly photoactive compounds. Not only strongly photoactive compounds, looking at their redox potential values, we realize that these molecules can be used as this organophotocatalyst to oxidize 2H azerine also. And also because we use them as dipolarophiles, so we thought of using these molecules in dual role, that is as dipolarophile as well as organophotocatalyst. And this is what we do without taking any other photocatalyst. We take these molecules. They serve as tri-substituted pyrrolium salt, serve not only as dipolarophile but also as photocatalyst for generation of this chalcone substituted pyrrole moiety. The broad substrate scope, yeah. And again in this reaction, if you look at quenching, there is moderate quenching, but strong enough to carry out this reaction. This triphenyl pyrrolium salt undergoes strong quenching with the diphenyl azerine, and this radical was also trapped with tributyl allyl. Yeah. So the mechanism goes through activation of this pyrrolium salt first, pyrrolium dye, and this triphenyl pyrrolium salt transfers a single electron, accepts a single electron, single electron from this azerine. Azerenyl radical cation undergoes CC bond cleavage to generate this acyl-anenyl radical intermediate. And on the other hand, this triphenyl pyrrole radical, pyrronyl radical is generated. Now these two radical undergoes radical coupling. Now this radical coupling leads to the formation of this cationic intermediate C. This upon ring opening, this CO bond cleavage and 1,5 cyclization leads to the formation of this pyrrole intermediate, reduced pyrrole intermediate here, which in presence of, after workup, it leads to the formation of final this chalcone substituted pyrrole product. So I would quickly summarize my work. We used single electron reductants, Hans ester and rhodamine 6G for carrying out several reactions, where Hans ester was used for the formation of these phenanthridines and naphthol moieties, and rhodamine 6G using the concept of CONPET. We use these molecules for, we use these molecules in a reaction of diazo keto compounds for vinyl radical generation, and this vinyl radical was trapped by vinyl azides for the formation of phenanthridine molecule. And we use two different organophotocatalysts, a single electron oxidants. One is triphenyl tripyrrolium salt, which were not only organophotocatalysts, they also serve the purpose of dipolarophiles in this reaction. And another was mesylacridinium salt, which was used for the generation of same acyl-anenyl radical intermediate, which was trapped by different substrates for the generation of pyrroles, imidazolines, triazoles, etc. So in all, we could synthesize differently substituted phenanthridines, naphthols, hydroxyphenanthrines as well, imidazolines, pyrroles, triazoles, and again, different substitutions of pyrrole as well. So I would like to acknowledge my students. This work was started by Savita, my former student Savita and Lalita. Lalita is doing postdoc in IT Gandhinagar. She is here. And again, Punema is continuing this work. And I'm also thankful to my collaborators, Dr. Sarkar from ICER Kolkata and Dr. Mukherjee from ICER Bhopal, and also my parent institute and organization CSIR and CDRI for funding, and currently ANRF, formerly SIRP, for Women Power Fellowship. And thank you. Thank you. I hope I'm on time.
Yeah, thank you, Dr. Rastogi, for being on time. So probably there is only one question in this time frame. If not, oh, you have.
You showed that azarine radical intermediate.
Yeah.
Do you isolate that one by quenching into corresponding aziridine, azarine? Huh?
No.
Yeah.
As a arenyl radical.
Because in the Campbell synthesis, Hodge-Campbell aziridine synthesis, this is the intermediate they projected that, yeah. So just I was curious because, this, yeah, aziridine.
This one?
No, yes, this one, yeah.
This is radical cation.
Yeah.
So once it is generated, it quickly undergoes this CC bond cleavage.
Okay.
Yeah.
And how do you prepare that one then?
This one?
No, the first one actually.
This one?
Yeah.
2H azarines, known synthesis.
Okay.
I don't even remember the starting material, what did we use. It's a known synthesis. And these are stable molecules.
Okay, thank you.
Okay, if there is no further question, we thank Dr. Rastogi again for this wonderful talk.
I request Dr. Sudipto Basu to felicitate Dr. Namrata Rastogi with a token of our gratitude. Thank you so much, ma'am.
So good evening. And now the final speaker of this session is Sir Itish D. Patel from Gujarat University. He comes with a very rich background. He's a kind of all-rounder in organic synthesis, medicinal chemistry, chiral synthesis, organic chemistry, oceanology, etc. And for his accolades, recently he received ISCB Best Teacher Award 2024 and then ICC Award 2023. And the title today's presentation is very interesting. It's research makes a zero to hero. So we are looking forward. Professor. So 17 or 18 minutes and then 2 minutes for the discussion. 17 minutes, I'll give you a kind of all around. Yeah. Thank you.
Good evening. I, Professor Itish Patel, going to give presentation over here for the young researchers. Normally, when we start doing research, at that time we are having many limitations. But if you overcome all the limitations, then you can achieve something. So here we are going to, I'm going to talk about that the journey, how we have started. We are working in the, excuse me, can I have? Hello. So we are working in the different field of chemistry, computational chemistry, medicinal chemistry, asymmetric catalyst, synthesis, then natural products and process development. As the discovery of the drug, it required 10 to 15 years. But if we focus or if we use computational chemistry, then we can minimize the time for the drug. Here, computational chemistry can help us for target identification, then title validation, optimization, and preclinical studies. Based on the computational chemistry, several discoveries are there. And till date, today, some drugs are in the market And the drugs are developed with the support of computational chemistry. At Department of Chemistry, we are having a Schrodinger software. We are using it. To use the Schrodinger software, there are several steps that we are following. We are following ligand-based drug discovery approach, then structure-based drug discovery approach, target selection, ligand preparation, ADMET study, docking, selection of synthesis, then in vitro study, evaluation of results, modification of the compounds, toxicity study, lead optimization, and improve design, and if required, then synthesis of optimized compound. Here, in ligand-based drug discovery, we have followed the approach of molecular hybridization. In molecular hybridization, we have collected some molecule from the literature and we have modified those molecules. And then after, we have designed a molecule. This molecule we have carried out for computational chemistry first, that is called in silico study. For that, we have taken a PDB code 3R44, and then after, we have checked the docking score. In the docking score, we have found that our molecule is properly bind with the target, means that there is a target ligand complex formation is there. And after that, we have select some molecules for the further studies, that is called MD studies. And after that, we have also performed the Lipinski rule, followed the Lipinski rule. And then after, we have started the synthesis. During the synthesis, we have found that our compound having chirality. And first of all, we have isolated or separate the molecules by resolution technique. And after that, we have synthesized or modified the pathway in such a way that we can synthesize only single isomers, and we have carried out the reaction under microwave, so we can reduce the time of the reaction also. And after that, we have completed the process, and finally, target molecule we have synthesized. The structure of the single isomer we have confirmed from the single crystal XRD, and we have reported to CCDC, and we are having its number. So looking to the wonderful work, we have filed a patent, and the patent is granted by Indian Patent Office. Then next approach, that is called structure-based drug discovery. In structure-based drug discovery, we have carried out study for MTB. In MTB, we have find out some PDB codes, and based on PDB codes, we have find out the cavity is there. Based on the cavity, we have designed the molecules based on the molecular hybridization concept. After designing and after completion of the computational study, we have synthesized the molecule, and those molecules we have carried out the study for the anti-tuberculosis activity. And after getting the good result for the anti-tuberculosis activity, we have filed a patent, and that patent is also granted by Indian Patent Office. Likewise, other derivatives also we have synthesized by the same approach, that is called structure-based drug discovery. From nature, we have inspired, and in nature, we have found that some molecules are there. Those are having dihydrospiro derivatives. We have modified natural products, and we have synthesized this derivative. After completion of all computational study, we have carried out the anti-malarial and anti-tuberculosis activity of the synthesized compound by us. We got the good result for the same. We are not only working on the malaria and tuberculosis, we are working in the field of anti-cancer agent development. In anti-cancer agent development, we have targeted it. We are working on targeted protein that is called P53. P53 is known as a guardian protein, but if any defect is there, then P53 is bind with other receptor. Its name is MDM2, and the normal work of the protein is disturbed, and that's why we have to develop some inhibitors. That inhibitor having capacity to not react with mutant P53 and MDM2, so we can control the growth of the cancer. Looking to the literature available, we have designed a molecule by available data or based on the available data, and we have carried out the synthesis. After synthesis, we have confirmed the structure by single crystal XRD and other analytical data. After that, we have submitted our molecules to National Cancer Institute USA. They have carried out the study for the single dose. In single dose, our molecules are showing good results, and that's why they have carried out the further study for 5 doses concentration too. In 5 doses concentration also, our compounds are showing good anti-cancer activity. So after getting the results from a NCI, we have carried out the toxicity study of our molecules, and our molecules are showing less toxicity. This is another derivative. Based on the previous result, we have followed the another pathway that is called bioisoterism, where we can modify or we can replace one ring. Here aromatic ring is present. This ring is we have replaced by another 5-member aromatic ring, and we have carried out the synthesis of the molecule and confirmed the structure by single XRD. After that, we have submitted the molecules for to NCI, and at present we have filed 2 patents for the work which we have carried out for anti-cancer activity. We have carried out some work for the thiazole-based thiosemi-carbazone derivatives, and those synthesized compound we have submitted to NCI, and at NCI we have got good result for anti-cancer property. Likewise, other benzimidazole thiosemi-carbazone derivative, this derivative is also showing anti-cancer activity, and we have received good result from NCI USA. Based on the ring and based on the characteristic of benzimidazole thiosemi-carbazone derivative, this derivative target that is called RNR, ribonucleotide reductase. This target is also found in malarial parasite also. So we have carried out anti-malarial activity, and in that we have compared our results with quinine and chloroquine. We have found that some of the molecules are showing good anti-malarial activity. We have carried out the QSR study for the same, and then after some of the molecule we have sent to Rega Institute, Belgium for anti-HIV activity, and some molecules are showing anti-HIV activity also. Now we are not only working in the field of therapeutic, we are also working in the field of diagnostic also. Tuberculosis is the major problem of India worldwide. These are the methods, those are available in India for the diagnosis. But the methods are having limitations, and that's why we have developed a fluorescent dyes in our lab. Then after we have screened our compounds against the MTB, and we have found good results. When we have found good results, we have tried to convert our lab-based technology into strip, and the results are good. And right now, the development at the last stage, we are working with the Premier Diagnostic Corporation, India, for the final technology transfer. We have received project from BIG Birec also for this kind of work. In this category, we have filed 5 patents, and all patents are granted by Indian Patent Office. Now, natural product. We had a project from SEC ISRO where we were working for Arabian Sea parameters of Gujarat coastal. During our study, we have found that near Dwarka, there are some species are available. Those are known as seaweeds, and these seaweeds we have collected. We have collected green algae, brown algae, and red algae. After collection, we have dried the algae. After that, we have converted into powder, and then after we have carried out the oxalate extraction. And after oxalate extraction, we have collected the crude from the different solvent. After collecting the crude from the crudes, we have carried out the anti-cancer activity, and the crude which having good anti-cancer activity, we have separated each and every isomers by using chromatography, and after that we have confirmed the structure of natural product. After that, we have synthesized the natural product via retrosynthesis method. And after that, we have carry out the further anti-cancer activity, and it's showing the same activity that we have isolated from the natural product. After that, we have carried out the computational chemistry. In computational chemistry, we have modified the natural product, and then after, we are at present, various derivatives we are synthesizing with the help of our basic chemistry knowledge. These are the methods how we carried out. Now the last topic, that is called process development. We have designed and synthesized one catalyst. Its name is CuPCS, which is known as copper phthalocyanine tetrasulfonic acid. This copper phthalocyanine tetrasulfonic acid, which is known for the nitration reaction. Normally for the nitration reaction, we require sulfuric acid, but instead of that, we are using the solid acid. This acid is recyclable and reused for many times. This is the reaction mechanism. I'm not going to talk in detail, the reaction mechanism. By using our catalyst, we can synthesize different types of nitro compounds. Now, this catalyst is also useful for the Sand-Meyer reaction. In the Sand-Meyer reaction, first of all, diazotization of amine will take place, and then after, by addition of Ki only, we can convert it into iodo derivatives. This method is also useful for azide formation, means entry we can prepare by using this catalyst. We have synthesized several derivatives by the catalyst which we have developed in our lab, that is called solid acid catalyst. This catalyst is also useful for the reduction as well as esterification reaction. For this work, we have filed 2 patents, and both the patents are published. From this work, 4 publications are also there. Based on the government guideline, that is called DSIR guideline, I am having my own company. Its name is SD4U Diagnostic and Therapeutic Research Private Limited. This is the certificate. We are having MOUs with national and international organizations. These are the list of the projects which we have completed and our supporting partners. These are the name of publishing houses where we have published our articles, name of journals. Here for the young researchers, whenever you are having good results, you should file a patent. If you file a patent, then the value of your work. Why it is important? In 2014, our Prime Minister said that India should be in the top 25 in GII index. Before 2014, we were at 81st position. Now in 2024, India is at 39th position. This is only because of stable government. Government is extensively promoting research parks and technology business incubators and giving further other supports to the researchers. So I think almost most of the students are here. They are familiar. What is patent? Patent is an exclusive right granted by the government to the inventor. So I'm not going to talk about it in detail. Yeah. This is my research group. I have received several awards, ISCB 2025 Best Teacher Award, then WA Science Laureate Award, Senior Scientist Award I have received from Vikram Sarabhai Community Science Center. I have been honored by Indian government on especially on Republic Day parade. I have received ICC Award 2023, IPR League 2022, BIDDS Award 2022, then Innovation Award from Anurag Thakur. He is a cabinet minister. I have taken a training from IIT BHU on AI-based drug discovery. This is our research center where we are working, and some other patents are under pipeline or published. Our 12 patents granted by Indian Patent Office. And last one, I am not only a researcher, I am an athlete also, runner too. Thank you.
Thank you for finishing on time. So, any questions from the audience? Okay, if there are no questions, let's thank the speaker for his wonderful talk.
I request Dr. Ekambram Balaraman to felicitate Professor Hitesh Patel with a token of our gratitude. Thank you so much, sir. Okay, now I would like to invite Professor Ramendra Pratap from Delhi University to felicitate our distinguished session chairs, Dr. Sudipto Basu and Dr. Ekambram Balaraman, as a token of our appreciation for their invaluable contribution for this session. A big thank you to all our speakers and session chairs. We now move on to our final technical session of the day, session 10. We now begin the final technical session, which will be chaired by Dr. Namrata Rastogi, CGRI Lucknow, and Dr. Vijay Lakshmi, Thappar University. Welcome, ma'am.
Good evening, everyone. Welcome to the last session of the day. So the first speaker in this session is Professor Amreen Kumar Singh. He's Associate Professor in Department of Chemistry, IIT Indore. And Dr. Amreen Singh earned his PhD from IIT Bombay under the supervision of Professor Pradeep Mathur. He also received Shyama Prasad Mukherjee Fellowship, CSIR, as a student. He conducted postdoctoral research at the Lund University, Sweden, with Professor A.B. Nordlander, and later at Michigan State University with Professor R.N. Odom. He joined IIT Indore in 2016 and became an associate professor in 2022. His scientific contributions include research in inorganic and organometallic chemistry, focusing on catalysis. They mainly are focused on designing pincer and NHC ligands and their transition metal complexes for a small molecule activation, investigating their reactivity and mechanistic pathways for renewable energy and sustainable chemistry applications. Title of his talk is Rhenium Complexes as Promising Anti-Cancer Agents. Welcome, Dr. Amreen.
Thank you, Dr. Nita. Am I audible?
Okay. So at the outset, let me thank my good friend Vishnu Maya for giving me this opportunity to showcase some of our recent research work. And as the title gives away, I'm going to talk about some ruthenium complexes which have shown promising anti-cancer agents. All these, some of the famous ruthenium complexes which have been popular for showing the anti-cancer agent, NAMI-A, KP1019, NKP1339. These are some of the popular ones. But before I jump onto this anti-cancer agents, let me introduce our group activity, because this is not what we are known for. Our group is not known for anti-tumor or anti-cancer investigations using ruthenium complexes. We are inorganic synthesis and catalysis group. And during our investigations, we have prepared a huge library of new ruthenium complexes. And this is a recent thought, which I will talk about later. But let me first introduce what type of work we are doing for last few 6 to 7 years, so that you will have an understanding where we are coming from, how I'm going to talk about ruthenium complexes as promising anti-cancer agents. So what we generally do, ruthenium metal complexes, we synthesize and we use them for some organic transformation as catalysts. Our tool are ruthenium complexes having N-heterocyclic carbenes as ligands, which are very popular type of ligands these days. And what we have fundamental, interest is the understanding of structure property relationship of these complexes. What are the effects of different ancillary ligands on reactivity of these complexes? How we can tune their stereochemical properties for some desired catalytic activity. So this type of complexes, this N-heterocyclic carbenes in pincer architecture, ruthenium complexes with those pincer ligands. We have explored these complexes for 3-4 years, and we use them for acceptorless dehydrogenation catalysis for hydrogen production from organic alcohols. And the thing I was talking about, tuning stereoelectronic properties and observing the effect of ancillary ligand. So we observed that the effect of ancillary ligand goes in this direction. So we see that carbon monoxide gives us very good catalyst for acceptorless dehydrogenation, while the phosphine containing complexes are not so good. And another related reaction is dehydrogenative coupling between alcohols and amines, and to give the imine or the hydrogenated product of amine. But what we saw and what was very interesting is the reversal in catalytic activity. Normally what we will think is that when we have some complex which is very good at alcohol dehydrogenation, since the dehydration process or the condensation process is a subsequent step. Normally, we think that something which is good at alcohol dehydrogenation should also be good for imine synthesis. But we saw a reversal in this trend, and we were able to explain that trend using this intermediate, that after the alcohol dehydrogenation, one aldehyde bound metal complex intermediate will be formed. And the next step is, depending on this type, the reactivity of this intermediate. When we have ligand like carbon monoxide, which is very good transdirecting ligand, aldehyde is dissociated easily, and the resulting intermediate undergoes another cycle of alcohol dehydrogenation. But when we have some ligand which doesn't have as strong trans effect as carbon monoxide, this remains on the metal, and we also know that aldehydes are orthodirecting groups for CH activation, and that happens, and this metal-bound aldehyde after CH activation becomes a chelate ligand, and on that metal-bound aldehyde, amine can attack, and condensation then happens, and imine forms. So we were able to explain the difference in reactivity and this surprising reversal in catalytic activity using this thought process, and we were able to show the effect of ancillary ligands. Then we moved on to new subclass of protic and heterocyclic carbene ligands. Protic and heterocyclic carbene ligands are related to this common non-protic NHCs. As you can see, when we have an NH group in close proximity to the nucleophilic carbene carbon, we will definitely say that should not be very stable. And of course, in the free form, azole form is preferred. But the strong metal carbene bond makes this protic and heterocyclic carbenes stable, and they can be formed when they are bound to the metal, but not in the free form, only bound to the metal. They can be formed and characterized. Why they are interesting is having an NH group adjacent to the metal gives us some interesting properties. They, with the small and reactive NH group, we can expect that they can be utilized for substrate recognition, or maybe heterocyclic cleavage of dihydrogen, and maybe if we are lucky, metal ligand bifunctional catalysis can also be possible. So these are the things that we are interested in. We have developed a new type of unsymmetrical NNC pincer ligand. We call it CPAP, where we have on the one side carbene ligand, middle we have pyridine, and then we have a aminopyridine sidearm. And this, because of this amino group in between, this becomes an unsymmetrical ligand. On the one side, it will make a 5-membered methyl. On the other side, it will make a 6-membered methyl. And because of that 6-membered methyl, we can also expect that this pyridine can be hemilabile. And that gives additional point of reactivity. So with the N-alkylated azolium salt, we are able to make normal, classical NHC complexes. And if we don't use the N-alkylated ligand, then the metal-assisted tautomerization happens, and we are able to make the protic NHC complexes. And we have characterized them extensively using NMR and single crystal X-ray diffraction. And we have utilized them for complete study we have done and published these papers. We have utilized them for small molecule activations. The protic NHC can be easily deprotonated, and then this thing is called now an anionic NHC ligand. And anionic NHC ligand is able to do hydrogen splitting and make a hydride complex. And when we have this type of reactivity with one metal complex, we can easily guess that, okay, if it is able to do heterolytic hydrogen cleavage, it should be able to do some kind of hydrogenation-dehydrogenation reaction. So that is something that we are very interested in. We'll talk about it. In addition to that, it is able to do CO2 reduction to formate. And in addition, we are similar to hydrogen splitting. We are able to do terminal acetylene splitting and form metal acetylide. And also the alkyl halide so-called activation we can do, and this can be converted to a classical NHC complex. And hydrogenation, dehydrogenation reactions are something which we are interested and many other people are interested because of the potential applications in energy-related domain. For example, the hydrogen production and renewable energy things related with hydrogen, one of the bottlenecks where everybody struggles is, even if we are able to make hydrogen using renewable sources, how we are going to store the hydrogen, how we are going to transport, because it is a gas, it has very low energy density if we look into the gas form. So that is a problem, and there is a thought process that instead of hydrogen, use the liquid organic hydrogen carriers, well, LOHCs. What are liquid organic hydrogen carriers? Some organic molecule, some small organic molecule, which can be hydrogenated. So we can transport the hydrogenated form to some place safely. And then when we have need for hydrogen, we dehydrogenate that hydrogenated organic compound to get the hydrogen. And the dehydrogenated form will be transported back to the source and can be rehydrogenated again. So that is the entire thought process behind liquid organic hydrogen carriers. In this way, you can see that it involves a hydrogen regeneration from renewable sources, and then hydrogen and LOHC will be combined, of course, which we know that it will be called hydrogenation, which will need a hydrogenation catalyst. And the release of hydrogen from the hydrogenated LOHC will require dehydrogenation process, which will require another catalyst, dehydrogenation catalyst. But wouldn't it be nice if we have one catalyst which can do both, right? It will streamline the whole process. Same catalyst if you can be used for hydrogenation, dehydrogenation, and this is a very strong need is felt about this, that we need to have a bifunctional catalyst which is able to do both. And I would say that we are lucky that our protein protic NHC complex is actually able to do both. This part, the hydrogenation part of hydrogenation of nitriles to amines, or in heterocycles, in aromatics to N heterocycles, hydrogen form, can be done using hydrogenation process using our catalyst. And the same catalyst under slightly different, little bit harsher conditions is able to do dehydrogenation also. So this hydrogenation part we have submitted as a paper. It is under revision right now. After that, we will go for this dehydrogenation part, publishing that part. So this we have done. Another thing that we have developed in our lab is ruthenium-3 NHC complexes. Ruthenium-2 NHC complexes, ruthenium-2 complexes are very common. But ruthenium-3 not so common, and ruthenium-3 N-heterocyclic carbene complexes extremely rare. Before our paper came out on this, there were only 4 examples in literature. And all of them were prepared from ruthenium-2 NHCs by oxidation. We were able to make this, as the topic of this conference is also involves sustainable. This is a very simple process. We start with ruthenium salt, trichloride salt, and a simple ligand which is also commercially available, although we make it in our lab, it is also commercially available. And simple THF reflux, no base, nothing required. You can make this compound, which if you look closely, it is actually a ruthenium Cl3 analog. It is still is in ruthenium plus 3 oxidation state. We just have installed one bidentate chelating ligand. One of the ligand part is N-heterocyclic carbene. So now, using this, all the chemistry, all the type of precursors that are prepared using ruthenium trichloride, as I said, our compound is analogous to ruthenium trichloride. We can prepare the phosphine complex with this, similar to how it is prepared from ruthenium trichloride. Okay. After that, similar to ruthenium trichloride, DMSO complexes, how it is prepared, same process we follow, we can make DMSO complexes. Then we also showed that the DMSO complexes can be substituted for the other different ligands. Bipyridine complexes can be prepared. And in that, we also showed that we can also utilize the DMSO, photolability of DMSO, to make these bipyridine complexes easily. So in a series of publications, we have shown that these ruthenium-3 NHC complexes work as a fantastic metal precursor, and we can prepare a lot of complexes starting from here. Additionally, what we found that not only the THF reflux reaction, these compounds can be easily prepared in HCl solution. Normally, preparing N-heterocyclic carbene ligand complexes from the precursor imidazolium or the azolium salts requires base. But here we are able to show that we can prepare these complexes in the acid medium, where the Lewis acidic ruthenium center does a CH activation, halide assisted CH activation in HCl, and it makes carbene complexes. And in this method, we were able to get abnormal and heterocyclic carbene complexes. Okay, so after that, we have shown a lot of catalytic applications of these complexes and complexes originating from this project. And then this is something that we have recently noticed. Although the catalytic application for C-alkylation of secondary alcohols and ketones for CC bond formation reactions is something that has been done by a lot of groups, and the substrate scope is also very wide. But that is not the interesting part. Interesting part came out when we explored its mechanism through DFT. What we found, normally we know that when we have something like this, what happens is that we form an alkoxide intermediate. This alkoxide intermediate goes beta hydride elimination to produce the hydride intermediate, and aldehyde will be released. That aldehyde can interact with the ketone to make the chalcone. And then the chalcone will be reduced by this hydride. But what we noticed during our DFT calculations, that this hydride generation and beta hydride elimination can easily happen through the inner sphere mechanism. But when we went for the subsequent reduction of the chalcone intermediate, we found that the inner sphere mechanism is too high in energy. And at that stage, we explored the outer sphere mechanism, and that turned out to be lower in energy, although we don't normally see that these type of complexes can do outer sphere mechanism. And during that, we also explored how this crossover, this hydride intermediate, converts into this another type of hydride intermediate, which is able to do outer sphere hydride transfer. And even more interesting part is, during the outer sphere hydride transfer, we see that the substrate is parallel stacked on the ligand. That is something very new, because it appears that the hydride transfer for chalcone reduction happens outer sphere mechanism, that's fine. But this outer sphere hydride transfer is supported, stabilized by pi-pi interaction between the ligand framework and the substrate molecule aromatic rings. For exploring that, we have done DFT calculations where we have shown that if we look at this structure, both side phenyl rings, so 2 pi-pi interactions. If we look at the extreme end where we have replaced the phenyl rings with alkyl groups, so no pi-pi interaction. And in between, 2 other structures we have found where one side remains, one side alkyl, or in this side, the ketone side has the aliphatic chain, while the other side has the phenyl ring. So 1 pi-pi interaction intermediates. So transition state. So no pi-pi interaction, 17.3, 2 pi-pi interaction, 10.3. And when we have 1 pi-pi interaction, you see that both are nearly halfway in between these two energies. So it further supports that actually the pi-pi interactions are supporting or favoring this the outer sphere hydride transfer. So after that, now I move to what I gave the title for, because we have prepared a huge library of ruthenium complexes. We thought of, other than our primary interest of developing energy efficient processes or sustainable processes for various catalytic organic transformations, catalytic organic transformations, or hydrogen storage, hydrogen transport mechanism things, why not explore these things for anti-cancer agents? As we know that the NAMI and some other related compounds have entered in clinical stage trials, but after that, another type of mechanism was found. One of the popular concepts in the anti-tumor activities of ruthenium complexes is activation by reduction, in which it is believed that ruthenium-3 oxidation state complexes, when they enter in body, they are first reduced to ruthenium-2, which is the active form of the drug. And this reduction can happen more in cancerous cells because they have more acidic and more reducing cell environment. So this should give us some more selectivity towards cancerous cells. So that is the thought process, and the activation has been explored for the KP109 and KP1339, and these are the mechanism report that ROS species will be formed, and that will cause mitochondrial damage leading to the apoptosis, or some other mechanism is also possible which results in cell cycle arrest. Other types of ruthenium complexes, RAPTA, where the name comes from R from ruthenium, A for coming from arene, and the PTA. So these are also, one famous family for ruthenium anti-cancer compounds. And then a new type of ruthenium polypyridyl complexes are known. So you will notice that I'm coming to the structurally similar complexes from our group. Okay, so main bottlenecks, as our previous speaker reported, that the dynamic therapy is one of the popular concepts. Ruthenium complexes are also utilized for PDT and photoactivated chemotherapy. So encouraged by this, what we think that we have this ruthenium 3 energy, which we can explore, and the derivatives coming from these ruthenium 3 NHCs, the DMSO complexes, the bipyridine complexes is similar to the polypyridyl examples we are seeing, and the abnormal NHC complexes. So right now, we only have data about the ruthenium 3 NHC complex and the abnormal ruthenium 3 NHC complex, and we see that there are some differences, slight differences. I'm not showing that data because it is too early stage, but we see that they show a little bit reactivity towards anti-cancer explorations, and the structural differences between normal and abnormal NHC complexes are also visible in their differences in their behavior. So with that, I will summarize that in our lab, we have developed different types of ruthenium NHC complexes, protein NHC complexes using a CPAP ligand platform, where we have shown small molecule activations and hydrogenation, dehydrogenation of liquid organic hydrogen carriers. Then in our alpha alkylation of ketone explorations, we found the pi-pi interaction supported outer sphere hydride transfer for chalcone reduction, which can be considered as a new type of metal ligand cooperativity, but still it is a little bit early in stage. And then we have just started our ruthenium complexes for their anti-tumor, anti-cancer activities. I would like to acknowledge all my students, and biological study is being done with Professor Hemchandra Jha in collaboration with Professor Hemchandra Jha at IIT Indore. And thank you for your kind attention.
Thank you, Professor. Thank you, Professor Ambreen, for finishing on time. And in the interest of time, I can have 2 questions.
So first, I say there's very interesting piece of work, but I have 2 comments. In the very first slide, you showed the structure and other KP-related ruthenium complex, but in the instead of ruthenium, you mentioned platinum in the very first slide. So maybe it's a kind of mistake, maybe. The second comment I have, maybe, yes, the first, second, third.
Oh, yeah.
The KP, NKP, and NAMI, they are ruthenium
yeah.
complex. I mean, just mistake probably.
Yeah.
But second comment is that these ruthenium complexes are known kinase inhibitor. So have you thought of trying those molecules for kinase inhibition?
No, I have not yet. Like I mentioned, it is a very early stage, but still, when Vishnu Maya, sent me invitation, I was wondering, how did you know that we have started working on this? Because our group has not published anything in this domain. Okay, so I asked you, how do you know now that we have started working on this and you are calling me in this conference? So, but yeah. So it is
There are
early
quite
stage.
few reviews on ruthenium complex as kinase inhibitor.
Yeah.
So if we refer to those, maybe it will be helpful.
Thank you.
Thank you.
So as you have prepared a lot of complexes,
Yeah.
and ruthenium, these types of complexes are very much popular for the electrochemistry.
Yes.
So have you tried on that? Because a large number of these organic molecules you are trying to synthesize, but that is only to find out in what is.
Electrochemical CO2 reduction is one thing that we have tried. And we are getting some result, but I have not included that in this. But electrochemical CO2 reduction, yes, we have seen format is easy, but we want to go to either methanol or to methane. So format, we are able to see that we are easily getting the format, but we would like to go few steps further to methanol at least. That we are exploring.
Thank again, Professor Amreen Singh. Thank you.
I request Dr. Namrata Rastogi to felicitate Dr. Amrendra Kumar Singh with a token of gratitude. Thank you so much, sir.
So the next session is a special session by RSC. And on behalf of the conveners and organizers of the conference, I would like to thank RSC for sponsoring 5 poster prizes and 5 flash talk prizes in the name of their flagship journals. And this session will be convened by Dr. Rajdeep Roy and Dr. Ayushi Arora. Dr. Ayushi Arora currently holds the position of Assistant Editorial Development Manager at RSC. She did her PhD from Doon University, Dehradun, and she has been a recipient of Indian-US Fellowship for Women in STEM. She completed her postdoctoral research training at Texas A&M University, and her research work focused on advanced organic and inorganic chemistry. Currently, she's, as I already told, she is an Assistant Editorial Development Manager at the Royal Society of Chemistry. And her work on platinum group metal chalcogenides, palladium complexes, and metal organic framework has significantly advanced the field of catalysis. Another speaker in this session is Dr. Rajdeep Roy. He holds the position of membership executive at Royal Society of Chemistry and peptide chemist. He's a peptide chemist. He did his PhD from, he did his PhD from, I'm sorry, I can't see that. Currently, he is serving as membership executive at the Royal Society of Chemistry. He has an extensive research background. He did postdoctoral training at University of Notre Dame and Hebrew University of Jerusalem, Israel. He earned his PhD from ISCS Kolkata, and his research focused on dissipative non-equilibrium self-assembly and biomedical polymers and peptide self-assembly. So over to you, Dr. Rajdeep Roy and Dr. Ayushi Arora. Thank you.
Thank you for such a nice introduction. And at the onset, I would like to thank the organizers for giving me the opportunity to present. So let me share my screen with you. Is there any permissions required, because it's not allowing me to share my screen?
You already have permission. Please check your system, Can.
Are you able to see my screen now?
Okay, it's fine.
Yeah.
Yes.
It's in slideshow mode, right?
Yeah, and
Yeah.
Dr.
Okay.
Arora, just to remind you, you
Yeah.
have 20 minutes for the presentation. Thank you.
Yeah, sure. Thank you. So, good evening, everyone. I am Ayushi Arora, and I would like to very quickly walk you through some of the initiatives of Royal Society of Chemistry in integrating chemical sciences community and beyond. So, as you know, Royal Society of Chemistry has been there since 1841, and currently we hold a network of more than 60,000 members across 125 countries. We are a leading non.
Dr. Arora, can you
Is it?
please, yeah, full screen mode?
Yeah, is it in full screen mode now?
Not yet.
Oh, sure. Now, because it's in full screen mode.
No, we cannot yet see that.
Okay.
Press
Maybe you
F5.
can stop share and reshare, Ayushi. I think that will help.
Yeah, sure. Just one moment.
5 on your screen.
Ayushi, please share F5.
Okay, okay. Um, yeah, 5. Is it now?
I think we are good. We can see. It's okay. Let's move on.
Dr. Arora, please go ahead. Should be fine.
Okay. So as I said, we have a network of more than 60,000 members across 125 countries, and we are a leading non-for-profit publishers. We are also a part of our Royal Charter. We have our head offices in Cambridge, and we are well represented by our offices from Washington DC and Philadelphia in the west to Beijing and Tokyo in the east, with our India offices in Bangalore and in Delhi. And just to give you a glimpse of the submissions and the publications, we are India is the second largest contributor in terms of both submissions and publications from India across all portfolio of our journals, which starts from analytical to food chemistry to nanosciences to environmental sciences and all. And in this regard, I would like to introduce you to one of our very recent journal, which is RSC Pharmaceutics, which is the scope of which lies in drug delivery, formulation, and pharmaceutical technology. So, as led by Yvonne Perry, who is the editor-in-chief of RSC Pharmaceutics, the journal has completed its 1 year anniversary in February 2025 this year. It was launched last year, and there are the issues that are out. So, please do read out to those issues. And also, some of the latest theme collections from India include the recent one in ChemCom, which is a Chemical Research Society of India medal winner collection. Also, the Journal of Materials B collection celebrating the 100th birthday of Professor Sukhdev, which is in organic and biomolecular chemistry, and also the one in environmental science journals. So, apart from this, we have an open call for papers in one of our gold open access journals, which is Sensors and Diagnostics, on advances in nanomaterials for sensors in early diagnosed diseases, which is guest edited by two of our CSIR scientists, and Dr. Suman Singh and Dr. Murugan. Are my slides moving?
Your slides are not moving, Dr. Vira. Now it's moving, yes.
Oh, so I don't know what the
And
problem is. I mean,
currently it says, sorry, time stuck.
it's okay. So okay, it's okay. I'll just briefly explain these things again. Is it moving right now, right? So RSC Pharmaceutics is a newer,
Yes.
yeah, it's a newer journal that has been launched in a drug delivery and formulation, and it's completed its 1 year anniversary this it completed its 1 year anniversary this year in February 2025. So we also have several themed collections from India in various journals across Royal Society of Chemistry, starting from Chemcom to our environmental science journals. And the latest themed collection that is in progress is in sensors and diagnostics, which is on the topic of advances in nanomaterials for sensors in early diagnosed diseases. So not just these, there are several other open call for papers that are live, and according to your interest, research interest, you can browse and submit using the link, which is call for papers in chemical sciences and related discipline. So the one is for advanced catalytic materials for energy and environmental applications, and the other one is NRSC advances on a spotlight on recent appointees. We are well represented by our board members across all our journals. We have several associate editors on board with us, right from Chemcom, which is Professor Sandeep Verma, who is an associate editor, and to JMCA, in which Professor Kundu is one of the associate editors with us. We do welcome our new Indian board members. Uh, uh, on several journals, from Professor Mallareddy, who is the editor in chief for Crystal Engineering Com, and Professor Sachidanand Tripathi, who is the advisory board member on environmental science for atmospheres. Also, Professor Singhal, who is the associate editor for sustainable food technology. In addition to that, we have several of our events, which is one of them is like Meet the Editor, RSC Meet the Editors, which gives you a chance to ask directly to our editors your queries, which one of them happened at ISER Pune and CSIR NCL last year. So we have several others lined up this year, so please do follow RSC in their Meet the Editors programs. Also, Chemcom completed its 60-year celebrations with our community, so we had a road show at IIT Chennai, IIT Kharagpur, and IIT Kolkata. Also, we have one of our major flagship event, which is Chemical Science Leader in the Field Symposium, which last year happened at ISET Trivandrum, and uh, we and this year also, please do check out for the dates of this symposium, as it is an annual event. We have several publishing resources available for the authors in our authors and reviewers hub, which is one of them is how to publish your research. So not just how to publish your research, several other guide booklets like how to increase your impact or how to review for Royal Society of Chemistry. So there are several research guides which are available for you, so please do look out and download and read them accordingly, so that you get the best impact for your paper. And there are several publishing resources on how to write a paper. There are certain things that we need to think about, title and abstract being the most important, since these are the first readable items on your manuscript. Also, spelling and grammar, and proofread your paper very, very carefully before submitting it to the journal. So the next thing is, just the life cycle of a manuscript. I just wanted to give you a glimpse of it, which is, you submit your manuscript, then we have an initial assessment. Then if it passes the initial assessment, you go into the peer review. Then it's edited, evaluation by editors, which either leads to acceptance or the rejection of the manuscript. Or if at any stage, you can always make an appeal for the decision that is made on the manuscript. Also, one of our recent launches is the Journal Finder tool of Royal Society of Chemistry. So you can keep a track of your open access options, search with certain keywords using your research keywords, and you can get the required journals which will be the better suited. So not just journals, this all things were about journals, not just journals. We do publish books as well. For over 5 decades have been there. RSC has been publishing books. So, and these include specialist periodical reports, short form books, professional reference books, and several other options. We have a new platform which was launched very recently, and it has several options available for books. So please do check it out. And also, if you have a proposal in mind, so you can reach out to either me or us at books@rsc.org uh, for any book proposal. All book proposals are subjected to a peer reviewed process before they are accepted. But if you have a proposal in mind, we would love to hear from you. And apart from the publishing activities, we have an education outreach program, which is a Yusuf Hamid inspirational science program. So it's divided into 2 modules, which is teacher training and the chemistry camps, in which we train and equip teachers across India for their specialist knowledge, as well as we motivate some of the brightest chemistry minds from all backgrounds to study chemistry and take up chemistry at a higher education level. So with this, we have actually trained so many science school teachers across India, and also more than 32,000 school chemistry students have been trained through their 52 Yusuf Hamid camps. So with that, I would hand over the presentation to Professor Rajdeep Roy, who would now take you through how you can be a member of Royal Society of Chemistry and what benefits would it serve to you to be a member of Royal Society of Chemistry. So this is the team that I work with, and thank you. So over to you, Rajdeep. I'll stop sharing.
Thank you, Avishi. So first of all, am I audible to all of you?
Yes, you are audible.
Yes, you are. Thank you. Thank you. Thank you. Let me just share my screen as well. Okay, excellent. So hopefully are you are able to see my slides, right?
Yes, it is visible.
Thank you. Thank you. Thank you. So is it in that slideshow mode?
Yes,
I
yes,
think
that,
it's working fine. Got it.
yes.
Got it.
Maybe you can
Thank you.
just go to the next slide so we can confirm if it is moving
Ah,
properly.
yeah, yeah.
Perfect,
Uh,
perfect.
Oh,
Please go ahead.
excellent. Thank you. Thank you. Thank you. So yeah, as my colleague Aishi has given a very fantastic, overview overview or an idea, I would say, regarding our publishing activities. Thank you, Ayushi, first of all. And at the outset, I would also like to kind of thank you to the organizers, Professor Bisai, and all the other organizers at the Industrial University for organizing this nice conference and allowing us to talk over there in virtual mode. So what I will do for the next 10-15 minutes would kind of take you to an different aspects of our organization. So as Ayushi has quite rightly mentioned that we are a non-profit uh, uh, society publisher. So as a part of this society publisher, we do have a very strong membership activities throughout the world. And myself, Rajdeep, I'm taking care of the membership activities here in India. So for the next 10-15 minutes, I will give you an idea that how our membership would eventually give you some assets or kind of some inputs in your career which will help you to move forward. So as a whole, I will talk a little bit about our membership as well as our networking opportunities, our policies and work, and our different funding opportunities. And at the same time, I will give you a little glimpse about our professional recognitions like chartered camps. So right at the very beginning, let me just tell you that our membership provides mainly 3 kinds of benefits to our members. You can categorize it as simple as like resources, support, and connection. And one by one, I will come into this. So as far as these resources are concerned, but before going into that, I will just talk about a little bit on this particular slide that what is all this membership is all about actually. So the membership is all about just to provide you a platform where all the people from chemical sciences or related to chemical sciences can come together to a platform. They can exchange their knowledge like any conferences which is going on in at your place or worldwide. They can exchange their knowledges. So chemistry has no boundaries. So it has no boundaries like in terms of color, in terms of gender. So we never believe in all these things, so that everybody should exchange their knowledge and can influence each other. So that's the main purpose of this membership. Now, before going into the details, I just kind of want to give you a little highlight on this slide, and I believe that this slide is very important for all of you, because in RSC we do have different categories of membership. Now, what does these categories actually mean? So right from the left side of the screen going towards the right hand side of the screen, the seniority of the membership increases. That means that this affiliate or students member are for those who have started their career. Mainly student members are those who are in UG chemistry or chemical science related courses, and affiliate members are for the mainly PG and those who are pursuing their PhDs. Now, as soon as you will have a PhD degree, you can apply for an associate member, which is in short called AMRSC. And then from AMRSC, the next step is you can upgrade your membership to MRSC. So MRSC needs at least 3 to 4 years of post PhD experiences, and that experience can be any academic experience or any experiences working in an industry or any academic institutes whatsoever. So you do need a post PhD 3 to 4 years of experience to apply for MRSC, and the senior most category is obviously FRSC. Now, before going into that, in on top of this membership, different membership categories, we do have some professional recognitions as well for our members. Like for our junior members, we have a registered scientist technicians. You can apply for that. At the same time, for AMRSC, you can have a registered scientist category, and those who are in senior professional categories like MRSC and FRSC, they are eligible for chartered chem. Now, before going into the details, I will focus my talk mainly on the different benefits aspects of our membership, as I have already mentioned. So as far as the resources are concerned, but before going into that, I will just tell you that this membership would give you a recognition. But what does this recognition means? This recognition means that you can use your membership identity as a post nominal letters anywhere. Like in social media, I have shared my own LinkedIn over here, that after your name you can share your post nominal credentials as per your membership category, which will give you a wide opportunity for your career advancement, in terms of your career advancement. And as far as your professional development is concerned, we do have lot of career consultations available for you. Now, as far as these resources are concerned, my colleague Ayushi has mentioned in detail that what is our publishing aspects offer for our community. Now, I'm not going into the details, but as you can see from this nice cartoon image, that apart from different journals, we do have couple of magazines also published with us quite regularly. One of the magazine is called Chemistry World, which is a monthly magazine, and the other magazine is called Voice, which is a quarterly magazine. And obviously, apart from journals, we do have our historical collections, a lot of books archives, and those who are teachers, for them a lot of educational resources are also available in RSC. Now, as long as you are a member, you can have an access of all this, but there is a few restriction in some of the cases. I'm coming into that. So before going into the details, I just want to give you an idea that, as I have mentioned you, that there are a couple of magazines published with us, so those members can always access both of these magazines. For the junior members, it will be an online version of this magazine, and for senior members like AMRSC onwards, the hard copies will be couriered to your physical address in regular interval. As far as the journal accessibility is concerned, which is a major asks from our community, I would say that in currently we do have 59 journal portfolios available with RSC, but out of those 59 journal, according as per your wish, I would say rather, that you can choose any one of our journal, and you can have a free unlimited access of surfing of that particular journal unlimited time. But you have a restriction as far as the downloads are concerned. You have a restricted downloads, like for the junior members starting from 10 downloads a year. For AMRSC it will be 15. For MRSC 20 and FRSC 25 downloads are available for all the members. Now those who are, suppose for example, is interested in organic chemistry, he or she can choose OBC, or maybe in any general chemistry journal like Chemcom or whatsoever. At the same time, that downloads also includes different book downloads as well. And at the same time, we do have a database available with us with a lot of molecules. It's called Chem Spider, and as long as you are a valid or active member, you can have an access of this Chem Spider as well. As far as the support is concerned, we do have a dedicated consultation team available in our UK office. They are always happy to kind of support you online anytime, either via Zoom or any kind of a virtual network, for your CV assessment, for your any one-to-one consultations. Like for example, in distress, you can also have a your mental counseling as well with our expert team. And as far as the CV assessment is concerned, it is very important for our younger members, because we believe that the CV which we use for our suppose academic purpose is quite far different, which we usually use for applying for any job. So how to construct a good CV? So this kind of an information or support is available with us. You can always have an access of this kind of an career consultations as long as you are a active member. This is the particular web page from where you can book your consultations anytime. Now I'm coming to the connection part. So membership always means connection. As I have already mentioned, that this will give you a unique connection worldwide. But starting from our smaller diameter, I would say that as long as you are a member, you will be a part of our local section. Now in RSC we do have local sections throughout the world, and in India we have 5 different local sections geographically situated. Suppose for example, as your university is located in the western part of the India in Gujarat, so you'll be a part of the West India local section as long as you are an active member with us. So anything regarding West India local section, any events or any kind of activities is going on, you will be notified in your registered email ID. As far as the subject community is concerned, so we do have 8 different subject communities available with us, and 71 special interest groups out of those different 8 subject communities. Now what does this subject community and special interest groups mean? It means that subject communities are like the vast area which you usually come across when we study chemistry, like starting from physical chemistry, organic, inorganic, analytical, energy, material. This kind of 8 different subject communities are available, and you can choose up to 8 different subject communities, and relevant informations, current coins about those discoveries, job opportunities will be notified in your registered email ID. And out of those specific 8 subject communities, suppose those who are working in a in inorganic chemistry, maybe in artificial photosynthesis, or those who are in organic chemistry working in peptide, they can also choose the special interest group, like very specific interest groups associated with us in our RSC, like our peptide group, artificial photosynthesis group, like coordination chemistry group. This kind of different 71 specified groups are available. You can be a part of it. But regarding specific interest group, max up to 3 you can choose for junior category of membership, but for senior category of membership like AMRSC, they can choose up to 4 special interest group, and for MRSC and FRSC 5 interest groups you can choose, and you will be a part of that one, and relevant updates and different opportunities will be notified. As far as the event, symposium and workshop is concerned, as long as you are a member, you will have a flat 15% discount, mostly in our worldwide conferences. But right now here in India, most of our RSC events are free. I'm talking about the global RSC events which are going on throughout the calendar. For those, you can have a 15% off in your registration fees as part of your membership benefits. And here I just want to mention one thing, that as part of our program here in India, from our membership team, we do have a specific program which we usually conduct throughout our country is called Chem Careers. So it's a typical RAC in-house events where we usually focus on different career aspects of chemistry, consider keeping in mind the benefits of our younger generations. So we usually do this kind of a program in an institute, and we usually try to invite other neighboring institute in that particular events as well. This is very interesting, where we usually ask our resource persons to talk about different career opportunities that can be from academic point of view or can be from industry point of view as well. So I believe that for the younger generation, those who are conducting PhDs are going to submit their thesis very soon, or those who are conducting even MSc also, for them it will be a very nice and informative event. At the same time, I will just focus a couple of minutes on the last point, that is called the Student Chemical Society, which is pretty much similar like student chapter available with us. So as soon as number of students, like for example 20-30 students join from a particular campus, they can form this Student Chemical Society anytime within their campus, and everything will be governed by the student community. You have to select one chairperson, you have to select one treasurer as well, and uh, RSC would be happy to support your activities throughout the year, and you will be given up to 250, which is almost like 25,000 INR, that is 250 GBP per year, to conduct this kind of an events like small popular lecture or chemistry quiz with your surrounding or neighboring colleges, something like that. So I would always encourage students, if you are joining in bulk from your campus, you can register for this one. We do have a specific web link for this particular SCS network. You can go, you can visit this network, and a form is available anytime. You can register, but for this one, everybody, those who are part of this network, has to be a member with us. Means at least they have to be affiliate member for the younger in terms of student community I'm talking. So next, as far as the AMRSC and MRSC benefits are concerned, mostly the benefits are same. Apart exceptions are for few supports for funding related thing, which I'm coming later. And as far as the FRSCs is also concerned, we need more advocacies and collaborations from their side, and we use our FRSCs as a resource persons going forward in our different events as well. Now I will take a couple of minutes just to quickly go through some of the slides, which is I believe very much informative and helpful for our younger generation, and as well as for those who have started their independent personal career as well, like assistant professor or associate professor, even associate professor as well. For example, this is one of the funding opportunities available with us, is called Researcher Development and Travel Grant. This particular grant is available for every membership category, and this grant can be attending any kind of conference abroad and even in India as well, and you can use this money in your registration or in your airfare expenses. This can give you up to 500, that means little more than 50,000 one time. And as you can see, 31 successful candidates were there last time from India in 2024. This is another grant. This is applicable for senior members like AMRSC onwards, AMRSC, MRSC, and for FRSC only. This is called Inclusion Diversity Fund. This will give up to 5,000, that means little more than 5 lakhs. This is another very interesting grants. This grants is called Sustainable Laboratories Grant, and for this one, your proposals would be on green chemistry or sustainable chemistry, which is a major concern right now worldwide. And this particular grant will give you 10,000, that means more than 10 lakhs as a consumable fund. Last time, two successful projects were there for India. This research fund is available for everybody. Everybody means obviously MRSC onwards, but without any specific area of research. It depends on what area you are working. You can write a research fund. This is again a very floating ad. Rounds of advertisement usually come out in our website. You can check our website all the time, that when it is available, you can go to funding. You can simply write funding RSC, and you will get this kind of an options available. As far as the funding is concerned. And this is a very interesting one. 9 successful candidates were there in 2024 last year. Again, this will give up to 5,000. The last one again 5,000. This is a research collaboration grants. For this one you have to show a collaboration within, even within country as well. Even you can show a collaboration within your campus as well. So it doesn't matter that where you are collaborating, but you can use this kind of a money. Again 5,000, that means little more than 5 lakhs, in using your travel, in using your any kind of research expenditures like consumables, etc. So these are the typical options that I have mentioned as far as our Indian members are concerned. So mostly these 5-6 grants will give you 5,000, except that sustainable grant which will give you 10,000. And on top of that, each and every year we always try to motivate our members. We do have a lot of prizes and awards for our members as well. Do follow us in RSC's prizes websites. You can have a lot of informations regarding this one as well. Now, how to become a member, as this membership is obviously dependent on your career stage. So as long as your application form will be submitted, you have to submit your resume and your past transcripts as well. And for junior category of membership, let me just tell you, no need of referees. You have to give one supporter's name or referee's name, just for filling up the, just for the form filling purpose, but nobody will be contacted for affiliate membership. But for senior membership like AMRSC onwards, AMRSC need one referee, and MRSC and FRSC will need two referee recommendations for applying for this particular membership. And obviously this is not a free membership, this is a yearly paid membership. But the good thing is that, keeping in mind the economic background of our country or whatsoever, the good news is that we do have a flat 50% reduction in each and every category. But most importantly, for junior category like affiliate, we do have a 90% almost flat reduction of our original rates. As you can see, it's only £11 a year, which is little more than 1,200 right now going on for the junior category membership, like student membership or affiliate membership, those who are still studying. And for MRSC it's 80. For AMRSC is little less than that, 49. So as far as the seniority is increasing, the membership fees is also increases. Fellow, you have to pay 92, which is exactly the half of our global rate. One more very important thing, those who are affiliate members, they can upgrade their membership anytime to senior category like AMRSC or MRSC as per their career progresses. And for that one, until they are not employed, they can still pay £11 for their senior category of membership as well, like AMRSC and MRSC. This is an exceptions for those who are still studying, but for those who are in their independent career stage, they need to pay the flat 50% rate, that is 49 and 80. So that's it from my side. Thank you. If you have any questions, you can write to me in this email ID. And thank you very much. Happy to answer questions. Thank you.
Thanks, Dr. Prashant. The session is open for questions.
Thank you.
Take a couple of questions from the audience.
I have a quick question to Ayushi.
Yeah.
So, hi Ayushi,
Hi.
about your book proposal, I would
Yeah.
like to know from student side and even from my side that if somebody is interested in that, hello,
Yeah, yeah.
If
If
somebody is
somebody is
interested
interested.
for your book proposal, what exactly for in those proposals, like
for selection.
Once you have a proposal in mind, so we have a, so you write to us that maybe that, let's say you contacted books team, so they will send you a detailed form which you have to fill, in which you have to fill uh, everything, what would be the topic of the proposal that you are that you want to submit, and other details regarding it, the guest editors and all that. And that detailed proposal is is peer reviewed by a team of experts, and after that they reach out to you with the decision, what decision they made on that proposal, that whether it is accepted or is it not accepted.
Oh,
So
so
that's
in your
the
form, exactly. So my question is same that
Hmm.
we would like to know what exactly, like, yeah, suppose somebody submits the proposal. Okay, so once we like complete the form, then only we would like will be able to understand that. But so
Yeah,
we don't know about
you
your
need.
form, right?
So
Or is it
we,
available?
I mean, yeah, it should be available. It can be shared with you. That what details? It's a very detailed actually form. So if you
Can
have
I
a
jump in
proposal
here?
file,
Yeah.
yeah.
I can jump in here,
Hi,
Vishnu.
Aparna.
I
Thank
think,
you so much.
hi. I think what we would need from your end is a list of topics that you would like to cover in the entire book, suggested contributors for the entire book chapter, and like who are the people who are going to contribute to that. How different is your proposal going to be to the existing literature? So we want a very comprehensive understanding of that aspect while you are sending us a book proposal. So all these things can be included in the book proposal. If you want to have a pre-look before you start thinking about the book, we can send you the form for you to have a look, and then I think that will guide you through the steps, and that will also bring in your thoughts together to put at a place, and you will also start thinking about who are the right experts that you would want to involve while you're writing or choosing the editors alongside yourself. So I guess that's how we would like to go ahead with.
Okay, okay. Thank you so much. Yeah, so before we conclude that and I give it to the session chair, I must mention all our audience here, students and our faculty experts and senior professors, that RSC for this conference on sustainable innovations in drug development from Indrachil University, RSC has sponsored us, extended their support in the form of 8 certificates, 5, not 10 certificates, 5 poster prizes, as well as 5 oral presentation prizes, along with cash prizes. Apart from that, we got 80K separately for to be spent in different heads to execute this conference. So that's a I think we RSC needs a big round of applause for this noble cause. Thank you so much, Aparna,
Thank you,
Ayushi,
Maya. Thank
team
you so much. Yeah.
RSC. We appreciate your efforts towards scientific communication. Thank you so much. I give it to the session chair, Dr. Namrata Rastogi.
Thanks, Dr. Arora, Dr. Roy, and Dr. Ganguly. So, on the behalf of organizing committee, I again, convey the appreciation for not only for supporting the prize, but also for your presentation. Thank you so much.
Thank you.
So I would now hand over mic to the next chairperson, Dr. Vijayalakshmi from Thapar University, Patiala.
The last speaker of this session is Dr. Kamaldeep Paul from Thapar University. Dr. Kamaldeep Paul did his PhD from Guru Nanak Dev University, Amritsar. Then he spent some time as a postdoctoral fellow in the Yonsei University, South Korea. After that, he joined the Ranbaxy Labs, Gurgaon. And after that, he moved to Thapar University for the academic position, where he is currently at a professor position. He has more than 120 publications in his credit, and the title of his talk is Transition Metal Catalyzed CH Functionalization of Bioactive Heterocycles, a Directing Group Approach. I welcome Dr. Kamaldeep Paul on stage.
Okay, thank you, Dr. Vijay Lakshmi, for the introduction, and I would like to thank organizing committee for inviting me to share my research work on this form. So the topic of my research work is this transition matter catalyzed CH functionalization of bioactive heterocycles, and here I'm using the directing groups. So this approach I have used from the last 5 years. So this work is started with my first review article that is published in ChemCon. After that, I have started this work. Okay, I will go through directly with short introductions. So why we are using this CH activation? So these are the beneficial for the using of the CH activation than the other reactions. So this is the simple substitution reactions where the various limitations are there. Some limitations has been removed with the coupling reaction, but with the catalytic CH activation, so that we are getting the atom step economy, low wastage, reduced reactivity can be achieved, and the various functional tolerance are there. So using the CH activation for this reaction. So here can be formation of the various types of side products with the desired product. So that's why there is a requirement of the directing groups. So directing groups, this direct only at the one position. Either it is the ortho position, meta position, or the para position. That is dependent upon the directing group. But we can achieve the reduced reactivity of the compound. So here the various approaches are used for the this CH activation. So in the one case, the directing group is there and the functional group is added, and this directing group is not removed from the substrate. The second one, where the transient directing group is there, where the directing group direct for the functionalization, and then it is removed from the substrate. And then traceless directing group, where the functional groups are used for the functionalization and it is also removed. But here in this study, I have used this no removable directing group. Here we have used the two pharmacophores, and one pharmacophore will act as a directing group, and the second pharmacophore will act as the substrate where the functional groups are added. So that's why we have made the various conjugations of these either directing group or the substrate. And we have combined these two moieties and formation of one molecule, that is the conjugated molecule. So here we have used the various pharmacophores, like we have used the quinozoline, which is used for anti-cancer, coumarin, that is used for anti-cancer, there's naphthalamide that is used for anti-cancer, and other biologically active pharmacophores we have used. So with this, we have identified some of the pharmacophores like quinozoline, coumarin, indole, pyrazole, naphthalamide, and phenanthromidazole for our studies. So in the first case, we have used the quinozolinone and the coumarin conjugates. Here, this coumarin and quinozoline, when use the various acrylates or other alkenes, the reactions can be possible at the quinozoline ring. So why we use the coumarin? Because the coumarin have various biologically active site. So why we have used the quinozoline? Quinozoline also have the various biologically activity. So we have combined these two and make the conjugate. So in the literature report, when the quinozoline are having the the CH activation taken place at this ring, that is due to the presence of this amide, and the NH is responsible for this reaction. So various types of products are known in the literature where these phenyl rings are alkylated. And when we tried this reaction, so we have changed this reaction when we have used the coumarin instead of this phenyl ring. And other, we have changed this carbon. This carbon we have substituted with the methyl group. And surprisingly, we are not getting this type of a product. That means we are getting the other product, that is the alkylation at the quinozoline, not at the coumarin. So here, this is the various substrate scope that we are getting the moderate to good yields of a compound, where we have used the various acrylates. We have used the methionine ketone and getting the 78% of yield. The substrate scope has also been increased with the substitution with at the coumarin ring, where the methoxy group electron donating or withdrawing groups we have used, and the electron donating groups getting the better yield than the other one. Even the phenyl rings we have used in the coumarin, and good yields of compounds we are obtained. when we study this mechanistic insights of this reaction, we have converted this amide group into the chloro so that we can find out if the amide group is responsible for the reaction, for it acts as a directing group. So here we found that there is no reaction, or even traces of compounds are not formed in this case. And when we have done the deuterated labeling, we got this carbon with 100% deuterated exchange. So that proves that the reaction taken place, this hydrogen is responsible for this reaction. And kinetic studies, we got this 2.63 value where we find out that this CH activation is a rate determining step. In the combination of methoxy and bromo, the methoxy with the electron withdrawing, electron donating groups are getting the good yields, and the reactions are faster than that of the bromo. So from this, we have proposed one mechanism that here the ruthenium is forming the complex with the oxygen, and then it forms a cyclic complex, and after migration and beta hydra elimination, that forms the final product. After that, after using this quinolidine, we have now used the coumarin at the separate moiety, and then we have used with the pyrazole. So we have combined these two moieties and formation of the substrate for the next CH activation reaction. So in the literature, we have found that this pyrazole is responsible for the CH activation, that this nitrogen is responsible for CH activation of the alkene. So we have tried this reaction with the acrylate. So we have used the 3 substrate, and we tried to this reaction in one pot, and we are getting this product. But again, we have separate find out the intermediate, what intermediates are formed in the reaction, and even we are able to separate these intermediates. So we have used the time-dependent studies. So in 10 minutes, we are getting the shape base, and after 1 hour, the cyclic products are formed. But, now we have checked if this ruthenium catalyst is responsible for the reaction. So we have done the reaction in the presence of ruthenium, in the absence of ruthenium catalyst, and we are getting the same product. And after that, when we have used the ruthenium catalyst, then we are getting the alkylination at this carbon. So with the deuterium experiment, we are getting the 50-50% on the both side of reaction. And with the kinetic isotope, this value comes up to be 2.05, which is again the CH activation is the rate determining step. So from this, we have find out the mechanism, and we have proposed this mechanism with 3 cycles. One where is the formation of the ship base, and this ship base is used for the cyclic product where the coumarin and pyrazole conjugates are formed, and then the using of the ruthenium catalyst and formation of this product by the migration insertion and the beta hydride elimination reaction. So here we have separated the intermediates, and excellent yields are obtained with these intermediate, and these intermediate are further used for the reactions with the alkene and formation of this final product, which is characterized by single crystal as well as the proton and C13 NMR. So in the substrate scope, we are using the various electron donating groups and electron withdrawing groups, and here we are getting the good to moderate yields of compounds. And these reactions are also compatible with the vinyl sulfonide, and again we are getting the even the low yields of compounds are formed in this case. So next we have conjugate the coumarin and the benzimidazole. Why benzimidazole? Because benzimidazole is also used as a very biologically active moiety. So here this conjugate, when alkylated with the alkene and in the presence of palladium acetate, and the formation of this product with the cyclic product formation. So here when we alkylated this carbon, then there will be no formation of this alkylation only. We are not getting this product, but we are getting this cyclized product. And we have changed And we have changed this ruthenium. We have also tried this ruthenium catalyst, but we are getting the low yields of compound with the ruthenium catalyst. So we have changed the catalyst to the palladium catalyst, and the yields are improved. So with the ruthenium catalyst and the previous reaction conditions, we are getting only 70% yield. But with the optimized reaction conditions and using the palladium acetate and in the DMF and copper oxide, we are getting the 80% of yield. So with this standardized reaction, we have used the various substrate scope of this reaction. So here we are getting the number of compounds with the different acrylates, and even vinyl with high vinyl ketone, we are getting the good yields of compound. We have changed the, we have increased substrate scope by using the electron donating group and electron withdrawing group. And the electron donating groups are giving the better yield than that of the electron withdrawing groups. And again, we have done the control experiment. Either we have to find out this NH is responsible for the reaction, because we did not get any crystal structure of this reaction. So we have proved the reactions with the various control experiments. So we have alkylated the nitrogen so that the reaction cannot be possible with the nitrogen. And here we are getting the compound with the open chain alkylated. And with the D2O experiment, here we have found that there is 100% of D2O exchange with this proton and the NH proton. That is proved through this NMR. So we have proposed this mechanism for this reaction. Here the palladium is coordinated with this nitrogen, and then it coordinated with the carbon formation of this cyclic structure, and then coordinated with alkene, and then elimination, reductive elimination, and formation of this final compound. Now, after that, we have used this naphthalamide moiety. Naphthalamide is a very good biologically active moiety. It is as used in the various clinical trials of amonafide and matronafide, but it was failed because of the increase in the toxicity at phase 1, phase 2. So we have tried this alkalination of this naphthalamide, and only 2 reports are noted with in the literature for the CH activation of this naphthalamide moiety. One with with OH coming at this position, this phenyl ring, and because this is favorable for the reaction formation of the 6-member ring with the ruthenium catalyst. The second is using the acrylonitrile and the formation of this product with the CH activation and this carbon. Otherwise, the reactions at this carbon is very difficult, and most of the reactions are reported at this carbon only. And we have now tried these reactions with alkylation and at this carbon using this amide group. And here we are getting the good yields of compound. So here, this is the substrate scope. The only limitation of this reaction is that we are not getting the single alkylated compound. Even we are getting the dialkylated compound, and we are getting both single and dialkylated, and specifically dialkylated compounds can be formed here. So with this, we have also increased the substrate scope. We are using the electron donating group and electron withdrawing group, and the electron donating groups are getting the good yields of compound. And in the electron withdrawing group, the alkylation, single alkylation cannot be taken as this ring. It is taken on the other ring. This is because this nitro is electron withdrawing group, so it decreases the electron density of on this ring. But the electron density is more in this ring, so the first reaction taken place at this position. That is proved with the proton NMR. So with this, we have done the mechanistic studies, the D2O experiments we have done, and here found out that 49% of D2O exchange is there. And when it is monoalkylated, the second proton is deuterated with the 44%. And then we have to find out if this amide group is responsible for the reaction. So we have convert this amide group with the anhydride, and we did not get any compound, any final compound. And with this, instead of anhydride, we are only use the saturated carbon, and still we are not detected any product. With the mixture of methoxy and nitro, the electron donating a withdrawing group, and we are getting the good yields and the faster reaction with the methoxy than that of the nitro. So next we have used the phenethromidazole, that is also biologically active moiety used in various anti-cancer agents. So in the literature that reported with the alkyne and the phenyl. So there is no report with the alkene. So we have used the alkene for this reaction, and we are getting this type of compound. So the optimization reactions are done with the various oxidant. We have used the various additive and solvents, and best yields we are getting with 88% with the silver acetate, silver antimonate, and the DMF. So we have used this reaction condition, this optimized reaction condition for the further reaction, and the substrate scope has been done with this reaction. And here we have used the electron withdrawing group and electron donating groups for this reaction, and getting the final compound. Here we have used the ortho derivatives also, so that only one type of alkylation can be taken place, and in meta and para, the dialkylation are there. But we have lucky to separate the monoalkylated, and then can be performed with the reaction with the dialkylated. So here again we have increases substrate scope with the other other functional groups on this ring. Here, in the case of this vinyl sulfonane and vinyl cyanide, we are getting only the mono, this ring form only. There is no formation of the alkylation and this ring. And in the case of styrene, this open chain is formed. There is no ring formation. Okay. So we have used the mechanical investigation of this reaction. So we have changed this ring with the methyl. So we have to check if there is a reaction with the sp2 hybridization, or there will be reaction can be possible with the sp3 hybridization. So we are not getting any product. So that means this ring is responsible for the reaction. So next we have done the D2O experiment. So here this ring having the hydrogen getting the 65% EA, so 65% exchange, and this carbon hydrogen is also exchange with the 25%, but we did not get any product with this substitution at these positions. And here, when this cyclization is there, then we are getting the 60% hydrogen exchange on this. That means the second, the acrylate is added on this carbon only. So here we have identified the intermediate. So we have tried to separate out the open chain, and then the cyclized product. So this is cyclized product is formed, and then the reactions with the acrylate, we are getting the substitutions at this carbon only. So from this, we have proposed the mechanism. So here we have the substrate. So where first it forms the ruthenium complex, and the coordinate with the alkene, and with this we are getting this open chain structure. We have also separated this structure, and then as a Michael reaction, and then it is this product 4 is formed, and then this product is then in the presence of ruthenium catalyst coordinate with the alkene, and then formation of this final product after the reductive elimination. So last, I have used the conjugation of indole and quinozoline, and then alkylination of indole with the help of quinozoline. So various optimization reactions conditions has been done. Just 1 minute only. So various substrate scope we have used for this reaction. I will not go through the substrate scope. The mechanism is also same. The deuterium in labeling experiment has been done where the 55% has been, this carbon is deuterated. So that is prone to the reaction. So at last, in the conclusion, so I have used the directing group for the various conjugates for the reactions where we are getting the atom and step economic reaction. Regioselectivity we have achieved. Wide substrate we have done the functional group tolerance there for the reactions, and the amide that is used as a weak directing group that directs the reaction for the final product. And now I would like to acknowledge my collaborators and my research scholars. And this work has been done by my two students. One, this is Dinesh Singla, that is, he is now doing job in Jubilant Campuses, and the Rekha Thakur, he is seeking for the postdoc, he got the opportunity from the Imperial College of London. And I would like to thank SERB, DST, CSIR, DRDO, and Thapar University. Thank you.
Thank you, Professor Kamaldeep Paul. One quick question from the audience. Yeah.
Sir, nice work. Sir, I want to ask, you have taken all the activated coupling partners, olefin coupling. What about the non-activated such as styrene and acrylamides?
Acrylamides, styrenes, I have used, but styrenes is the somewhat less reactive. So sometimes it not working in some reactions. No, I have tried that, but reactions are not happened with the styrene. Yeah.
Okay. Any other question? Okay, if there is no more question, join me to thank Professor Kamaldeep for the nice presentation.
I request Dr. Vijayalakshmi to felicitate Professor Kamaldeep Paul as a token of her appreciation.
Better if someone else will come. I have done project with
Uh, Now I would like to invite Dr. Gauri Shankar Roy Mahapatra to felicitate our distinguished chairs, Dr. Namrata Rastogi and Dr. Vijay Lakshmi, as a token of our gratitude for their invaluable contribution on this session.
I would like to make a special mention here that Dr. Gauri Shankar Roy Mahapatra is also a representative of Indian Photobiology Society, and we are thankful to the IPS Society for their generous support for this SIDD 2025 in the form of 5 oral presentations. Thank you, IPS. Thank you, Dr. Gauri Shankar Roy Mahapatra. Thank you.
It is the India before and after mutiny. I think writing this essay, Professor Chandra get some money to visit all over the Europe. So it is a nice book for the chemistry students.
So ultimately, art and science, they're complementary to each other. Thank you everyone. Please join us for dinner. And tomorrow, it would be great if you all kindly join for the valedictory and other. We have two technical sessions as well. So I would like to request everyone, all the to kindly come and tomorrow also. Please take it as important as all the sessions yesterday and today. Thank you.